Per capire cosa significa far parte di Parent Project, vi chiediamo di ricordare e immaginare il mondo prima di Parent Project. Eravamo soli, senza nessuna rassicurazione e senza prospettive future. Avevamo affrontato il momento più difficile della nostra vita. In così poco tempo, una sola breve parola era riuscita a stravolgere tutta quella vita che avevamo da sempre programmato. I viaggi che sognavamo di fare, le partite di calcio in cui avremmo ammirato il nostro bambino, le 1000 avventure che avremmo potuto vivere insieme. Prima di sentirci dire sì, parliamo di distrofia muscolare di Duchenne, sognavamo e programmavamo semplicemente il futuro di tutta la nostra famiglia. Abbiamo pianificato, siamo caduti e abbiamo provato a ricominciare. Era il tempo in cui ci dicevano che non c'era nulla che avremmo potuto fare. Non c'era una cura, non c'erano informazioni, ma stentavamo a crederci. Il nostro bambino era lì, era bellissimo, nulla sembrava diverso e non potevamo rassegnarci a questo. L'unica cosa che potevamo fare era rimboccarci le maniche e lottare. E così lo abbiamo fatto. Abbiamo cercato altri pazienti, conosciuto chi viveva la nostra stessa ed identica situazione. Quello era il tempo in cui la forza e l'intuizione ci hanno portato a fondare Parent Project e così abbiamo ricominciato a vivere. È stato il tempo in cui abbiamo seminato, in cui abbiamo coinvolto tante famiglie, medici, istituzioni, ricercatori. È stato il tempo in cui abbiamo capito che avremmo dovuto imparare a conoscere la patologia, che avremmo dovuto spiegarla ai nostri figli, che avremmo dovuto vivere di speranza e che questa sarebbe diventata l'unico punto fermo della nostra vita. Ma è stato soprattutto il momento in cui abbiamo capito che prima o poi una cura probabilmente sarebbe arrivata. Abbiamo lottato, perdendo e vincendo. Le vittorie ci hanno spinto sempre più a fare di più e a cercare altri medici, altri ricercatori, altri progetti. Ma sono le sconfitte che ci hanno lasciato il messaggio più importante. Ci hanno fatto capire che per poter vivere la nostra nuova vita dovevamo raggiungere un equilibrio tra il nostro presente e tutto ciò che sarebbe arrivato dopo, combattendo le nostre paure. È stato difficile ed inevitabile, ma ci siamo riusciti. Quello è stato il momento in cui la nostra speranza ha incontrato una certezza. Abbiamo capito che in tutto il mondo altri genitori stavano lavorando per creare un futuro per i propri figli. Gli ingranaggi sono diventati sempre più numerosi ed erano perfetti, sempre più perfetti. La ricerca andava avanti e quello è stato il tempo in cui abbiamo iniziato a raccogliere. È arrivato il primo trial, le strategie di presa in carico miglioravano e noi ci prendevamo cura dei nostri figli. Sapevamo finalmente mettere in pratica alcuni accorgimenti che ci permettevano di guadagnare tempo da trascorrere con i nostri ragazzi. Loro continuavano a crescere, il nostro tempo era sempre più pieno, ma le opportunità aumentavano. Abbiamo raccolto, abbiamo imparato, abbiamo continuato a costruire, ma anche a cadere. E tutto questo continuiamo a farlo ogni giorno. Questo è il momento in cui dobbiamo anche saper guardare indietro e unire tutti i punti, perché tutto quello che abbiamo vissuto ci ha portati qui oggi ed ha trasformato la grande intuizione in tutto quello che abbiamo ora. La linea del tempo che abbiamo tracciato insieme in questi 22 anni non ha ancora un punto di chiusura. C'è ancora tanto che possiamo e che dobbiamo fare insieme, perché i nostri figli sono qui, con una vita diversa da quella che avevamo programmato, con un futuro che non conosciamo ancora, ma con tantissime opportunità. Tutto quello che abbiamo costruito, tutto quello che abbiamo demolito, tutto quello che abbiamo conquistato non sarebbe stato possibile se non avessimo unito le nostre forze. Ci siamo immersi nei nostri sogni, in quei sogni che ci sembravano e che ci sembrano così nitidi e chiari. Ci svegliamo spesso, solo perché molto spesso arriva la pioggia e tutto è più difficile sotto la pioggia. Ci siamo nascosti e coperti, ma sempre insieme. E sono proprio queste speranze, queste convinzioni e queste certezze che ci hanno trovato tutti uniti ad affrontare la vita. La nostra forza è il motore di tutto quello che stiamo progettando. La nostra partecipazione fa la differenza e continuerà sempre a fare la differenza. Partecipare significa aggiungere quel pezzetto in più al mondo che vogliamo costruire. Partecipare significa, nel bene e nel male, dare un contributo costruttivo, sempre consapevoli di voler raggiungere tutti lo stesso obiettivo. Siamo davvero tantissimi, da ogni parte del mondo, perché cambiare il mondo è davvero un gioco di squadra e le persone insieme hanno il potere di cambiare il corso delle cose.
Grazie, grazie davvero di cuore a tutti voi per essere qui.
Thank you very much. Thank you all for being here today. You have come from all over the world, and it is indeed very hard for me to say anything further to what we've heard so far. Anyway, today and tomorrow we're going to have intense working days. We're going to listen to people providing us with new data. However, what a parent project really wants to highlight is the emotions that will manage to convey, of course, with the help of our physicians, of our researchers, who are always on our side and have been on our side for many years. Of course, we've also made mistakes, but we've got out stronger from such mistakes. So we need to move forward, and we'll have to stand united because, as it has been said, being together is what can truly make us stronger. And then, of course, we need more and more parents to come join the association because it is indeed of paramount importance. Actually, it's our moral duty to help our sons and other parents' sons. Hopefully, this pathway will be as short as possible because there will be a cure, hopefully, one day. Anyway, I wish to thank all those who have contributed to making this conference possible. You will find several documents in your file, and we owe all of this to the work of many people. By the way, this is a sort of a handbook for families that has been the result of hard work carried out by many people, starting from Ms. Mazzone. So these are important aids for us all, namely allowing us to find that balance that we desperately need in our daily life. So let me start by giving you the very first piece of news regarding this conference. That is a poll, a survey. Actually, you will be telling us what you think. I mean, in real time, what you think of this conference. I'm sure you're now going to see an address. Actually, visit the Parent Project website even now, okay? Just click on the conference banner with this catchphrase, changing the world is teamwork. And there you'll find a link where you're going to find the first 4 questions. Okay, I think you can find the address to click on. Okay, just go visit parentproject.it, then you'll click on the banner and find the link to this poll with the first 4 questions. I can't read them from here, but anyway, Why are you here? Are you here as, relatives or researchers or whatever? Can anybody help us through? Okay, so www.parentproject.it, then there's a banner. Just, click there and you'll get to the part of the website that we need. Don't Google it because you won't go anywhere. So question number one, why are you here? I mean, are you here as a relative or a researcher or what? So your answers will be shown online in real time. So that was question number one. Are you here as families, associations, researchers, or clinicians, companies, other professional figures somehow connected to DMD or BMD? And of course, others.
C'è un problema tecnico. Va bene, come in tutte le buone famiglie c'è un problema tecnico.
Well, it's always like this, I'm afraid. Technical problems arise quite unexpectedly. So anyway, do provide your answers because maybe at a second stage we'll be able to read them through. Oh, okay. Apparently, we are actually watching the questions, but Francesca Ceradini is coming up and she'll be taking the floor soon.
Eh sì, però questa è la seconda, l'altra domanda, giusto? Siamo andati avanti. Va bene, nel frattempo il meccanismo si metterà.
Okay. Let's take a few more minutes for such innovation to get started. You know, unless you try, you'll never get anywhere. So this is another question. What do you think the best possible strategies to cope with DMD and BMD may be? Money, teachings, and so on and so forth. Okay, let's move forward. Let's move forward in the meantime. And let me therefore give the floor to Francesca Ceradini. I'm very glad to give her the floor. She is the person in charge for our association. She's in charge for the scientific section. So we are running a bit late, but hopefully we'll be able to catch up. So Francesca Ceradini, you have the floor.
Grazie Luca. Thank you, Luca. Good morning, everyone. I'll begin by giving you an overview of the therapeutic strategies being developed for DMD and BMD throughout the world that are going to be illustrated during the conference itself in greater detail. Since we're talking about change this year, this is the theme of the conference this year, I decided to develop a timeline which shows how or the extent to which research has changed on DMD and BMD. The first description of Duchenne was made 150 years ago by Guillaume Duchenne, a French neurologist. But the important event happened in 1987 when the gene of dystrophin was discovered, and this paved the way and opened the doors to scientific research that began to develop towards the 90s. And it is in the 90s that in the States the parent project was created by Pat Furlong, who's here today, and 2 years later Filippo Bucella created the same parent project initiative for Italy. Then it took all the way up to 2004 to witness the first trial on DMD that then led in 2014 to the approval of the first drug in Europe for nonsense mutations. And in 2008, the first trial started for exon skipping, exon 51 skipping. That followed a whole host of trials that led to the development and the approval of the first drug in the U.S. For exon DIS 51. And then last year, gene therapy trials started out in the States. So what's the situation today, now that 30 years have elapsed since the beginning of research, and that 40 trials are taking place around the world, of which 10 are in Italy on the different strategies. And there are over 10 patient associations that are part of the network. And this is quite a significant change, which is the result of teamwork, definitely, between clinicians, researchers, patient associations, and pharmaceutical companies. Let's begin with dystrophin. Dystrophin is a protein, a fundamental protein when it comes to muscle function, and it's found right in the internal face of the membrane of muscle fibers. It's anchored there, and it has different functions. The first is mechanical. It functions as a shock absorber of the muscle during muscle contraction. And also it organizes the assembly of their proteins, and it also sends signals. What happens is that whenever there's a mutation in the dystrophin gene, and there can be many, this can lead to a complete absence of the gene, as is a case of DMD, or reduced levels of this protein in BMD and Becker's muscular dystrophy. In both cases, anyway, there is the triggering of a cascade of events, molecular and cellular events that lead to damage of muscle tissue and its degeneration. And here I'm going to show you what these events are. First of all, the total absence of dystrophin, as a result of which cells cannot play the role of shock absorbers to muscle contraction, and this results in weakness and fragility of muscles. What happens to the cells is that the membrane becomes more and more frail, that are micro fissures, small cracks that are created, and substances come in like calcium ions, which accumulate in the cell, and the cell undergoes necrosis, and they kind of burst, and an inflammatory process sets in. At that time, of course, there is an immune response that happens because the immune system recognizes these dead cell debris as necrotic tissue. And the necrosis does not, of course, provide for the adequate repair of the tissue on the part of stem cells, and the muscle tissue is replaced with fibrotic and fatty tissue. Researchers throughout the world started to devise and develop strategies that could actually affect and change the different steps in this cascade process. One was, of course, to restore dystrophin with a whole set of strategies. Then they tried to reinforce the muscle, and they tried to limit the accumulation of calcium, and of course reduce inflammation and fibrosis. It's really important to remember that there are also respiratory muscles in the heart that are affected, so there are several molecules that focus on these specific muscles. Amongst the material that Luca mentioned, we have this graphic information pamphlet that we organized for this event, which explains the different strategies that I will illustrate to you in a very direct manner to inform people. So, restoring dystrophin. Let's start with a gene therapy is a strategy that many know. It's based on the concept that if the gene is not working, the idea is to provide a healthy gene, the so-called therapeutic gene to the cell, which is introduced into the cell with the virus, which is manipulated in such a way that it can penetrate the target tissue and not be detrimental to the body. What's the problem with dystrophin, however? Dystrophin is the largest gene that we have in our body, as you know, and it is not able to be encapsulated into these viruses like the AAV virus, which is used for gene therapy. So what researchers did was to try to devise smaller constructs called mini and microdystrophin, which, although they are smaller, are still viable. And at the end of 2017, 3 different trials were undertaken in the U.S. with these strategies, which will then be illustrated shortly. Another strategy is this. Instead of providing a healthy gene, stem cells are provided which contain all of dystrophin and therefore cell therapy. The first consists in taking stem cells from a healthy donor, and of course, in the lab, they are then introduced into the patient. This strategy proved to be not very effective, I'm afraid. Another method is to harvest stem cells from the patient himself. These stem cells are genetically manipulated so that they can express dystrophin and reinfuse it in the patient. And Giulio Costa will describe this process shortly. Let me get a drink of water now. Then there are other methods to restore dystrophin. One is to correct the damage and the mutation directly. So either work with the DNA, and then gene editing is part of that strategy, or work on the mRNA molecule, which translates into the final and functional product of the protein, like exon skipping that we know quite well. And we can also have an impact on the translation process through nonsense mutations. Let's begin with genome editing, which is an extremely innovative technique. I imagine most of you have heard about this because there's a lot of talk about this in genetic and even non-genetic disorders. This is the last frontier of genetic engineering because it can modify directly the mutation or the DNA in a very specific and definitive way, which could actually create some obstacles. But anyway, how does it work? Well, there are different systems of genome editing, but the most famous right now is CRISPR-Cas9, which is based on 2 elements. One is a guide molecule, which indicates the exact spot where Cas9 has to act. Cas9 is an enzyme that cuts the DNA. It's important to remember that this technique would modify the situation once and for all because it acts right on the DNA. But trials are just in the preclinical phase. Only dog and rat cells have been used for these experiments. Let's move on to exon skipping. That shows you the 79 exons of the dystrophin gene. These are building blocks. Whenever there are deletions that change this pattern, dystrophin ceases to be produced. This is an example, the deletion of exon 4950, for example. I don't know if you can see this, but because of this deletion, the building blocks are wrong. So we use short RNA molecules that lead to the skipping of another exon, like 51, exon 51, which restores the reading pattern, although it's for a shorter molecule. There are several molecules being developed for this purpose. Eteplirsen, for example, was approved in the US, but not in Europe. So there are different molecules for the skipping of different exons, and they're going to be shown to you later on. Let me show you the last process for the restoring of dystrophin through nonsense mutations. Reading of the gene, is like this. It starts with the reading of a gene. It started with a capital letter and it ended with a period. Now, in some cases, mutations are such that the sentence is almost interrupted. So the dystrophin is not viable. It's degraded. It doesn't work. And Translarna, this is a commercial name of this molecule, makes the mutation continue, or the sentence, if you like, continue. There are clinical trials in different phases that are going on, even in the post-approval phase. Let's now move on to the next step. Instead of trying to restore dystrophin, let's try to reinforce the muscle. As I said before, dystrophin is like a shock absorber for cells. So when it is absent, it triggers many events downstream. And some strategies are based on the idea of compensating, offsetting the absence of dystrophin with other proteins. So one is utrophin, which is almost identical to dystrophin, but it's that protein that we produce in the fetal phase, and at birth it's replaced by dystrophin. Now, there are molecules that can induce the production of utrophin. And another strategy is to use surrogate gene editing methods that reinforce the dystroglycan protein complex. In this case, we have the development of euzitramid, but the trial was actually interrupted. It was stopped last summer because it didn't show any clinical benefits. But instead, the trial using GALG2 as a surrogate gene therapy is continuing. Another way is to increase muscle mass. Our muscle cells produce a protein called myostatin, which hinders the uncontrolled growth of muscle. The idea of researchers here is to inhibit the action of myostatin in order to have a little bit more muscle growth, muscle development. In this case, again, there are two trials underway. There were actually, because one was stopped in September because it didn't show any clinical benefits, but one is continuing instead. Then another strategy is to improve muscle metabolism, again with the view to reinforcing muscles. In our cells, we have mitochondria, which are small organelles that are like the energy power plant of cells, and they are fundamental for the production of energy. It's so important for muscles. In dystrophy muscles, we have a functional deficiency of mitochondria. So one strategy is to try to develop molecules that can increase the function of mitochondria and their number as well. And here we have other molecules that will be presented during the conference later on. Then we have a calcium accumulation, which brings about necrosis in the cells. And as I explained, this triggers the inflammatory process. And here again, we have two molecules that are being studied. Rumeparide, that controls the calcium channels and that limits the accumulation of calcium, and then P188NF, which is a molecule that seals the microfissures or microcracks in the cell membrane that are created with the damage. And finally, of course, many molecules that combat and that reduce inflammation and fibrosis, because as I explained to you briefly earlier on, in a healthy muscle, when you've got damage happening, there is an inflammatory process that happens, but we also have regeneration that happens through the intervention of the stem cells of the muscle. But in a dystrophic muscle, when the damage appears, and by the way, it's a chronic type damage because cells just don't have dystrophin. So the stem cells that set in do not contain dystrophin, and in a dystrophic environment, well, there is no enabling of the regeneration, and so the muscle tissue is replaced by adipose and fibrotic tissue that creates the scar, and the muscle no longer works. Let me conclude by saying that this year also we've updated our brochure of trials. It's becoming historic, and we are circulating it more and more. And finally, this is a picture of the scientific office of Parent Project. Let's now begin with the first session, Therapeutic Strategies for the Restoring or Restoration of Dystrophin, Part 1. I'd like to call upon Filippo Buccella and Francesco Montone, who are the chairs and moderators of this session.
Buongiorno a tutti.
Good morning, everyone. I'm so happy to see a lot of you and to see you here every year. And you're more and more numerous. I really do hope that this new conference continues to usher in a lot of groundbreaking news, as has happened in previous editions. This is the 17th time that we meet. So thank you for coming back and for making it so wonderful every year. Thank you to the parents, to the pharmaceutical industry, to the clinicians and researchers. I'd like to give the floor to Professor Mercuri, who's sitting right next to me, and who I'm sure would like to greet you all. I was introduced as Professor Mercuri, and I don't quite know whether this is good or this is bad. It's actually an excellent thing. It's really a great pleasure for me to be here. I know that you did it on purpose, I know, to introduce me as professor. It's a pleasure to have been mixed up with Eugenio. The program this year is once again extremely rich. I'm not sure that you're aware that the findings that are presented here are often complete novelties in the sense that they are completely unprecedented and unpublished. So I'll be really interested to hear about findings that are introduced for the very first time here. Our program will begin with efforts. I'm sorry, I don't speak very well. First of all, because it takes me a long time to wake up in the morning, and also because I've been living in England for 26 years. So for me, English is a lot easier than Italian. So the first session will begin with an overview of gene therapy. The various speakers will have about 15 minutes. The first is Michael Things from Pfizer.
Presentation.
se vuoi stare qua, stiamo qui o scendiamo? No, stiamo qui su.
Again in Rome to this wonderful meeting. So I want to talk to you this morning about our gene therapy program, which has been running since the beginning of last year, and give you a little bit of background to the technologies that are involved and some of the challenges that have been overcome to get to this point, and some of the challenges that lie ahead. I present a safe harbor disclaimer. So, what is gene therapy? Francesca gave you a nice introduction to the idea that this is all about providing a replacement gene for a faulty one. It's clearly most applicable in those diseases where there is a single gene mutation or a single gene disruption. And it's an approach that may provide a solution to improving symptoms or to correcting a disease towards health. Our hope is that this is a one-time intervention with long-term benefit, but it will clearly require many years of study in the clinic to establish just how long any potential benefit that is achieved is maintained. So we talk about vectors, which are the vehicles to deliver a gene therapy. They're usually based on the protein capsule or capsid of a non-replicating virus that allows us to introduce the gene into tissues. The virus that is most often used in gene therapy, and certainly I think is the basis of most of the projects you hear about today, is the adeno-associated virus that's been selected primarily because of the efficiency with which it's able to introduce a new gene into tissues, but also because it induces a minimal inflammatory and immunological response that enables this to be delivered at fairly high doses in a safe manner. The one downside to this virus is that the carrying capacity of the genome is limited to around 4.7 kilobases. That's 1,000 base pairs. You will recall that the DMD gene is the largest in the human genome at 2.3 million base pairs and is located on the X chromosome. That's transcribed into RNA, which is a much smaller molecule, but still around 14,000 base pairs in this characteristic exon structure that you've seen many times before. And that is then translated into a protein with multiple repeating parts of the protein. We call them spectrin-like repeats, but they're essentially little bundles of 3 coils of peptide all together repeating. And it's found in many proteins that are related to structural, the cytoskeleton, so the structural components of the cell. So this offers some real challenges for us. How do we get that huge piece of DNA and that huge protein expressed by gene therapy? And I'll talk to you a little bit about how that was achieved. We clearly need to deliver these viruses to muscle, which constitutes 40% of the body mass, and that's going to require really high doses relative to other tissues which have been explored in gene therapy, like the eye, or direct introduction into the central nervous system, or even to the liver. And you can see that compared with the one approved gene therapy in the US, which is a directed injection into the eye, the doses required are 10,000 times larger in order to achieve delivery in muscles. We have challenges in selecting the right subtype of virus, and each of the companies has gone about that in a slightly different way. We have challenges in terms of the immunology, the immune response to virus, and our ability to dose patients who may have been exposed to it before in the environment or through therapy. There is also a consideration around the possibility that introducing the expression of dystrophin when it hasn't been there before could induce an immune response. We also have a challenge in our understanding of what levels of dystrophin expression need to be achieved to translate into positive improvements in function, and that's an issue that the whole field is grappling with. We have ideas, but there's nothing with certainty that we can point to. However, this is potentially a one-time therapy that can make a big impact for patients and potentially for patients of all mutation types. So Francesca nicely showed a timeline of the field in general. I just want to talk a little bit about the timeline in terms of our progress towards the introduction of gene therapy here. So 1987, the identification of the gene, some mutations in Becker's muscular dystrophy suggesting that very small parts of dystrophin expression can be adequate to compensate for function. And then the introduction or the identification of synthetic constructs of dystrophin that could potentially replace function. AAV as a virus was first identified in the 60s, but was identified as a potential gene therapy vector in 1984. And these came together around 18 years ago with the publication of a demonstration of real benefit in mouse models. We've since seen the demonstration in the golden retriever model of muscle transduction and of efficacy in terms of preserving function, and even seen that function being sustained, functional benefit being sustained out for many years. I think the longest we've seen is around 13 years in a dog. We've had to undergo toxicology review of our constructs, and the first human AAV trial in the mid-2000s was completed, delivered locally, and was able to show evidence of transduction. But it was very local, and the doses were rather low in that study. And now we are here with as you know, 3 active clinical programs in the gene therapy arena. So our program focused on one particular miniaturized dystrophin, which, among other things, includes the Hinge3 region, but based on this knowledge about Becker's proteins. We've chosen to use the AAV9 serotype because in our hands it delivers more efficiently to muscle than AAV8. And we're using a muscle-specific promoter. The gene has been optimized in terms of the precise sequence for maximum expression in muscle. We introduced our gene into a rat model of Duchenne. We're able to show a very nice dose response with increasing expression of dystrophin, here shown in green, both in skeletal muscle and in the heart, achieving a good proportion of fiber transduction. And in that model, we also showed a very clear dose response in terms of the reduction of creatine kinase and in improvements in sustained muscle strength. So at that point, we consulted widely with experts in the field and with many patient advocacy groups, including Parent Project Onlus, to work out the best way to get the scientific information we need to progress the program and bring it towards a wider population of patients, but also in a way that was acceptable to patients going into it. And we've learned an awful lot from interactions with groups such as this, and continue to provide support in targeted, practical ways to many of these organizations that align with our mission, which is to bring breakthroughs that really impact people's lives, change people's lives. So thank you very much for keeping us generally informed. This is the study that's ongoing. We've designed a study that requires three muscle biopsies at baseline after 2 months of treatment, and then again after 12 months of treatment, and then boys will be followed up for 5 years for safety. The participants are between the ages of 5 and 12, and they need to be negative for neutralizing antibodies to AAV9. We have to stagger the cohorts, so the individual patients. So we can dose one, and then we have to wait a while, watch for any safety events before we can dose the next one. But we're also measuring in this study function, and we're looking at the quality of muscle via MRI and measuring activity at home in the real world with digital devices. So we have 3 sites active, all in the US. We've screened 11 patients. We've had only 3 patients fail. Only one of those was actually because of a neutralizing antibody. And we've enrolled 6 boys and treated 6 boys to this point. So far, the study is progressing very well from a safety point of view, and there's nothing emerging that will get in the way of us moving the project forward. And we hope to be able to share some initial data from our biopsies, which are being pooled together so that we can look at them all at one time in a blinded way later in the year, around the middle of the year. So with that, I'll stop and ask whether you have any questions. Thank you.
Penso possiamo tenere le domande tutte quante alla fine.
I think questions can be asked at the end of the session until you have something to ask that you can't, wait for. We are going to have a Q&A session at the end, so maybe you'll want to write them down, okay?
The speakers together, so you will be asked to come back here. Continuo. Il prossimo, rimaniamo nel campo della gene therapy.
Okay, let's stick to gene therapy, of course, and we're going to give the floor to Carl Morris from Solid Biosciences. He will be telling us about the progress achieved with SGT-001.
Very much for having me attend and fantastic meeting, and feel privileged to come here and be part of it. So today I would like to talk about our SGT-001 program, which is our lead gene therapy program, looking at gene transfer of microdystrophin in Duchenne. Similarly to Mike, have a forward-looking statement. So first I'd like to talk a little bit about Solid and what Solid is. Solid was founded by parents of a boy with Duchenne, seen in the picture here, Atani. Milan and Annie decided to start a company to go after Duchenne. And so since that time, we're now a company of about 140. I was number 4, and I've been there about 3.5 years ago. So we've grown tremendously over the last 3 years, and we're fully dedicated and focused on Duchenne and nothing but Duchenne. Our lead candidate is this sort of differentiated gene transfer program that I'll talk about. And as many are aware, we did have a recent press release around some of our data, and I'll talk a little bit more about that. Particularly, one key aspect that always goes a little bit unnoticed is the manufacturing of drug product. It's critical, given what Mike Binks was just talking about. We have to deliver high doses to large patients and we have to get a lot of drug produced. So we have focused a tremendous amount of time and resource and energy on getting to a point where we're very comfortable with our manufacturing process. So we have a real 360-degree approach, and so we're not just doing gene therapy. We do have other approaches that we're looking at and exploring. As Francesca pointed out, there are a lot of different pathologies and issues that are associated with the loss of dystrophin, and so we are focused on a number of those. Also importantly, we're looking at assisted devices in a collaboration with a company called Seismic in California that we hope to help augment function and improve the patient's quality of life as well. And then given that we have a 100% dedication to Duchenne, we also focus a lot of our time and energy on biomarkers and evaluation of alternative endpoints to help improve the overall clinical trial setting and approaches being taken to identify meaningful therapies for Duchenne. So I'll now turn directly to our gene transfer program. And given that there's now been 2 introductions to gene therapy, I'll make it short, but I still want to sort of emphasize some of those points. So what is gene transfer? So there's 3 components that are critical to deliver a corrective therapy for Duchenne. So you need the gene, so which for, in our case, microdystrophin. You need a promoter that will control expression only where you want it. So we have to deliver to the muscles, the heart, diaphragm, and so we want to make sure that we have a strong promoter that will produce significant amounts of expression of your gene, and therefore the protein. But we don't want it going to producing in the liver or other tissues where dystrophin is generally not made. And finally, the vector is critical because that's the delivery vehicle. That's what gets you to the muscle, gets you to the tissue in high levels. So we put all of that together, and then that's what's administered to the patient as a package. So for us, we have the 3 components that I just talked about. The first one is the SGT-001 microdystrophin construct, the DNA that we produced, and I'll go through that in a little bit as to how we selected the different components of our product. The second one is a muscle-specific promoter that's based on Jeff Chamberlain and Steve Hauschka's work from the University of Washington, where they looked at creatine kinase promoters, and so we use one called CK8, which is one more than CK7, so it just numbering systems, but it's just a creatine kinase muscle-specific promoter. And finally, we also use AAV9, like Pfizer, because we did consider that to be in our hands the best for muscle-based diseases. So up the top here, you can see that the full-length dystrophin protein even looks big. It's a huge protein, and it has a lot of the spectrin-like repeats that was communicated. There are various hinge regions. The issue is that the DNA of that does not fit into the AAV. The packaging limit of the AAV, as said before, is about 5,000 base pairs. And this does not fit. It's 14,000 base pairs. So Jeff Chamberlain spent a lot of time building different constructs and identifying functional small proteins that would produce a good level of force in the mice. So we took his constructs, and then Dongchen Duan started looking at a specific region called the nNOS binding domain. It's highlighted in R16 and R17. So the nNOS binding domain, we can think it will provide a little bit more functionality by calling in and allowing sort of localizing of nNOS, which is neuronal nitric oxide synthase. So basically it helps with the functional ischemia. So it allows a little bit more vasodilation, and so if you have dystrophin being expressed, it holds the nNOS there. You can get vasodilation and get a bit more oxygen flow to the muscle. That was the goal, and that's how we designed our construct. excuse me, and that was from both Jeff Chamberlain and Dongshen Duan, and really based on years and years of research. So you put that all together, and when you dose a mouse, you can see in the heart, we're looking at cross-sections here of both the heart and the quadriceps, and you can see as you go up in dose, and you can get more microdystrophin expressed in the heart, in the muscle tissue. And as mentioned, we want it in the heart. We want high levels of expression in the target tissue, the muscles, but we don't want it in the liver. So this is work from a dog study, so a golden retriever muscular dystrophy model that we worked on. And you can see that we're getting good expression of the microdystrophin as shown on the left-hand side in both the diaphragm and the quad muscle, the vastus lateralis. And while if you look on the right-hand side, there's no expression observed in the liver. So when you put it all together, what we're trying to do is get a high level of the delivery vehicles, the vectors, into the muscle tissue. So we can then look at the muscle tissues and take a small biopsy, or in the mice, sort of take the muscle out, and we can look at the dose response of how the vector is getting into the muscle and staying there. So we call that biodistribution. And you can see going from black to the gray bars as you go up in dose in all three tissues, independent of whether it's muscle or not, you see a dose escalation or increased dose, an increased level of vector genomes in that tissue. So now we know it's there, but is it functional? Is it doing something? So in the center, we look at target engagement, how much protein is being produced. And you can see from the Western blot that we're highlighting up the top, when you increase the dose level, you get increasing amounts of microdystrophin protein being produced. And down the bottom in that curve, it's sort of an S shape. And this is really just sort of driving a general dose response. If you start low, you have very low levels of microdystrophin being produced. But as you go up in dose, and very small levels, like 4-fold up in dose, you can start generating a lot more microdystrophin expression in the muscle tissue. Ultimately, it doesn't really matter how much protein you produce if it's not functional. So on the right-hand side, you see what functional changes are observed. And highlighting just force production from our dog study, you can see that as we go up in dose, we do see functional improvements in the muscles. So that, which is the most important thing at the end of the day. So talking about our gene therapy study and where we are in that terms. So first highlighting, we call it IGNITE DMD, and it's a phase 1/2 clinical study that's ongoing at the University of Florida. We have a single site right now. We have the ability to enroll both ambulatory patients, 4 to 11 years of age, and non-ambulatory patients, 12 and older, 12 to 17. And the way the study is designed to be a dose escalation study. So our first cohort was our starting dose. So safety is paramount, and the primary endpoint is safety and tolerability of the drug. So we initiated this study at a low dose to make sure that we went in reasonably safely and evaluated that before moving up to higher doses. So our first cohort started at 5e13, and we've dosed a couple of patients. But importantly, our study has this matched control group. So each time we enroll, we screen 2 patients and enroll 2 patients. And there's a randomization at the time of screening. One goes into the control group, which is a delayed treatment arm essentially, while the other patient goes into the active treatment and is followed. So once we've enrolled patients, after 12 months, and we get about 3 patients, 4 patients, we look at the DSMB, which is the Data Safety Monitoring Board. And what they do is they allow for us to dose escalate if they look at the safety profile of our product and look at all the patient records over the last year and determine whether there's enough sort of safe to proceed and escalate our dose. And so we've always planned to do a dose escalation study, escalation cohort, and we're in the process of now moving forward with that. I think our primary endpoints are also looking at microdystrophin expression, as well as the the usual suspects in terms of the evaluation of muscle function and other improvements. So as an update, so we dosed our first patient a year and 2 days ago, actually, in February 2018. To date, we've enrolled 6 patients, and all at the University of Florida. 3 have been put on the active group and have been dosed at 5E13, and 3 have been in the delayed treatment arm, so they're still ongoing with the study, but they have not been dosed as yet. The safety profile is good and hasn't changed over the last few, and they continue to remain on protocol and in the study on protocol. We have dosed 1 non-ambulant and 2 children. So recently, we performed preliminary analysis on we had the opportunity to look at our 3-month biopsies from the first 3 patients, and so we took that opportunity. And although we were expecting more, we did see signs of microdystrophin expression in all three biopsies. So I think that's important to note that we started at a very low dose based on our safety and safety being paramount, but we were able to detect microdystrophin in all 3 patients. And importantly, in 1 patient, we saw about 10% positive fibers via immunofluorescence, and we were able to detect the microdystrophin band on the Western blot of that patient. And then again, to provide a little bit more confidence, and we did see co-localization of the nNOS, the neuronal nitric oxide synthase, and beta-sarcoglycan, which is one of those dystroglycan complex members. So it means that the protein that we're producing is able to attract and sort of stabilize the proteins, the other proteins in the tissue. So as I mentioned before, manufacturing is critical, and we spend a lot of time going from the left, where in cell stacks, sort of just in Petri dishes. We're now move up into a 250-liter scale, where we're looking at it in suspension. So basically, we're producing the AAV in cells that are circulating in a big bioreactor. We have the ability now, even with the planned dose escalation, we have drug on hand to move forward with whatever dose is selected. And once we have patients enrolled, we'll be able to continue our dosing quite immediately. So we spent a lot of time on this, and really we've gone from this small scale, we're now up to a place where we're comfortable with our yields. Our yields are consistent, and we're getting good batches where we can produce material for multiple patients from each of those batches. So again, just to finish, we are moving forward with the planned dose escalation, and we're in the process of talking to the various agencies and regulatory bodies that are required to allow us to dose escalate. We're excited to continue with this program, and we look forward to hopefully next year presenting positive data from our next cohort. And with that, I'll end. Thank you.
Grazie Carl.
Thank you, Carl. Let us now move on to our next speaker, Rachel Potter from Sarepta. She will be telling us about gene therapy with microdystrophin, for, of course, DMD. Again, questions will all come in the end.
Thank you. I'd like to take this opportunity to thank everyone for letting me present this exciting data today. So we've heard a lot about gene therapy, so some of my introduction may seem a little redundant. I'll try to go through it a little bit quicker. Again, we have a disclaimer. Sarepta's therapies are not authorized in Europe, and this presentation is solely for educational and scientific purposes. So Sarepta Therapeutics has taken a multifaceted approach to target DMD. The goal of the company is to treat 100% of the DMD patients. And to do so, they've taken a 3-prong approach. So the first platform is the RNA-targeted therapies using exon skipping with PMOs and PPMOs. The second, which I'll focus on today, is gene therapy, specifically gene replacement using microdystrophin. And the third is using gene editing with CRISPR-Cas9 technology. So our scientific approach, again, is gene therapy, and it's relatively simple at its core. We've heard a lot about vector design and our transgene. However, our approach is to package the transgene in our modified AAV virus and deliver systemically to boys with DMD. The three important components are the vector, the promoter, and the transgene. And I'll highlight specifically on this slide our promoter. So we've heard that the promoter drives expression. Our promoter is a muscle and cardiac or heart-specific promoter, which has robust expression in the heart specifically.
Sorry, can I ask you to slow down a little? Some of my colleagues have difficulties in following you. Thank you.
I apologize. Okay, so a little bit of review. AAV is our delivery vehicle for gene therapy. And what AAV will do, it will target the tissues intended. So with our serotype, we're using something called RH74. RH74 is a unique serotype. It was isolated from a non-human primate model. The reason we've chosen this model and this serotype is because it has minimal immune response, low pre-existing immunity in the environment, and it specifically targets heart and skeletal muscle. So this AAV particle will find the muscle cell, the receptor, will move into the muscle cell. It will then move into the nucleus and undergo something called uncoating. Once uncoated, it will develop into a double-stranded DNA and produce functional protein. It's important to note that AAV does not integrate into the host genome, nor is it pathogenic, and it exists as something called a mini chromosome. So this mini chromosome allows for long-term expression of the protein of interest, and in this case, that's microdystrophin. Again, we've heard that full-length dystrophin does not fit into the packaging capacity of AAV. Therefore, we've taken an approach to modify it and shorten it into a microdystrophin. So moving into the preclinical design of this study using our muscle and cardiac specific promoter, which is referred to as the MHCK7 promoter. Again, this is a myosin heavy chain creatine kinase promoter that has been proven in our hands to optimally express in the heart and skeletal muscle. This study was a dose escalation study, so I will get into that in a few minutes. Our construct for microdystrophin, or our transgene, is shown here. So we include hinges 1, 2, and 4, which are important for flexibility of the construct. We also include spectrin repeats 1 through 3, which are extremely important for force production and optimal force generation, as well as spectrin repeat 24 and cysteine-rich regions. So the three important components that are shown are the vector, which have shown to have robust affinity for muscle and relatively low levels of pre-existing immunity. The promoter, again, is MHCK7, which is specific to heart and skeletal muscle, and our microdystrophin transgene, which is a functional version of dystrophin and contains spectrin-like repeats 2 and 3, which are important for force production. So this study was a dose escalation study, and I'll show some data regarding that in a few minutes, but at our clinical level dose, you can see in the third panel, we evaluated expression initially. So with AAV microdystrophin treatment, you can see that there is high, robust expression of the protein, and it is localizing to the cell membrane in skeletal muscle, heart, and diaphragm. We also looked at the complex called the dystrophin-associated protein complex, and we want to make sure that our microdys is able to link the inside of the cell to the outside of the cell. So we used a surrogate biomarker called beta-sarcoglycan to do so, and we saw that we have a beta-sarcoglycan assembly as well. So in the fourth panel at the bottom, you can see that there is expression and localization to the cell membrane of beta-sarcoglycan. So all of this together says that microdystrophin is expressing appropriately. And next we wanted to look at, is it functional? So the first thing we evaluated was CK levels in the animal models. So again, this is a preclinical study using MDX mice, so dystrophin deficient animal model. And when we have high CK levels, we know that this demonstrates muscle damage is occurring in the model. So CK is being released into the blood, and we have high CK levels. With AAV microdystrophin treatment, you can see that there is a significant decrease in CK levels. This demonstrates that the muscle is being protected and CK isn't being released into the muscle. Next, we looked at, in the middle panel, the specific force in TA. So this is looking at muscle force in vivo. And we saw with our dose escalation study, our mid and highest dose are shown here. And there's a significant increase in force production with both of those doses towards wild type levels. Finally, in the third panel, the eccentric contraction assay, simply put, is an assay to look at muscle damage, protection from damage. So these muscles undergo continuous stretch and release throughout the assay, and the fatigue or percent of force drop is tracked throughout the assay. So you can see the light purple is the untreated animals, and the top bar is the wild type animals. And then with treatment, both the middle dose and high dose demonstrate that there is significant protection from damage in these animals. So all this data together demonstrated that microdystrophin is expressing appropriately and at robust levels in skeletal muscle, heart, and diaphragm, and it is a functional version of dystrophin. We like to point out that the preclinical studies demonstrated 120% of cardiac expression. So the promoter, again, is highly expressing in the heart as well. To move on to a clinical update of the phase 1/2 trial for MHCK7 microdystrophin. The goal of study 1 was to validate the preclinical results. So as you can remember, the preclinical results showed robust expression in muscle and heart, as well as reductions in CK, and a favorable safety profile in animal models with no immunological responses. This is a figure that demonstrates the percent of dystrophin-positive fibers in the preclinical study. So with the 3 levels that were evaluated, the low dose, mid dose, and high dose are shown. And you can see that with dose escalation, the percent of dystrophin-positive fibers increases towards wild-type levels. And we move forward with looking at the gastrocnemius in our clinical trial. So the trial was an open-label trial design with 2 cohorts. I'm specifically focusing on cohort B, that includes 4 subjects, 4 to 7 years of age today, with confirmed DMD mutation and negative for AAV RH74 antibodies. The primary endpoint for cohort B was safety, with secondary endpoints in change in microdystrophin expression, looking at CK levels, and evaluating functional assays, including the North Star Ambulatory Assessment, as well as the others shown here. And finally, evaluating cardiac MRI at 1 year. The 4 patients' baseline CK are shown here, and at 3 months post-treatment, up to a year, these values have dropped by 70%. So this is very encouraging for us. We were very excited about the drop in CK. This is a significant drop that's been maintained throughout the trial. Finally, looking at expression levels in these gastrocnemius biopsies, we found that there is robust expression and appropriate localization to the cell membrane in all 4 subjects. So as you can see, looking at the percentage of dystrophin-positive fibers, we're above 80% across all 4 patients, which is highly encouraging. Finally, using Western blot analysis to look at protein levels, as you can see that all 4 patients had high levels of microdystrophin protein shown here. So this data was encouraging, and we saw a highly robust expression in the muscle at both the immunofluorescence level as well as the protein level. Again, all 4 treated subjects are doing very well. They had significant effects within 3 months of biomarkers including CK and dystrophin. And early results show that the boys are performing very well functionally as well. And importantly, there is an extremely favorable safety profile to date with up to 9 to 12 months of follow-up. To wrap up just where we are in the pipeline at Sarepta, the first 4 patients were included in trial 1, and this happened at Nationwide Children's Hospital. The first patient was dosed in January 2018, and that was using research-grade supply. Trial 2 has begun, and 24 patients were treated. This is a placebo-controlled, are being treated. This is a placebo-controlled trial, and finally this will move us into a confirmatory study.
Grazie mille. Il prossimo speaker è il professor.
Thank you. And now the floor to Professor Ronald Cohn, who works at the Sick Kids Hospital in Canada. Ronald Cohn won't be speaking about gene therapy, but rather of gene editing, which is a technology that is still not being done in patients, but that will complete the picture with AAV therapy. You have the floor.
Buongiorno. That's about all of the Italian I have for you. Thank you for inviting me to come and talk to you about what I think is the next step. So you've heard a lot of exciting updates about gene therapy. So what I'm going to do is I'm going to give you an update about what's going to come next after gene therapy. So I call genome editing gene therapy 2.0. And the system that we're going to use for this is called CRISPR, and it's really a system that has kind of revolutionized the way how we think about science and how we are going to think about treatment, not just for Duchenne muscular dystrophy, but for a lot of genetic disorders. And I'm going to give you a little bit of an update of what it is we do in our own laboratory and how we can use this technology. But at the end, probably most importantly, I'm going to discuss with you the issues that need to be resolved until we can really move this into a clinical trial. But let's start with the difference. So I was looking for the Italian alternative to Twinkle, Twinkle, Little Star, which I didn't find. So bear with me, that's a very common English sentence. So look at this sentence, and if you have a mutation in this sentence, it reads like this: twinkle, twinkle, big star. Now, if you now think about gene therapy, what you have heard in the last 3 talks, how would the sentence read? So the room is too big, you don't probably want to come forward with the answer, so I'm going to give you the answer. Current gene therapy leads to the sentence reading like this: twinkle, twinkle, little big star, meaning you're doing gene therapy by providing the gene that is missing, but you're not actually fixing the mutation in the cells. The mutation is still going to be there. And what CRISPR can do is it's actually going to fix the mutation and it's editing it in a way that you end up with what we call the wild-type product, and that's the eventual goal of CRISPR. There's one other aspect of CRISPR that many of you probably don't know about, which is equally powerful and potentially even the first aspect of how we are going to use CRISPR in clinical trials for patients, because you can inactivate the so-called scissor, which is cutting pieces of DNA, and then just use CRISPR to upregulate or downregulate expression of modifier genes. And I'm going to show you an example of this for Duchenne muscular dystrophy, but it is a very powerful way to alter gene expression in many different disorders. So I'm going to give you 3 examples before we talk about what needs to be true for this to happen. The first one is going to how we can use CRISPR for deletion mutations in Duchenne muscular dystrophy. As you know, most of the patients with Duchenne have deletions, up to 70%. And I'm going to particularly give you an example of a deletion of a patient. There are several patients that I know of who have a deletion of exon 52 to 54. And as you can see here, this leads to, not unexpectedly, to a dystrophic muscle. So what we did in our lab, we did 2 things initially. We generated the first multi-deletion model for Duchenne muscular dystrophy by recreating the exon 52 to 54 deletion in mice. We also generated a mouse that is removing exon 52 to 55. Why did we do this? We did this to test whether removal of exon 55 by restoring the reading frame would make mice better. And that's exactly what you see here. So if you look at the picture here, the middle picture shows you the animal model of exon 52 to 54, which has no dystrophin and is developing muscular dystrophy the way you would expect it. And if you remove an additional exon, exon 55, you can see that these mice have near normal dystrophin levels and have a normal muscle biopsy. And if you look at the function of these mice, you can see that they perform as well as the wild-type mice. So that was encouraging for us, and then we used this as our proof of concept to move forward and develop a gene editing strategy to remove exon 55. And we did this with a single guide approach, which is similar to what has been published by other groups. So Eric Olson, for example, is using a similar single guide approach. It's important that it's only one, because the more guides you have, the more potential side effects you have to deal with. So we use this guide, cutting out exon 55, and then looked at the results of this. So here you can see that we get very robust expression of a shortened dystrophin. It's important that we are restoring the reading frame, so it's a shortened version of dystrophin. With a single guide approach, we get similarly good expression in the muscle, and if you look at the performance of the mice, you measure the strength, the grip strength of these mice, you see that the ones who receive the single guide have significantly improved power in their muscles, so it's very encouraging. What we are currently doing, and we have a whole cohort of mice who have been injected now for 10 months, and we are going to wait 2 more months to see how do these results look like after 1 year of injection of the single guide, to see whether what we see early on can be maintained over a longer period of time. So the next example is an example of what I told you before. Here we are using CRISPR without the scissor, but using it to modify expression of a modifier gene called osteopontin. I'm not sure how many of you have heard about this, but it has been shown several years ago that by downregulating osteopontin, you can significantly reduce the fibrosis in muscle of mice with dystrophin-negative muscular dystrophy. So that was very good for us. It's also a modifying gene that has been validated in patients. So there are patients who have certain mutations in the osteopontin gene who either do better or slightly worse than one would expect. So it's a powerful modifier. So our hypothesis was we use CRISPR to downregulate the expression of osteopontin and then get better skeletal muscles. So this is exactly what we did. This is how this looks like. It's a technical slide, so we don't have to spend too much time on it, but this is the promoter we are using to downregulate the expression of osteopontin. And we did this in the mice that we generated before, the 52 to 54 deletion mice. And what we do is we use AAV9, like you have heard before, as a powerful vehicle, at least for the moment, and we'll discuss this in a second. And as you can see, we get significant downregulation of osteopontin. When you look at the power of the mice who have less osteopontin, you can see that they are stronger. They have less fibrosis as expected, and we were excited to see that we see quite a bit of a downregulation of fibrotic tissue. And you also see an overall improvement in the strength of these muscles. What we have to do here now is to see whether long-term downregulation of osteopontin leads to any potential side effects. At the moment, after 6 months, we actually don't see any side effects, but again, we need to make sure that that holds true for at least another 6 months. And the last example I'm going to give you is around duplications in Duchenne muscular dystrophy. About 12 to 15%, some studies say 17% of patients with Duchenne have duplications. And if you think about the idea of CRISPR, so here you have a scheme of a duplication. So you have AB, and the portion of the gene AB is duplicated. So if you think about a scissor, it comes to mind easily that you take one scissor on one side of the duplication, one scissor on the other side of the duplication, and you cut this out and you end up with a wild-type gene like this. The issue here is, if you use 2 of these scissors, it's always more challenging to put this into an AAV9 virus, and each of the scissors comes with its own side effect. So I had a brilliant person in my laboratory who figured out you can do the same thing with just one scissor. Taking the scissor, cutting it within one of the wild type and the duplicated area of the gene, and then you end up with a normal gene that looks like this. So we tried this experiment and did it in a patient who has a duplication of exon 18 to 30. At the current time, he's the only patient in the world who is known to have this duplication, which will become important for our discussion. So what we did is we took muscle cells of this patient, we exposed these cells with one single guide, and here's the result. So you get re-expression of now full-length dystrophin. So all of you know that any kind of therapy you hear about, you have heard about, in terms of restoring dystrophin expression either through exon skipping or through gene therapy, is always going to give you a short version of dystrophin. This, in theory, well, not in theory, this gives you the wild-type full-length expression of dystrophin. So we were excited to see this, and then we had to generate another new model because there is no duplication mouse model that was amenable to CRISPR therapies out there. So we replicated the duplication of exon 18 to 30 in a mouse. So I have to just give credit to my student. I'm showing you one slide that took about almost 24 months to make. It was very difficult to make this mouse. We used the CRISPR technology to make the mouse, but it was very difficult to finally get to where we are right now. But we are there, and I'm not going to bore you with all the 18 months of sweat and tears. Here you have a mouse model that has an exon 18 to 30 duplication that has no dystrophin expression and develops muscular dystrophy, as you would expect. We injected AAV9 virus with the single guide into these mice early on at the age of 2 days, and then checked whether we would get removal of the entire duplication. So here you have a Western blot of these treated mice, which is very exciting. As you can see, we get fairly significant expression and removal based on the removal of duplication in all tissues, including the heart, the diaphragm, and several of the extremity muscles. And I'm going to show you a couple of pictures about this now. Here you see restoration of heart dystrophin. Here you see restoration of dystrophin expression in the diaphragm. And again, I want to remind you, this is full-length dystrophin. Here you see it in the lower limb muscle. That's about the muscle that has the poorest expression. And then the forelimb muscles actually have significant expression. When you look at the histology, you see that the fibrosis is significantly improved in these mice. And the overall strength of these mice, it's not quite normal, but it's significantly improved. So where we are with this right now is we have to probably improve the efficiency a little bit. So we have 2 experiments that are going on right now. We have treated mice with a higher dose of the single guide to see whether we get even more removal of the duplication, meaning more expression of dystrophin. And we have another cohort of mice that is going to finish now almost 1 year in a couple of months, and we see whether this is a sustained effect. So before we talk about what needs to be true for this to happen, these are the people in my lab, and I want to maybe point out this person. She's the one who worked on all the duplication work, and I think it's important to acknowledge that I'm just here to tell you the cool stories, and these are the people who do the hard work behind it. So what needs to be true for CRISPR to get into the clinic? There's a lot of hyper excitement in the field, and I'm not here to tell you it's not very exciting, but I'm here to provide you, in my mind, a bit of a reality check. So the first thing we need to keep in mind is we have a number of incredibly promising gene therapy trials going on, and we've got to learn from what we get out of these results before we start clinical trials for CRISPR gene editing. I think it's critical that we use all this amount of data that we are going to collect, that we are already collecting, because it will help us inform how we're going to design a clinical trial for genome editing, whether it's duplication, deletion, or modification of gene expression. There's a lot of discussion in the field around what's the difference between genome editing of a patient versus an embryo. Who in this audience, let's see whether this works with translation. Who in the audience has heard about the Chinese babies that were edited? Raise your hand. There you go. That was terrible. It was terrible, it was unethical, and should not happen. Whatever I talk about and whatever we need to talk about as a field is the genome editing of patients. It's not affecting the germline. The main reason why I'm telling you it is terrible is if there's any side effect to genome-edited babies that were unintended, then it's going to carry through generations to come. If God forbid something happens in one of the genome-edited patients, whether it's for Duchenne or for any other genetic disease, it's going to happen in one individual and not in the generations to come. So everything around genome editing at the moment has to be related to living patients. Off-target effects is something you hear quite a bit about. It's very important that we come to a general conclusion as a field of when we feel like it's ready to go into a patient. I can tell you, none of my colleagues, and we don't ourselves, see any off-target mutation, meaning mutations in other genes that we don't want to edit. But you know what? All of these studies are either done in mice, and I could not care less about the mice because they must not represent the human, or they are done in cells in a petri dish in culture, and whether that translates into a human is something that I'm not so sure. We have to figure out when we feel like it's ready for us to go to a patient.
Il microfono non arriva dalle traduzioni.
1 minute. And the last thing I want to say is we need to think about other delivery vehicles. As you know, once you inject AV9 into a patient, you can never re-inject it again. So I actually believe that the future of gene therapy and genome editing trials is going to it's going to utilize different kind of delivery vehicles, and that's the way we have to think about it, and we can discuss it more during the questions. Thank you.
Ringraziamo Ron Cohn e
Thank you very much, Ron. I'd like to now call upon Giulio Cosso, who's from the University of Manchester.
Lingua parlo? Ditemi voi.
Sardo.
What language should I speak? How about Sardinian or Roman? Okay, that is what I'll do. I'm very happy to be here once again. For some years, the aficionados didn't see me, so I just wanted to warn you all that I'm still alive and kicking. And I spent these few years trying to understand what had not worked.
Uh-oh, I did something wrong.
In the clinical trial.
Di cui
that I spoke to you at length in previous years. Now, just to contextualize, let me say that we engaged in preclinical work for almost 15 years in 3 different models of muscular dystrophy. And the only model involving a big animal back then was the golden retriever, a dog model. That was based on the transplant of cells that were capable of forming muscle that we had called, and we still called, mesoangioblasts. So they're not real satellite cells like those that were used back in the 90s, but they do have the capacity of adhering to the wall of the vessels whenever there's an inflammation. And of course, there is an inflammation in a dystrophic muscle. They're able to cross the wall, and this provided us a unique tool, a unique delivery vehicle for cell therapy, because it carried these cells to most of the muscles that were downstream with respect to the artery that was being injected. Over the years, when I still worked in Milan at the San Raffaele, this led us to undertake a phase 1/2A trial that was based on the transplantation, intra-arterial transplant using a catheter of these cells that came from a donor who happened to be a brother that was immunologically compatible, and that was HLA identical. The findings of the study were published a few years ago. This is, the timeline of the study. And the findings, the results, I'm afraid, fell way short of the expectations that had been generated by studies on or trials on animal models. We wondered what the reasons were why the results were so modest compared to what we'd observed in mice and dogs. And we found various explanations. And for some trivial reason, for example, we were never able to achieve the dose that we had administered to the animals in questions. And then the patients selected were at an advanced phase of the disease, whereas the animals were treated very early on. And this is a lesson that in cell gene therapy was really crucial, and now it's become mainstream, of course. And also, patients were being treated with steroids, and we didn't modify that, and you'll see what the problem is with that. And finally, we're the only mammals that are bipeds, that walk on 2 feet, 2 legs. All the other animals have 4 legs. It's like a table with 2 legs versus one with 4. So reinforcing leg muscles only, even if we had been more successful than we were, would not have been sufficient to correct the posture of walking, because that's based on other muscles. For example, the back and the pelvic girdle that we hadn't even worked on. However, in spite of this adverse result, you will see that in the younger patient that had received the brother's cells, we found an amount of dystrophin that could be detected with the Western blot, shown in the green arrow. And this had to come from a donor, because this patient had a deletion, so that he was lacking about half the gene, so he could not have been able to produce a full-length dystrophin. And the patient that had the best response was not much worse than the dog. I hope you can see this, that the dog was amongst those that had responded less favorably. So the distance, the gap between therapeutic failure, so to speak, and the efficacy threshold was not huge, but still there was a lot to do, of course. At this point, we got down to work and we tried to improve the various phases of the process, and we devised 3 strategies that should, if everything works right, that should converge in one last trial, hoping that I am not wrenched away from this before that time comes, before this trial sees the light. The point is that there are many phases in the vascular transplantation that can be improved. Anyway, the second point is that we do need to reach the back muscles and the diaphragm, and these cells are just not in the condition, in the position to form cardiac muscle. So we do not expect this approach to solve the issues related to the heart in DMD. And we're actually checking this, validating this in a clinical trial that we are carrying out in Manchester. So how is it that we can actually improve the protocol? How can we enhance it? Well, imagine that a catheter transplants cells in the femoral artery, in the aorta, or in the subclavian artery. From that point on, the cells have to go into the capillary stream. They have to adhere to the endothelium, cross it, bind to the tissue. And the accumulation of fibrous tissue, of course, is a barrier that cells are just unable to cross, and therefore we have to stimulate fusion with the fibers that are being regenerated. And most crucially, these cells have to be used like a Trojan horse, if you will, to help the other nuclei of the same cells produce dystrophin. There are many nuclei involved in these cells. Anyway, we did in vitro studies with human endothelium, and we observed things that everybody knows about practically, namely that the proteins that the endothelium expresses whenever there's an inflammation bind white blood cells and take them to the site of the inflammation. The same proteins are used by stem cells. But if we treat the endothelium with steroids, the binding is reduced dramatically. Now, all of our patients were being treated with steroids, and so without knowing it, and I still can't come to terms with this, but anyway, unknowingly, we reduced the binding to the endothelium 3 to 4 times, which we could have very well avoided simply by withdrawing steroid therapy a couple of weeks prior to the transplant, which is totally legitimate because, as far as I know, there are protocols still underway that use alternated treatment. In other words, they provide for steroids on and off. So that was the second strategy, the first strategy. The second is shown here. The endothelium is in red. You look at what happens below. A normal cell is trying to go across the endothelium, but it's having a hard time. At the same time, another normal cell crosses the endothelium absolutely normally, but provided that the JAM-A is missing. So it would suffice to remove this protein or silence it to enhance the passage of these angioblasts 2- or 3-fold. Now, the antibody that was available at that time was not given to us for the clinical trial. However, recently, 2 groups published certain results pointing to the fact that rosiglitazone, which is used for diabetes, inhibits JAM-A. Unfortunately, rosiglitazone in the long term has cardiotoxic effects. Instead, pioglitazone doesn't seem to, and it also inhibits JAM-A expression, as you see by immunofluorescence in these 2 panels, and you see this in PCR. And it increases the passage, the crossing of the endothelium, and this is something else that we observed and learned. But, well, all this is for the future trial that I mentioned at the beginning, the final trial with systemic delivery. I have a slide that I can't seem to find, but anyway. The main issue that we had observed instead with our trial at the San Raffaele in Milan is that in the patient with that little dystrophin that you saw, the amount of cells of the donor was less than 1 out of 100. So how can we possibly hope that a single cell can help to correct the defect in the other 99 cells? And here, even if what I said were to work, we could get to an engraftment of the muscle up to 4, 6, even 7%, but not beyond that, because all the ex vivo gene therapy trials that worked involve epithelial disease and blood disease, where it is possible to remove the sick cells with myeloablation, for example, and make room for donor cells. But we can't do that. You can't remove the heart, the brain, or muscles. And so the transplantation strategy will never lead to a sufficient production of dystrophin to hope for a clinical effect. So how do we get around that obstacle? Well, take a metachromatic leukodystrophy. In that ailment, glia would produce a missing enzyme, and the neurons of these children captured it, and the disease was blocked, halted, at least for the time being. And this was actually done by the group of Nardini and others at the San Raffaele Hospital. But in that case, again, they did not try to repair or replace the neurons, but to interject the missing protein, which fortunately was a protein that was secreted, and so it could be captured. But that's not the case of dystrophin, as you well know. So we said, why don't we go back to what was observed in the past and try to make it fit the situation today? To prove that that idea could work, we began a clinical trial in a single muscle for safety reasons. Because although Mr. Antiblas have already used clinically, and although cells like this have also been used in a clinical trial, well, that wasn't necessarily the case for the other proteins. So in Manchester's, we took cells from a patient, we corrected them with a vector. They will be injected in the contralateral muscle to see exactly how much dystrophin we'll be able to generate. These are the participants in the trial. I'm not going to mention them all. Of course, the primary objective is safety and then efficacy, because we want to see exactly how much dystrophin we're able to produce. So there's a biopsy, muscle biopsy, there's transduction of the cells with this vector. We're at this point now, we have an intermediate product which has been frozen. As soon as we get the approval on the part of the regulators or regulatory authorities, the product will be injected in the contralateral muscle. If we do not get up to an additional 10% of dystrophin of healthy muscle, the trial will end right here, and I will retire immediately. If instead we do get more than 10%, some cells will be injected in the hand muscles. All of these patients are non-walking patients because we want to see whether the contraction power of the thumb increases proportionately. Why should it all work? That was the slide that I should have shown you before, but anyway, because we can use the genetically corrected cell as a horse, because it goes into a fiber that is being degenerated, and all the other nuclei in there are dystrophic, so they cannot generate dystrophin. But the vector produces a small RNA, mRNA, that was designed by Bozzoni many years ago, that produces exon skipping. This small RNA goes into the cytoplasm, it assembles, it comes together with proteins and goes into the nearby nuclei. So it is capable of correcting nuclei of the same group inside the fiber muscle. The muscle fiber. We checked this in vitro. The results are quite encouraging because you see the dystrophin, we can see even with the dilution of one corrected cell to 29 uncorrected cells, which mimics a colonization of 3%. If you use normal cells that produce dystrophin, as you dilute them, the dystrophin disappears. But if you use genetically corrected cells, I'm sorry, we've lost the microphone. There is a dilution of 1 to 30. And this is visible also when you inject these cells in immunodeficient animals. So the idea is that clinical efficacy may be achieved by enhancing every step of the process. We should get to 30-40%, which is considered to be therapeutic. We will only know the answer to this in about a year from now, and we'll see whether we will continue or not. Thank you very much for your kind attention.
Grazie Giulio, e io inviterei anche Giulio a rimanere qui con noi per...
Thank you, Giulio. I'd like to invite Giulio actually to stay with us for our question and answer session, along with the other speakers who have spoken.
Yeah, so that we can have a question and answer session. Quindi se cominciate a pensare chi vuole chiedere delle domande in modo da mettere le mani sopra, vi raggiunga.
Please raise your hand if you'd like to put questions so that you can be handed a microphone.
Allora, intanto che aspettiamo qualche domanda che matura.
As we wait for questions to come in, we'd like to talk to you about a new initiative. We have a new app. It's called Duchenne and Me, and it was devised to support families from the moment of diagnosis onwards. It's not available on the Apple or Android store yet, so it's rather complicated to download, but we do invite you to download the Italian version, I'm afraid. That's the only one we have right now. Please download it, but first you need to install a small application that will allow the Duchenne and Me app to work. And because this is a zero version, it's the pilot version, if you like, please report any possible glitches you might find so that as soon as possible, we might come to a perfectly functional version. So we ask you for your cooperation. If you can, please download it as much as you can. Try to get your data in there, and please write to us should you encounter any issues so that we will work on making it increasingly performing. Are there any questions? Yes, there is one down there.
Hi, hello. Janet Bloor from the UK. This is a question for Rachel, who I see has just left the platform. I'll give you a minute to get back. Hi, Rachel. This is a question regards Sarepta's recent statement on Goldesferin. What are the implications that the FDA are currently not planning to hold an advisory committee? Does that mean that the next round of trials will be expedited for exon 53, 44, and the rest?
La traduzione simultanea non è arrivata al gruppo. La domanda non era tanto sulla gene therapy, ma sulla come Sarepta porterà avanti i vari prodotti per antisense oligonucleotide, exon skipping. Quindi
Yes, great question. I believe tomorrow there will be a session specifically on exon skipping and these therapies. So I will, we will have further information on that tomorrow. It's very early, so I would focus tomorrow on the presentation.
Altre domande? Una domanda qui. E anche ce n'è una dietro, poi.
Salve.
Hello. I'd like to go back to Professor Costu's presentation. I was thinking about something and listening to him. Cortisone and corticosteroids that are administered as the sole therapy for our children could be a hindrance, it would seem, to possible new treatments because... So my question is, why is this not foreseen in a preclinical trial phase? In that phase, do you think about the administration of possible drugs following mice or rat models that are also receiving corticosteroids? And do you compare that against other animals that do not take corticosteroids, just to see the extent to which cortisone can hinder or maybe help the potential therapy? It's surprising to see that in 20 years, we've been able to treat all the rats of this planet, and it's surprising to see that after all these 20 years, the same has not been seen to happen for our kids. Maybe we need to start asking ourselves questions about that. Well, maybe I was misunderstood. Corticosteroids are a consolidated therapy and they're not being challenged in any way. In the specific case of cell therapy, you see, I don't have any information concerning viral vectors. I don't know whether the inflammation of the endothelium has an influence or a bearing on the passage of the vector. But with regard to cell therapy, we just need to reduce steroids and remove them for like a week. And that's it. And then reinstate them. So it's evident that the interaction between drugs can positively affect things. Let me give you an example. Any drug that slows down the progression of the disease increases the likelihood of success for cell and gene therapy. We just need to prevent these small incidents from happening. In our case, we hadn't realized that the reduction of inflammation would have reduced the passage of the cells temporarily, that's all. I mean, you need to contextualize. It's not that steroids are not good. It's just simply that you need to very accurately, when designing a clinical trial, you really have to factor in the possible interaction with other drugs, whatever that is. But a good clinician, and that's not me, should know about that.
Okay, c'è una domanda di là.
Okay, we've got another question there.
Mr. Binks of Pfizer. The question, I guess, is something that you would expect from a parent conference. Do we have any indication of the efficacy results so far from the clinical trial? And if we don't, what is the timeline that you foresee for that?
So we're intending to disclose some information about the muscle expression of our transgene later this year. We've taken the approach of batching up biopsies together from multiple patients to be analyzed at one time so that the analysis can be completely blinded and it can be assessed in a highly objective manner. So we will be sharing some of that data later this year.
Altre domande?
Any other questions?
C'è una domanda anche lì.
Yes, there's a question down there. I'm sorry, you came first.
From Brain Project Czech Republic, and my question is for all the gene therapy partners. If I understood correctly, then one injection pretty much means for the patient that he can't really get another one, that he develops immunity. Is that then correct? And if so, is it right that the patients who are now involved in trials would not be able to get the treatment? Thank you.
So yes, it's a single injection for all the gene therapies. And after administration of the virus, you do elicit an immune response and you would not be able to be retreated. The hope is that it will only require one treatment, and so therefore those on the study would already have received that treatment.
Il professor Cosu e poi c'era anche una domanda lì.
Professor Cosu, and then we've got two more questions. I see two hands.
Question for Ron. Maybe you understand me in English, you don't need translation. Do you see a significant drop of efficacy in editing when you go from 2 to 1 to a single guide?
So we currently don't see the drop in efficacy that has been reported by other groups. I think that's why you're asking probably the question. But I do think overall the efficiency of the editing that we see is not where I would like to see it before we think about a clinical trial. That's why we are going through the experiments of using higher doses of the AAV and the vector and see whether it's showing a sustained expression over time. I'd like to add a comment to the previous question because I thought I had 20 minutes for my talk, but only had 15. So I couldn't bring home the message about the new delivery methods. I do believe it's important for all of us as a field to think about alternative non-immune delivery vehicles. Because even if you just need one injection for gene therapy, it's probably fair to assume that we will never get close to 100% expression. So if we think about something like genome editing as a second step, as an additional step on top of what currently gene therapy has to offer, if we can use something that is not immunogenic, so not a virus-based vector, we'll have the opportunity to synergistically add on to what gene therapy is, what I believe is going to be a game changer soon. Still, there won't be 100% expression. We'll need to think about therapies on top of it. So personally, I feel one of the major hurdles we have to overcome is to move away from these immune-based delivery vehicles that are currently creating some limitations.
C'è una domanda qui in fondo.
Hi, I just wondered if any of the companies working on gene therapy could comment on plans to bring trials to Europe?
From Pfizer perspective, we're currently planning our pivotal trial, which we do expect will be conducted partly in the US and Europe and around the world. So preparations for that are underway, and I think you can expect to hear more information again later in the year.
And so for Solid, we've actually communicated before that we're looking to move into Europe and expand sites also in the US.
Sarepta also has all intentions of bringing trials to Europe.
Lì il microfono qui al signore. Sì, grazie.
Does it work? Yeah? Okay. Goran Vasovic, DMD Serbia. First of all, I would like to thank to all of you for great presentations, and short question for Rachel from Sarepta. As far as we know at the moment, your microdystrophin therapy covers only boys with mutations between 18 and 58. So my question is, do you plan to expand the range? And if yes, when do you plan to do it? Thanks a lot.
Thank you. Yes, the first study that was undergone at Nationwide Children's Hospital did have the inclusion criteria and the mutations 18 to 58. However, these are very early days, and we plan to have greater inclusivity in the trials and move to look at all mutations.
Ci sono due domande qua.
We have two questions right here. Please raise your hand.
Grazie.
I'd like to thank all the panelists for their presentation. And my question was for Dr. Cohn. I note with pleasure that finally there is a valid therapy for duplications. My son has an 8-9 duplication, and I was wondering, because it's in the initial part of the gene, is it possible to use this technique? And are there plans for undertaking trials on human beings and not just on rats and mice? And if so, when? Thank you.
Thank you for your question. The first part of your question, can you remove a duplication of 8 and 9? Yes, you can. We actually have done this. We have a patient sample of another patient. So if you can email me with your contact information, I currently have a running list of patients who have duplications. So the question about when are we going to start humans and move away from mice and rats. I feel your frustration that this is not happening fast enough, but I also believe there is the reason why I ended my talk with what needs to be true and to kind of put a bit of a reality check into place is we don't want to do something that's potentially going to be harmful. Everybody here in this room probably knows when the first gene therapy trial for not a muscular dystrophy but another genetic disorder happened many years ago, it paralyzed the field for almost 12 to 15 years. So I can promise you I have a little army of people working on this, and we do want to go into patients, but there are still a number of questions that we have to be conscious about and answer. And as soon as we have the answers to this, we can think about clinical trials. Personally, I believe CRISPR for Duchenne muscular dystrophy, whether it's duplications or anything else, still needs a little bit of time. I know we all don't have time, but we don't want to do anything wrong. I promise you there are a lot of people working on this, and we are determined to get this into the patients as soon as possible.
Can I ask, faccio not necessarily to Ronnie, but also to the AAV gene therapy guys, is duplication an exclusion criteria for some of your studies or not? Because in a way that may be also important to realize that not necessarily there is no things that may be happening for people with duplication. Scusi signora, non so perché, lei capisce l'inglese? Ok.
So Mike just said that for Pfizer and for Solid, we don't have any exclusion criteria based on the mutation, so we take all comers.
Okay. Yeah.
My question is regarding the pre-existing antibodies and also for re-administration of AAV. Are there any procedures to get that antibody out of the body? And are they feasible? Is it surgical operations? And are people working on it? Is it possible re-administration of AAV and therapy? Is it possible by doing these procedures? Thank you.
There are some approaches that are heading in that direction. So there are experiments being done to look at the use of immunomodulatory therapy at the time of treatment. And there are experiments being done to look at the depletion of existing antibodies prior to a treatment or a retreatment. At the moment, I don't think there's a procedure that we can point to that will guarantee our ability to retreat. So it's still very much in the experimental phase. There are approaches that are being explored. I don't think anybody has a solution yet. And so with current technologies, we'd have to say there isn't really the option to retreat. Having said that, the thresholds that each group is using to exclude may be slightly different. We're all approaching this in a precautionary manner. I think initially, to demonstrate efficacy, we're excluding quite stringently. In subsequent experiments, we'll be looking at low levels of neutralizing antibody and whether they can be overcome by one means or another. So it's very early, but initially people are being pretty careful about those exclusions. Having said that, in young boys, the seroprevalence of antibodies, certainly in our case, does look to be quite a lot lower than the published numbers out there.
Allora, io vorrei ringraziare
I'd like to thank all of our speakers of this first morning session. Thank you. It was extremely interesting. Don't worry if there are additional questions. We can maybe take them up in the next session, or maybe you can send them to us by email and we'll get the answers to you. So thank you for being with us this morning. I wanted to present another initiative. Parent Project has financed research ever since it was set up back in 1996 when we were founded here in Italy. And historically, it was really important for Giulio Gosso to be with us today because he was the first researcher that we funded through Parent Project. He also mentioned René Buzzoni, who was one researcher funded by us, and it's wonderful to know that this research is continuing to bear fruit today. This year we're funding 13 different projects, and so we took this opportunity to introduce you to them because it's really important for you to have this information. There are 7 that were already in progress last year and 6 brand new ones. So during the conference, you'll see some very brief videos and clips in which the researchers were asked to explain what they're doing in 2 minutes and how important their project is how important it is with a view to finding a solution to this problem. I'm going to show you 2 of them now, then we'll have a 15-minute break, and then we shall reconvene for another important session. So let's watch these very quick video clips now.
Buongiorno a tutti, sono Alessandra Ferlini e lavoro all'Università di Ferrara, sono un genetista medico e lavoro sulla distrofia muscolare di Duchenne già da parecchi anni. Tramite il Parent Project stiamo portando avanti 2 progetti, un progetto di ricerca che è mirato alla identificazione tramite anticorpi della proteina distrofina su liquidi come plasma e urine al fine di mettere a punto e identificare se possibile una metodica molto rapida, proteina sensibile e poco invasiva per dosare la distrofina sia nei pazienti con distrofia muscolare di Duchenne, ovviamente anche di distrofia di Becker e anche nelle portatrici sane. Idealmente, se questo tipo di metodica, di strategia funzionasse, il dosaggio della proteina distrofina nel plasma e nelle urine potrebbe essere estremamente utile sia per monitoraggio della patologia o della valutazione dell'andamento della patologia e della severità del quadro clinico nel tempo, ma anche in base soprattutto alla sua specificità in applicazioni di test neonatali come test newborn. Il secondo progetto riguarda una biobanca che raccoglie e conserva e stabilizza delle cellule staminali di origine urinaria, le cellule staminali urinarie sempre da pazienti con distrofinopatia, quindi in senso ampio su Duchenne. Becker, in questo senso, in questo caso la biobanca, ha come obiettivo quello di rendere disponibile per scopi di ricerca e di diagnostica queste cellule multipotenti nelle distrofinopatie. Grazie.
Mi chiamo Guglielmo Sorci, sono un professore associato di anatomia umana del Dipartimento di Medicina Sperimentale dell'Università degli Studi di Perugia. Con il mio gruppo di ricerca mi occupo di muscolo scheletrico da anni, con particolare riguardo alla rigenerazione muscolare, all'atrofia muscolare e alla distrofia muscolare, in special modo alla distrofia muscolare di Duchenne. Recentemente con i colleghi professor Riccardo Calafiore e professor Giovanni Luca abbiamo messo a punto un protocollo terapeutico per il trattamento della Duchenne in topi distrofici, consistente nell'inoculo di cellule di Sertoli microincapsulate all'interno della cavità peritoneale. Questo trattamento è risultato in un forte miglioramento del quadro morfologico e clinico dei muscoli scheletrici dei topi trattati. Abbiamo quindi deciso di approfondire vari aspetti di questo tipo di trattamento e grazie a un progetto biennale finanziato da Parent Project, che è ora in fase conclusiva, abbiamo potuto determinare la dose minima efficace delle cellule di Sertoli microincapsulate, abbiamo potuto documentare effetti positivi delle cellule di Sertoli anche sulla componente dei mioblasti del muscolo scheletrico, abbiamo potuto documentare la morfologia di queste cellule all'interno delle microcapsule nel tempo, nonché abbiamo importanti dati relativamente alla sicurezza dell'utilizzo di queste cellule negli animali e quindi presumibilmente nell'uomo. In particolare queste cellule hanno dimostrato, o meglio abbiamo dimostrato che queste cellule non sono immunosoppressive ma piuttosto immunomodulatorie. Tutti questi dati ovviamente favoriscono una potenziale applicazione dell'uso di cellule di Sertoli microincapsulate nei pazienti distrofici.
Allora io ne approfitto per ringraziare il professor.
I take this opportunity to thank Professor Mercuri. He keeps calling in Professor Mercuri, and thank you for being my co-chairman. No, now he's the real name, Francesco Muntoni, and we'll reconvene in 15 minutes. Thank you.
riaccomodarvi che tra 1 minuto e mezzo inizia la tavola rotonda. Se riuscite a riprendere posto, proviamo a recuperare qualche minuto del piccolo ritardo che abbiamo. Grazie. Per favore, se da in fondo riuscite a rientrare in sala e a prendere posto, possiamo provare a ricominciare. Ok, faccio l'ultimo appello a tutte le persone in piedi in fondo alla sala di accomodarsi e chiedo gentilmente a chi voglia continuare a scambiarsi opinioni o a chiacchierare di farlo allontanandosi dalla sala, altrimenti si sente un brusio fortissimo. Quindi vi chiediamo di prendere posto perché possiamo avviare la sessione numero 2, che è la tavola rotonda moderata dalla nostra...
We're now going to begin our second session, which is going to be chaired by Marika Pugliese from Parent Project. The title of this, when complexity increases, so do the problems. And this is what we're going to be discussing now.
Buongiorno a tutti. In questa sessione vorremmo...
Good morning, everyone. In this session, we'd like to talk about the issues and problems related to BMD and DMD, based on studies that show how in 1/3 of the cases there may be problems setting in behavior attitude, learning skills, and neuropsychological function. Special attention will be devoted to the ways that we can adopt in order to implement these approaches in the daily life of our children, in the school setting, at home, and so on. And we will culminate with witnesses from the families. I'd like to now give the floor to Ms. Pane, who's from the Children's Hospital within the Gemelli Hospital of Rome. We've worked with her for many years, and she's been dealing with the clinical management of DMD and BMD patients. Thank you, and good morning, everyone. It's always a great pleasure to see so many familiar faces that I often see in the clinical setting, a lot of friends and a lot of travel companions. This morning, we started with a lot of hope and expectations and innovations, hopes that we share on a daily basis, but of course we have to reckon with daily life and what it is that our children, your children, have to deal with day in and day out, school, rehabilitation, and so on. The cognitive aspect is something that is very dear to my heart. I'm a children's neuropsychiatrist. That's what I studied. And so I know children from their infancy. That's why I'm so interested in their cognitive skills. And I always like to see that aspect in children, even if they're affected by DMD. Of course, we do place a lot of emphasis on the motor aspect. We try to prolong deambulation. We try to do our utmost in order for our children to walk as long as possible. However, the cognitive aspect must be equally considered when managing a young patient with DMD, because this translates into quality of life and good integration into the various settings that they interact with. Parika talked about the cognitive aspects. It's known in the literature that 1/3 of children with DMD suffer from mental retardation. We also know that these children may experience speech impediments and a delay in speech when they're very young, and children affected by DMD may also have learning disabilities. Why is it that these children have learning disabilities? Well, there are various theories and various studies that have been published in the literature. Some authors say that this is due to the underlying disease rather than the psychological causes. It is said that they are independent of the motor disabilities, but several studies in the literature over the years have led to an observation, which is that some children with certain mutations or deletions and duplications. And by the way, we too published some studies in which we showed that children with mutations after 44 have a greater likelihood of developing cognitive problems, whereas children with mutations before 44, gene 44, have less odds of developing such disorders. There are isoforms of dystrophin, and DP71 and DP140 are those agents in the brain that are most responsible for these problems. Now, there are also behavioral problems. ADHD. And according to the literature, between 12 and 30% of children with DMD can suffer from behavioral disorders and hyperactivity. So they're impulsive, impatient, they fidget, and obviously this has a negative bearing on learning abilities and social skills in these children. Now, there can also be behavioral problems like obsessive-compulsive behaviors. So 60% of these children can suffer from obsessive and compulsive behaviors, although this type of diagnosis is misknown and not always recognized readily. Another phenomenon observed in DMD, is autism, because we have a more or less patent degree of autism, and percentages according to the various studies range from 3.7 to 11% of all children with DMD. We tried to determine whether there is a genotype-phenotype correlation within these children and the reasons why all this happens. In the brain, there are isoforms of dystrophin that are expressed in certain specific locations of the brain, as a result of which the absence or presence of these isoforms determine these problems within our young patients. We try to determine whether in very young children there is something like the possibility of detecting the issue rapidly, very early on, in order to administer very specific treatment. So in our group, we looked at 80 young children aged under 5 years. This is a Griffith scale that measures development and provides us with a cognitive level of a child under 2 years of age. We observed that most children had a motor retardation or delay, which we expected, but they also had delayed speech. And we started to think that maybe pediatricians should start thinking about conditions beyond speech impediment, and they should maybe think about taking up an CK assay. We looked at several items from these scales, and we realized that motor delay or motor retardation is a delay in acquiring a certain stage. So there are children that get to a certain stage, but with a bit of delay, like getting up from the floor or going up the stairs. Maybe they do so a bit later than their peers who are not affected by DMD, but they'll get there eventually. That's because it was observed that there's almost a delay in these phases caused by a delayed maturation of the child because of the dystrophin. So what we did was to take Conley's trial. She used a Bailey scale, and her findings were very similar to ours, motor delay and speech delay in the children she examined. But we did something extra. We wanted to determine whether the Griffith scale could be predictive in terms of the adaptation of the child. We wanted to see whether the children had the same cognitive level when they grew older according to certain scales. We wanted to study their evolutionary pattern because the cognitive level remains stable over time. It doesn't aggravate, doesn't exacerbate like the motor level. And this is really important because we can make a prognosis when it comes to the cognitive and behavioral aspect of these young children. Why is this important? Well, because the takeaway message that I want to leave you with, and that Dr. Lombardo is going to touch upon, is this: the need for early diagnosis and early rehabilitation. Because, of course, younger children experience a retardation because of a brain, a cerebral phenomenon, and not because of DMD necessarily. So the maturation does happen, but this means that early rehabilitation can help to intervene appropriately early on and therefore improve the likelihood of treatment of the child. Thank you.
Ringraziamo la dottoressa
I wish to thank Mrs. Pane, who gave us an idea of the complexity families must face when this type of diagnosis becomes known. And after the diagnosis, we must try and understand how all this is to be approached in terms of rehabilitation. And we give the floor to Mrs. Lombardo, who works in the region of Lazio. Thank you, everybody. I have some slides. Good morning. I wish to thank Parent Projects for inviting me. I am a pediatrical neuropsychiatrist and I work at a center that is specialized in DMD and Becker. Mrs. Pane clearly explained all the neuropsychological problems that may be associated with these diseases. Biological factors such as the absence of dystrophin in some areas of the CNS, but also chronic treatment with glucocorticoids and as well as social and emotional factors linked to the underlying disease lead us to define DMD and Becker as multi-systemic disease. This has a significant impact on the quality of life and the family burden. We can say that in some cases and in some stages of the disease, the stress caused by the management of these neuropsychological problems may be even more important than the management of the motor component. In DMD and Becker must be managed from with a variety of approaches in all their parts and complications. International guidelines devote a part of this psychosocial management, and they say that the neuropsychological management must start at diagnosis and must go on for the whole of the life of the patient. This is the multidisciplinary team that is essential for the rehabilitation care of these patients. There are different specialists, rehabilitation specialists, and also social workers and psychologists. As for neuropsychological problems, children neuropsychiatrists are very important, therapists of neuropsychomotor sciences, and then speech therapists and assistants and tutors, and also occupational therapists. This team must be trained on the specific problems of Duchenne and Becker. They must be aware of the disease, they must know what the disease is about and what they may expect over time. That is why we speak about this neuromuscular and psychiatric team. It is important to assess some areas, the neurocognitive area, language and learning, behavior, the ability of the child to manage his own emotions and adaptation abilities, an important screening on the psychosocial well-being in the family, so patients, siblings, and parents, and that is to be done at diagnosis, before starting school, and at any moment of change, be it school or the social environment or functional changes, and an overall assessment is to be carried out every, say, 2 to 3 years. As this to the things we know from the international guidelines vis-à-vis our clinical practice, if we're speaking about neuropsychological problems, rehabilitators, rehab medicine aims at having the best possible cognitive and behavioral approach for that child and his specific situation, and that is why we must step in very early, working on prevention, and that is why I subdivided my speech into 4 different age classes, and there the concept of early diagnosis becomes paramount, because at diagnosis we know, well, we get an alert for neuropsychological problems. So if we have the possibility of doing something early for rehabilitation, assessing the neuropsychomotor development, and also implement different things at different moments of development, that is extremely useful, of course. But then, after age 3, we can better assess cognitive status and more specific cognitive functions such as memory and executive functions and school course, and behavior. When the child starts primary school, well, we must concentrate on specific learning disturbances. The DSA diagnosis is expressed at the end of the 2nd class of primary school, but it is also still possible to identify a sort of pre-existing condition, and so we could do something even before the final diagnosis is made. And so we must try and develop an overall type of functioning, now helping them to face the transition phase, and so the possibility of developing a number of autonomous behavior with regard to social functions, but also motor functions, and helping the child to social inclusion, in social inclusions also through psychological support, which is something we do by devising a rehab protocol. So, chronic diseases, first of all, because they are chronic diseases, but they are also dynamic diseases because there is a progression of the disease. And the other concept is the complexity of the disability. So, to what extent the level of disability has an impact on the possibility of participating in the social environment. There are children with the same diagnosis and same age may not have the same level of disability, but the impact, the hindrance to participation may be affected by factors that are somehow linked to the disease, comorbidities or psychosocial factors that may be different from one person to the next. With the rehab project, with the rehab protocol, we must not concentrate just on symptoms, but on the biopsychosocial recovery, so to speak. And it all depends on the specific needs of the individual child. So we must identify the objectives we aim at, the programs, the rehab programs to be implemented, therapeutic programs, care programs or educational programs, and it is important also to define the time scale for these objectives. It could be short, medium and long term. Without the family, it is impossible to obtain any sort of result, because the family is essential part of the project, and it is together with the family that you must identify objectives. First of all, optimizing the cognitive and behavioral aspect, and so we must identify a number of objectives, let's say, on the way. And we must identify priority objectives and also the objectives that can be realistically achieved. And another aspect is school. School is the place where our children spend most of their time, where they learn and where they create a relationship with a relationship in a situation that is different from being in one's own family. So family and school, and that is why the family is within the educational project, but we must also care for the family. We must make a psychosocial diagnosis of the family at diagnosis and also over time, and we must also involve the caregiver, the person who daily participates in the care. There is a higher rate of anxiety and depression in patients and family because they face a progressive disease with very well set worsening stages, phases over time. So at every step, the family must get organized for a sort of new disease, and that determines, great stress, causes great stress. And school is essential too. It is essential, and there is a provision in Italy that regulates this relationship between health care staff, school and family. And now there are many acronyms in Italian referring to working groups that, for example, in this case, GLHO, they meet once a year, and there are doctors from the local healthcare unit and parents and teachers and also the support teacher as well as the headmister or headmistress, and they plan the necessary resources to be made available. It is impossible to reach an objective if people are not willing to work together. We must work as a team. Thank you.
Grazie alla dottoressa Lombardo con cui...
Thank you, Dr. Lombardo. We've often spoken
poi della pianificazione di un progetto riabilitativo che passa anche attraverso.
And she talked about the various professionals that are involved in these projects. And now I'd like to give the floor to two individuals who work in neurocognitive and neuromotor rehabilitation. Dr. Rosie Galbo, who's a developmental neuro and psychomotor therapist, and Dr. Serena, who is a speech therapist who's acquired a lot of experience in the treatment of our children. Thank you. Good morning. I'd like to thank Parent Project and Dr. Pugliese for having kindly invited me and giving me the opportunity to give my contribution as a neuro and psychomotor therapist and give my contribution to this roundtable. I'd like to go back to what both Dr. Pane and Dr. Lombardo said about the problems related to the diagnosis of BMD and DMD, and on how important it is to have an early diagnosis and early as well as multidisciplinary rehabilitation.
in light of what emerged in trials concerning the comorbidity of behavioral issues and neurological issues, of course, psychomotor therapists and speech therapists and neurotherapists too play a very important role in the management of the young patient. Above and beyond the mere motor and physiotherapeutic approach, which consists of stretching and functional maneuvers, what really is important is to focus on very young children and an early approach to them. Indeed, we need to work from the very outset to achieve neuro and rehabilitation goals based on various aspects, regaining attention, the control of motor activity, and everything else, because all of this obviously has an impact on educational proficiency. This type of neuropsychomotor approach dovetails with rehabilitation training, and it really does dovetail with approaches that can also be playful in order to achieve muscle functional and other rehabilitation goals in children affected by this condition, so as to recreate the necessary neuropsychological bases to allow for good educational proficiency and interaction with others, and to enable speech therapists to work. I'd like to report on my personal experience, on my professional experience in the rehabilitation clinic that I work in here in Lazio. It's for developmental age patients, and we see younger and younger children coming into us. They're affected by DMD and BMD, and they show, obviously, pathological signals, but not fully. There are often very slight symptoms and signs. In this phase, in this very early phase, because it is not impelling or incredibly urgent to work on already patent and evident muscular symptoms, we can take the liberty, so to speak, to have a playful approach to the therapy and lay the foundations for a successful partnership between the child, the therapist, and the family as well. The ultimate goal, of course, being that of providing for a functional bodily identity on the part of the child. Regardless of how the pathology will evolve, it's important for us to see to it that the children become self-aware and are able to control their bodies and orient themselves in space and time and relate to people and communicate with the outside world. These goals are very important to us as therapists because often in rehabilitation, children, and this happened especially in the past, were taken on board when it became urgent and when it was very pressing to take action, which sort of disregarded the experience of the child and their self-perception. Instead, it's really important to have time to create this intermediate and crucial therapeutic step. This approach is really important, whatever the child you're dealing with and whatever the disease affecting the child.
because it is a way to access the inner world of the child, and that's what you need if you hope to be effective in your approach. And this, of course, we can do with the tools that we have at hand in neuropsychology, children's neuropsychology. Based on this, we can support the child or children as they acquire these skills. They probably would have acquired them over time anyway, but maybe not. And we can support them in maybe developing a balance, standing on one foot only, and then jumping, and then doing other activities. All these acquisitions are supported by the neuropsychomotor therapist through work that often involves merely motor aspects. For example, the children are asked to go through hoops or to climb over small bridges or to sort of creep along inside a tube. But they also involve other aspects of development because the children are asked to build something concrete or to organize physical space. They're asked to create boundaries and to respect those boundaries and the rules that are set. And they are also encouraged to learn to do a somersault, which means that they're developing some new movement. So this is a rather traditional neuromotor approach, but it does lay the foundations for better school proficiency, and they provide fertile ground for the work of other professionals like the speech therapists, and they can help address behavioral issues and speech retardation. In my experience, we gained excellent results with this approach with 2 children affected by BMD. One child came to see us at age 4, so as soon as a diagnosis was made. And that was the typical example of early management. And we supported the acquisition of motor skills according to a playful approach. And the family reported on the fact that the child was able to stabilize the previously acquired motor skills and didn't get as tired as he used to, and he became more self-assured. Then there was another older child who was 10 years old, and with him, of course, we did stretching work, and also, oh, by the way, initially, not only did he suffer from BMD, but also a learning disability, although his IQ was within the norm. But he tended to be a little bit hyperactive and impulsive and had attention issues. With him, we prescribed general motor exercises and also the triggering of working memory and functional exercises. The results were very good with this patient too, not only in motor terms, but also in terms of school proficiency. I'd like to conclude by thanking you for your attention and giving the floor to Dr. Serena Calabrese, who will illustrate to us how the work of the speech therapist is part of this team effort. Thank you.
Buongiorno a tutti, grazie mille.
Good morning. Thank you. I'm very glad I can share my experience with you. Over the last few years, I've been working with families with DMD and Becker diagnosis. Speech therapists have a special relationship with words. If we choose some words, we do it deliberately, and that is why I spoke of families with a diagnosis. Those who work in developmental age know very well that working with children means that you work with all the people participating in the personal, emotional, social, and cognitive and educational aspects of the child life. Well, a train needs rails to reach its destination, and the same goes for the rehab treatment of a child with muscular dystrophy. Well, similarly, we must develop it. We must have a two-tiered approach. So we work on targets. So the deficits, the existing deficits, and where we work, of course, one-to-one during this hour of therapy, but that must also be accompanied by the management of the family and of the school situation. Because unlike an adult that can modify his or her own environment, with a child we must sort of work upside down, because it is the environment where the child lives. The environment must somehow facilitate, become a sort of mediator for the acquisition and improvement of some skills. So the speech therapist is called when a multidisciplinary team has already been set up, and so so the speech therapist steps in with a diagnosis that adds to an already existing diagnosis, and that implies a number of problems, because working with this disease makes it necessary for us to try and help with the tools we have available. When the child comes with a difficulty that is a consequence of another disease, of course, the difficulty increases. In our range of tools, we have in fact a variety of tools. Possibly we should further increase the number of these tools, because also in terms of counseling, because in fact we do counseling whenever we speak with the parents, for example. It is an everyday fact. So the speech therapist must step in very early. As Rosie said, we need an early care of the patient, and the developmental neuro and psychomotor therapist must not only work in that area, but also in the psychomotor area, because in that area we can work and strengthen fundamental tools, and that not only to acquire education, reading, writing, and so on, but for developing language. I'd like to make a practical example. When the colleagues spoke of psychomotor psychomotor projects, well, the child is asked to develop working memory. Of course, speech therapy works on the working memory, but if I have the possibility of building on what someone else has already done, well, of course, I can work in a more direct way, and I can go straight to the objectives and to the deficits that can be solved. If this is not the case, of course, my work is much slower. And also a dialogue with teachers, and we hope to make all this very real, also apart from formal meetings, so as to have a better generalization of what has been discussed during these formal meetings, because then the different skills must be generalized. I mean, finding appropriate, personalized, tailored tools to transfer contents, but then these content must be exploited also outside the therapy itself, so in the social arena. And this type of work requires a precondition, and their support teachers for us are the necessary link, because what is being done in our one-to-one relationship to the child can then be extended to the classroom, especially if we are speaking of primary school. We are not only speaking of learning as such, but also the possibilities of socializing within a small group that is somehow an example of society. And now I would like to give the floor to the other participants to the round table. Thank you.
Grazie ancora. Abbiamo...
Thank you again. We've talked about the school several times and the important role that it plays within the context of a project that aims to provide comprehensive well-being for the patient and their family. And that's why I'd like to give the floor to a former support teacher and trainer, Giancarlo Onge, who will speak to us about his experience and the tools that school have at hand when working with these children, and families too, of course.
Bene, grazie per questo invito.
Thank you for this kind invitation. It would be rather difficult for me to tell you everything that our colleague announced, I would say. It's not enough to have 15 minutes. But I would like to speak about a young man whose name was Angelo, and I got to know him at school because I was a support teacher at the elementary school, which we now call primary school, but anyway. And he was affected by DMD, but nobody knew anything about DMD at the time, and so I like to remember that I met Angelo. I didn't meet with DMD, and through my experience with him, I learned that the first thing to consider in our classrooms is that we meet with students. They may have disabilities and pathologies, but each one is an individual. Each one has their individual dimension. So I don't like to speak of children with DMD, but children, period, because each one of them is a child, whether they're named Angelo, Maria, or whatever. And this, I think, cuts across all professions, because we should see the person before anything else. So, obviously, this is not enough. This kind of approach is not sufficient. We also need to have knowledge. And at that time, I didn't know anything about DMD. All I knew was the life expectancy of patients with DMD, because that's the only thing that people talked about. So we decided to establish a network, and that goes back to the title of this event. It can't be just a definition, though. It has to really be Well, it has to be implemented. It's got to be put into practice. It's difficult, but it's crucial. And we were able to do that to create a network together. We didn't have the medical aspect because there were no trials at the time, or there were very few anyway. But, we got families together, the social context, of course, some few medical suggestions, but that was that. The fundamental role, however, was played by Angelo's classmates. They made the difference because they adopted him, but they didn't have pity on him. We did not have pity on him because he was having a hard time. We helped him to bear to put up with what he had, and we tried to give a meaning to what he was doing. And after 15 years, Angelo got a degree from university. They should have given a degree to his mother too, because she really helped him a lot. She should have a degree ad honorem. But anyway, I think that these forms of synergy are ones that we need to really focus on above and beyond words. We really need to focus on facts and behaviors. Obviously, we also had the problem of who would take him to the bathroom. I would take him to the toilet at school. I never said that, I can't do this because I'm a teacher. Maybe I ran some risks, but that's what Angelo needed because he didn't want a female assistant or somebody to help him, a woman, because he was embarrassed. And I took him. Imagine that bathrooms were on the ground floor, so they were 30 steps down, so I had to take him in my arms and carry him to the bathroom. I hope that that's changed in the meantime, because that was a real architectural barrier. Anyway, we don't need new norms and provisions. We really need to implement the existing legislation, which tells us that we must all do our part, that we must all rally around one word, our joint responsibility in providing concrete answers to those that are having a hard time. And then personally, I really think that when we give answers to those that are having a hard time, we will be giving an answer to everyone, because we'll be better at everything that we do. We must all give our support. It's not just support teachers. We must all support each other. And when I say everyone, I mean that we have to take on board Article 34 of the Italian Constitution, which says that schools are open to all. They must be open to all. And there have been improvements. I mean, back in my days, when I went to school, there were no disabled children and there were no facilities. Instead, now we have disabled children. They're visible to us, as a result of which we've been able to improve our schools. We've made them more accessible. Had they not, been with us and visible, we would have just gone up and down the stairs, and that's all. So I do think we have significant legislation in place. For example, Act 104, but others too. And we really don't need any new laws. Sometimes people ask me, you're a support teacher, can you work with everyone? Well, unfortunately, there are still people who think along those lines, but what can we do? Let me just conclude by quoting a child from kindergarten. He's Moroccan, and actually pre-kindergarten, there was a workshop and a teacher made couscous. And at the end, the children had couscous and they said to their Moroccan classmate, "Did you like couscous?" And he said yes. And they asked him, "Is it as good as the one your mom's makes?" And the child said, "No, the one my mom makes is better, because my mom makes a layer of couscous and then below that a layer of tortellini." And this is a wonderful example of inclusion, I think, of being together, of respecting traditions and actually putting them together so that we can become better and more capable of responding to all needs. Thank you for your attention.
Grazie a Giancarlo Onder, e entriamo sempre più nel vivo cercando di capire che cosa accade poi. E quindi lascio la parola alla
So now the floor goes to Sabrina Parazzini.
Con cui spesso
she's a supporting teacher, and we have been working with her about the disease because of course the idea is to try and find the best possible way for us to cope with it. Good morning everybody. I would also like to thank Parent Project for inviting me here today and for giving me the opportunity to share my work experience with you. I'm actually a supporting teacher and work in primary school. I'm not a fan of mics, in case you're interested. But anyway, I have been working as such for the last 26 years or so. But 4 years ago, it got particularly richer because I met quite an exceptional boy. His name is Emanuele.
Ok. Allora, Emanuele.
So, Emanuele was 7 years old when he started the first year of primary school, and I was definitely not ready to deal with DMD, apart from some theory and a highly superficial reading of some news I Googled. So that was indeed a challenge for me. The boy did not like playing with other, boys, and he loved watching the washing machine, but I immediately loved him. I mean, I fell in love with him immediately. After some time, of course, I got deeper into my knowledge about DMD, and I realized that Emanuele had a cognitive heritage, if you wish, a cognitive potentiality that should have been helped to emerge. Of course, I became aware of the fact that my work could only be possible through cooperation. I mean, welcoming diversity is not enough, or being nice, to the boy. That is not enough either. You need to work together with the boy and other people through inclusion, orbiting around the pupil's potentialities. At first, it was very difficult. We had to know each other, of course. We had to know, I had to meet his family and know them. I also had to get to know the Parent Project Association too. But my colleagues, Valeria and Loretta, were on my side all through. Actually, they were with me when I started. After I had contacted some, of course, counseling centers and, DMD counseling centers, and therefore realized that much of the energy would be spent in order to build build social relationships of paramount importance to the boys' emotional well-being. And of course, the harder I worked, the more easily I realized that all Emanuele-oriented activities should be properly targeted because the language ought to have been the same as the one used with the other pupils, but of course methods might have had to be changed, and the dialogue should have been searched for with all the stakeholders. Well, of course, there are some aspects to be considered when we talk about DMD based on what truly characterizes DMD: learning problems, mood, habits. Actually, with Emanuele, one day is never the same as the following one. There's always this, imbalance moving around. So you are never allowed to, sit back. But Emanuele is definitely a very clever boy, the one that Lowenstein talks about. It's a modifiable kind of an intelligence. And indeed, I went back to study his publications. I decided he should make up my methodological choice. This is, the background of the methodology we have embraced in dealing with Emanuele. Even such a mood imbalance should be properly understood in order to allow Emanuele to live through his school experience as nicely as possible and as playfully as possible, if you wish, as Maria Montessori rightly said. Well, so far it seems like we have managed to share all the activities, even with Emanuele's family and speech therapist. We use concept maps or, recap patterns, videos, pictures, and we also change, teachers as much as possible with Emanuele, but that is not really what I would like to stress at this stage. After all, this has become quite customary in our schools. But the point is, how can Emanuele's work be made easier? Well, indeed, through the help of his schoolmates. And by the way, this happens certainly with the aim of having someone working on Emanuele's side, but actually the other way around too. For example, Emanuele knows how one's diaphragm should be used, how to approach music. He knows the meaning of a single word and tells us about it, and he's very good at making connections between words. Let me say a few words now about the need for Emanuele's teachers change all the time, namely with the one who teaches humanities, who teaches actually Italian language rather. And the idea is that inclusion should be an all-embracing approach, because if a pupil considers the support teacher as the only one there for him, that will not talk to him about inclusion. And the same goes, of course, for pupils. The class is indeed the one group that can account for the most precious gift for us all. Last year we wanted to help Emanuele to cope with the fact that one of his schoolmates was not that was no longer coming to school. So we had to invent some stories by somehow involving the whole class. This is very important. Peer relationships are of paramount importance, especially as the boys and girls grow up, because they get better aware of their own body, because they start teasing each other. So relationships are of paramount importance for the intellectual and social development of a pupil. And class dynamics change with time, of course, because indeed they keep growing up. Hence the choice for setting up smaller sized working groups that could also meet at home through a flexible teaching based on success stories and self-esteem stories, including sitting volley approaches and physical abilities. Now, of course, social relationships have almost become pervasive, because indeed the children have grown up and we feel like we have to start it all over again. Hence the need for doing something based once again on the dialogue with the family, for example. Actually, we should not mean this as something that we just give. We place as a burden on the back of the family members. Actually, that is not the case. So we want an operational working group to be set up. Operational, what does that mean? Well, it means that within this framework, an ecological approach should be embraced, which means an all-embracing approach, one that encompasses and includes all the various stakeholders through flexibility, so as to better respond to the boys' needs. Let me conclude with a remark. We are teachers, and therefore we are responsible for these children vis-a-vis their families too. We should not lag behind. We should actually try to be open to welcoming diversities in a constructive and moderate way. All stakeholders should be enabled to work together on a daily basis. Thank you.
Grazie a Sabrina Parazzini.
Thank you, Sabrina Parazzini. As I've said on other occasions, thank you for your emotion, for sharing your feelings with us, because that is an indication of the passionate way with which you approach your work. And now we come to the crux of this session, which is the testimony of families. I'd like to give the floor to Jean Crizia, who, with her husband, will tell us about their family experience. Good morning, I'm Crizia, Giorgio's mother. I'm not going to go into detail. I'm not going to tell you how one feels when dealing with the tragic diagnosis of DMD, because every parent involved knows what kind of chasm they fall prey to. And going back to our story, however, I feel like saying that we were quite fortunate, because in only 2 months, although they seemed endless at the time, we were able to get a diagnosis. Initially, we went to the Meyer Children's Hospital in Florence, and once we obtained the result of the DNA study, we were directed to the Nemo Clinical Center at the Gemelli Hospital here in Rome. After the first visit, we were contacted by Parent Project, and so we realized that we were not alone. We realized that there were many other families in our same situation. In addition to the medical part, we were greatly supported in our completely new life. Our life had been totally revolutionized. We treaded uncharted territory, and we needed to know how we could make it without growing discouraged. With the great commitment of, I'm really sorry, I'm getting a bit emotional here.
Abbiate pazienza. Dicevo, col grande impegno di crescere comunque 2 figli, mantenendo il più possibile la loro.
With a great commitment of raising 2 children. So Giorgio was in the front line of this battle, and Rachele, together with us, trying to overcome all the problems. In 2015,
L'indicazione era di
we received the diagnosis and speech therapy was suggested, and conceptual maps were provided to Giorgio in order for him to take stock of new learnings. After the DMD diagnosis,
maggior ragione
we were able to more readily understand the kind of difficulties and limitations that we needed to work on. Initially, we really had a hard time, but we didn't grow discouraged. And actually, we explained this to Giorgio, telling him that this was necessary to make him feel more self-assured at school. Unfortunately, during this rehabilitation time, he changed 4 doctors, which meant that Giorgio had to start from scratch every single time, and he felt very frustrated. Giorgio does not collaborate readily and right away, for those who know him, especially when he feels that he's being put to the test. And so even without the help of a specialist, we try to work on his self-esteem as parents, to the extent that that's possible. Physical degeneration has been slow so far, so we've been able to grow accustomed to the different steps of the pathology. And we live in a small city, so we were afraid of not having access to local support. And in time, we spent many hours having arguments with day hospitals and offices. We had to even have recourse to legal experts and lawyers in order to have access to what was due to us. We were able to also organize over the years a minimum level of rehabilitation. We turned to the Stella Maris Foundation, where we were greatly welcomed. With them, we have periodical follow-ups. For a year now, Giorgio has been involved in a physiotherapy treatment at a local facility. He has a gym and finally a hot water pool that he can swim in. Through this association, Stella Maris, we also had the opportunity to be supported as parents. We went through a program, and I think it was very useful in order for us to find a balance in the family. We took the time to stop, exchange, and listen to each other. As regards school, because of bureaucracy, unfortunately, we weren't be able to benefit from support teachers right away. But as of the 3rd grade, Giorgio finally was able to have a support teacher who followed him with the dedication, love, and professionalism up until 5th grade. Now he's in 6th grade, and unfortunately the educators are a lot more trained and a lot more professional than the support teacher. over the past few years, they've helped us greatly to come to terms with this and to come into contact with families like ours who are going through difficult times. For example, we were hosted as a family at the Dynamo camp. It was fun. It involved disabled children with their siblings. Everyone was equal and everybody did everything. They danced, they sang, they acted, they swam, they went horseback riding. There were many different activities amid medical and physical safety. It's a magical place, and it teaches both adults and children that it may be possible to live beyond one's boundaries. Giorgio hasn't been able to have this experience on his own yet, but hopefully he'll be able to soon. Then there was another association that we benefited from called Sportabili in Trentino in the mountains, and Giorgio was able to ski with us in the summer and go on rafting excursions. And there are many volunteers that alternate in the summer, and the wonderful thing is that many of them are parents of seriously disabled children, although I'm not really sure that it's fair to say that. Another wonderful experience was the trip to Sicily with Parent Project. We were really happy and satisfied because Giorgio, up until the summer, had not had any opportunity to meet with children and youth suffering from DMD. My husband and I decided to go on this holiday because we wanted him to get to know other kids, and we also wanted him to discover in a very natural way a whole new reality that we had kept him away from, I'm afraid. My husband and I today feel that we did a good job, although we still have a lot of obstacles that we need to overcome. We have always remained united in spite of our arguments sometimes, and we've been able to respond to the various needs of our family along the way. To other parents, with great humility, I advise that you live on a day-to-day basis and enhance the abilities of your child, and do ask for help when you're afraid of not being able to make it. And like I say to Giorgio, sooner or later we will find an alternative. I'm really sorry for being so moved.
Grazie.
Thank you. Thanks. And now the floor goes to Francesca and Elena. They will also tell us about their own experiences. Good morning, my name is Francesca, and on my left-hand side there's my husband. We are the parents of Alessandro and Eleonora. We are here today to talk to you about our experience. I am already a bit nervous, so I'm going to read out. Alessandro is a nonsense mutation, DMD son. We learned about that when he was 3 years. We got that as we learned that at the hospital, at the local hospital, and of course that was a problem for us all. We felt like that disease would change our life completely. That regarded us as single persons, as a couple, of course, and as the parents of the boy's sister, Eleonora. Since then, we've been going a long way to find a balance, but also to live with this terrible thing that had happened to us. So first of all, we asked a neuropsychiatrist to help us. Secondly, we met the Parent Project people 4 years ago. We attended our first conference, and then we chose the Gemelli Hospital in Rome as a center where we felt welcome as a family. Well, then we were diagnosed the learning disorder connected to short-term memory. We first realized that through the cognitive test that Alessandro underwent here in Rome at the Gemelli Hospital when he was 5 years old. We had actually started to realize that. Once I remember that at primary school, even his teachers had realized that he found it difficult to remember things. He just could not retrieve anything from his memory. And as he was doing his homework, he often said, I keep forgetting things, Mom. My head is empty. I'm a forgetful boy. Well, this has led us all to getting this correlated diagnosis. But we did not think that we could have problems also with his learning functions. We thought, therefore, that we had wasted time, that we were not doing enough. I personally was angry with myself. I thought I had always observed my children very carefully, but that was apparently not the case. So I had to realize that a parent is not an expert. Of course, there are drawbacks and limits, and I was not happy about that. But then, together with my husband, Leonardo, we realized that that was not helping Alessandro or indeed Eleonora. So we decided we should really try and work harder in order to listen to them, to their needs. We wanted them to feel at ease with us, so as to allow them to tell us how they felt about the real truth, the true truth, as Eleonora used to say when she was a younger child. So now our children started to ask questions, and sometimes they even got some answers. Sometimes Alessandro, as he was doing his homework, watched me as if I came from Mars. But then he came up with a question: "Can you help me? How can I solve this problem?" Well, that was okay enough, but I realized that I still needed the help of a neuropsychiatrist, and so I wanted to get a handle into his actual difficulties. So in the meantime, Alessandro became better aware of his own difficulties, and therefore we realized that he was much better aware of many more things than we thought. So he came on our side and helped us cope with a difficult situation. Well, that was a very important step because it helped us truly understand how difficult the whole situation was and how in need we were of help. So, he trusted us. He was not ashamed, not even when his sister was around. He decided to ask for help. So he was so patient with us grown-ups. My husband and I are very different people. We, of course, respond to the unexpected in very different ways, but he helped us come up of our fears, realizing that we might not be able to solve the problem on our own. Well, our experience certainly starts from clinical data, but the key component is Alessandro's placement at school. We tried to find local experts with whom to go deeper into this, and we actually detected two people, a neuropsychologist and a speech therapist. They were supposed to work together, and also they were supposed to be available to be part of a working team at the Gemelli Hospital, where Alessandro used to go on a regular basis. So they all told us and Alessandro that much of our memory could be made better. Therefore, some speech therapies exercises could help them. Aids could be used. Concepts might be practically experimented through other senses, such as the touch or the sight. So we needed an all-embracing approach. And as far as teachers are concerned, we also tried to help them understand Alessandro by envisaging some training courses on DMD, thanks, of course, to Parent Project. And then we detected those tools that could be best used at school. Of course, at the end of the first class of primary school, we realized that a support teacher could have a role. We needed help because in the meantime, it is very difficult to find the right balance to, not to consider us as parents, as ourselves as parents, as experts. So we've worked together with a psychiatrist in order to find that balance that I mentioned before. And anyway, performances should not be the key word. As for Alessandro, well, he has changed. Now, of course, he has a different work at school. And by the way, that does not regard only him. Actually, he now feels as a privileged boy because thanks to him, much innovation has been brought about in class that had not been envisaged until that time. Now, as a family, we have tried to work out something well balanced, hopefully orbiting around our son. We wanted each member of this group to work closely together, but Alessandro has always played a key and an active role in this. He knows, of course, the plan. He knows he needs to have a planning of what everybody is supposed to do, but he has certainly taken up the challenge, and he has started this new program with us all, and it seems like results are encouraging. But of course, this work must all revolve around the son. It must be sustainable. Some activities have had to be temporarily discontinued because we wanted everything to revolve around Alessandro, but at the same time we did not want to nullify what had been done until that time. So cum labor, work together with Alessandro, with the team. And by the way, there's another key element at this stage, that is the very close relationship that Alessandro has with all the people in and out of the family that have embraced our cause. This is very important, the notion of a team, because Alessandro needs to be helped now and all along his growth, as it goes with every other child. So a neuropsychiatrist must be on his side all along, and the same goes for the physical therapist. So it's a long way, and not only for Alessandro. And every single step entails something new and something difficult. Sometimes you feel like you have found a perfect balance. Everybody in the family has their own role. That's great. But then once you've almost got used to this, something unexpected comes up, and you feel like you need to revisit it all. So that's not easy at all. You may stumble and fall, but then you stand up again, possibly asking for other people's help. I believe we as parents can make a difference in showing how things can be done, in daily life. Alessandro is learning how to consider himself not as a slow boy, but as one who is maybe more introverted than slow. How can that be achieved? Well, the problem must be coped with. Projects must be envisaged. Teachers must be contacted. People must be involved. Experts as well as friends, because you need friends. You need to smile at what you're doing from time to time. That will change things, because the quality of life will change. Alessandro works hard, but he's okay when he knows that he needs speech therapy. He is not too unhappy about his stretching, exercising, for example. So we know that the family has a key role to play. The family is the carrier of a great value, because this is how we can help our children. So sometimes therapies, may be talked about in a given way, and this may help the child undergo them in a different way, because we need to believe in those therapies. This is the case of the boy as well as all of his family, and I will never stop doing that. I will keep doing things, and I will keep doing that in the right way, because this is a life that must be lived at the best. Thank you.
Ringrazio quindi tutti i relatori.
I'd like to thank all of our speakers, especially our families. We know how difficult it was for you to share our experience with us, so a very special thanks goes out to you. Because thanks to your testimonies and the contributions of our experts, we realize how complex it is to manage these young patients in the family when other elements of complexity add to the diagnosis. And we realize that the approach can not only be focused on the child, but that we also necessarily need to consider all the contexts in which the child lives. Because this multidisciplinary approach is conducive to the overall well-being of the child. It therefore has to be defined, but also reviewed and changed according to the changes that take place in one's life. And now I'd like to open the floor for any questions on the part of the audience.
O anche, i relatori a fare
Or the speakers too might want to make additional comments if they'd like.
Certo.
Mi sono dimenticato.
I forgot to say something because I was rather hasty in my presentation. Please do not stick to only acronyms. Behind the acronyms, we have children. Use their names. Often, I hear people say, in my class, there's a Down syndrome child, or there's a child with DMD. And then every now and then, they give you, numbers like, oh, look, we have an F-81, but an F-81 could be an aircraft too, right? So please don't settle for these simplifications. ECF talked about a holistic approach and the need to consider all dimensions. Please rebel against this. We don't have only DSA, but we have DSAs, DSIs, DSOs, DSUs, and DSEs. So we can speak privately if you wish to know what lies behind these acronyms, but I invite you not to use them, please.
Dica.
Il ragazzo ha 26 anni e le ricerche che hanno fatto, cosa...
Aspetti signora, dobbiamo fare la domanda al microfono così l'ascoltano tutti.
I'm afraid you have to speak into the microphone unless you want to speak to the person privately.
Dopo, quando finiamo la sessione...
After the session, you'll be able to speak to the person privately. Thank you.
Ci sono domande?
Are there any questions? Yes.
A proposito della scuola, mio figlio ha 16 anni.
I'm sorry, but we don't have a working microphone. There we go. I wanted to speak about my own experience as a parent. We must never give up, I think, because unfortunately, as somebody said, we do have existing legislation, and it's good legislation. We don't need to produce more laws. The problem is enforcement. My son, for example, he loved to draw. He wanted to go to art school, but in our city, Treviso, it's not accessible. When the various schools were presented, I said, is it accessible to disabled children? And they said, oh yes, we have a lot of disabled students. But then privately they stopped me and they said, you know what, it's not really accessible. And my son's friend, who was right there, looked at me and said, why didn't they say it in front of everyone? Were they ashamed? Yes. They were ashamed, I think, because things should be different in our country. I learned later that the dean of that school had made it a point not to make the school accessible to disabled students. So the school occupies different buildings throughout the city with stairs and no elevators. So anyway, my son said, does that mean I can't go to that school? And I said, yes, I'm sorry, that's the way it is. So we thought about something even better because sometimes things lead to good solutions. And we found another school that's better suited for him, that specializes in graphic art and communications. And so now he goes to this other school, and they use computer-aided design, so it's a lot better for him, I'm sure. He'll be able to find a job more easily. It's too bad, however, that this school, which is pretty large, has never had disabled students. When I talked to the headmistress, I said, you have no disability culture. And she said, yes, but we have a disabled child. Too bad that he was an autistic student and didn't go to class. So the faculty was simply not prepared to manage a student who didn't have a learning disability, but a physical one. But we really worked hard, and we were able to break through the barriers, not with all the teachers, unfortunately, because there are physical, but there are also mental barriers. The physical barriers here didn't exist because there were elevators and computers, but then the teachers were simply not ready. For 2 years, we fought. My son had an Italian teacher who gave him really bad grades, and he passed that year only because the other teachers and the headmistress acted as a team. Finally, I have to say, his teachers and professors understand him and try to help him in his difficulties, but it was hard. We opened a gap, really. We created an aperture because after him, there have been several disabled students. And this was confirmed by the headmistress, who said that her school was an example in their area. In the past, all the disabled kids went to one school, and that school became a ghetto, really.
Grazie. No, sì, frequentemente anche nella pratica del centro ascolto di incontrare situazioni molto simili.
Well, actually, this often happens to us at the counseling center. We know that this is an inclusion culture that still needs to be built up. Even in Rome, where we certainly have many disabled students, but we still have gangways that cannot support the electronic wheelchairs. Sometimes lab classrooms are not accessible, so there's a whole lot to be done still, and we know that we need to raise public awareness to that respect. We don't want that to occur again. Any other questions? Good morning. I have an 11-year-old son. Well, of course, the burden on our back has turned heavier than the one at primary school, which, by the way, goes together with what we do on a daily basis with my son, taking him to the swimming pool or the therapy exercises, class and so on and so forth. So homework is a bit of an issue there. For example, there's a time for physical therapy, one for stretching, one for speech therapy. Okay, what about homework? Well, quite often 24 hours are not enough, and sometimes we need to take him off, the playing time with his schoolmates. Otherwise, the homework will simply vanish. So, what to do? Well, people tell us you should give up something, but the point is I can't give up this, I can't give up that. So again, 24 hours are not enough. Thank you.
Le rispondo io, signora. Ma allora il discorso è sempre cercare di trovare un giusto
equilibrio. Let me answer. Well, of course, the point is to find the right balance. The right balance. Because sometimes there are your son's needs, medical needs, for example, but on the other hand, there's the need for him to do his homework. And what about the quality of life? Well, from my viewpoint, this is a key issue. So I often say that a boy might find it better or easier maybe to get in touch, with his classmates. After all, this might be better than learning all war dates by heart. My apologies to the teachers in the room, of course. But I mean, this is something I always do with my own daughter. I need to find something in between. I can see that, of course, the burden on your back is much heavier, but maybe 1 week you'll give up the swimming pool, the week later you organize, some time with his schoolmates and so on and so forth. Let me add something to that. When these operational working groups are set up, well, they are supposed to be operational, which means that the teachers must be made aware of the different engagements that the child must meet. So you might want to give up something 1 week and something else, the week later. But I don't know, maybe homework could be revisited in terms of quantity, provided, of course, that the curriculum is properly complied with. Well, let me add something to that. Talking. I mean, there are many people that you need to, get in touch with. at this stage of your life. Hence, the need for sharing your fears with all such stakeholders. I don't know, physical therapists or physicians. Of course, I am a therapist myself, so I believe that we may get in touch with the child in a better way, on a more regular basis. So finding a rule is a need for us all, I mean, for the planning of each one of us. However, there are some cases when a planning can be particularly reassuring, but we need to find the right framework for that. So first of all, let us talk about all the problems arising. Let us put them all on the table, because maybe some of these problems could find a different place based on a priority list that may change with time. 6 months ago, that list might have included some items. Today, the items may have changed. So let's try, to open up and talk to the people.
Faccio un ragionamento sullo spunto che ha dato la signora dei compiti.
About homework.
Direi che
I think
è una interessante riflessione sui modi di fare scuola.
Teachers can teach in very different ways. I mean, homework is not a must. Of course, a teacher may, give some homework. Other teachers may not. In the former case, the whole class is led accordingly. The point is that when we must cope with someone who has some specific difficulties, we need to think of that child in different ways from the standard ways. Blue eyes, brown hair, and that's it. The point is that whenever I need to approach a child, whether it's a boy or a girl, I must come up with a customized approach. Neuroscience is supposed to be part and parcel of a teacher's work. We now know that what was envisaged in 1975 seems to have somehow, written down the rules of neuroscience, because at that time the children were disabled but still saw their able-bodied mates at school. Well, they found that quite regular. So I mean that quality of life should once again be highlighted. When we talk about stairs, for example, this has nothing to do with the single disabled person, but with quality of life. So I think that even other parents should get pissed off, if you allow me, because we need this issue to become our issue, not the issue of the disabled child's family. You may go up the stairs and break a leg, even if you're not disabled at all. So the point is this: we need to consider the needs of someone who are having special difficulties. We need to consider this from a general viewpoint. We don't want disabled children's parents to be left on their own. And to conclude, let me just say that the word poor old thing, the phrase poor old thing, should be erased from our vocabulary. Maybe we should replace that phrase with another one. Let's start from your name, okay? What's your name? Let's forget about acronyms and use names once and for all. Thank you.
Abbiamo quindi l'ultima riflessione e poi chiudiamo la sessione.
Mi rifaccio a quello che diceva, all'esperienza.
So, As far as schools are concerned, again, actually, as far as all institutions are concerned, well, there are laws in Italy whereby such places must be accessible to all. Act 13, for example, envisages that all public areas must be equipped, public area units must be equipped so as to be accessible to disabled people. And if that is not the case of a given school, well, that school shall be shut down. So let's consider it all. Let's put everything into question, including the school administration. Let's speak up, because, this is not the case right now. Therefore, such schools are still open and are only accessible to not disabled people. In Forlì, for example, northern Italy, the kindergarten where my son used to go has changed 3 different places, 3 different buildings, so for different reasons. And apparently the third time there was no access to disabled people. So, we talked to the media, and after 1.5 years, the place was at last innovated with, say, only 3,000 euros after all. So if we don't speak up, this will never happen. Families are important because the child is truly at the center. Thank you.
Grazie. Avete visto sugli schermi i materiali che in questi anni Parent Project.
Thank you. On the screens, you've seen the material produced by Parent Project on these issues, and the pamphlet on diagnosis that you see right here, projected on the screen, and on school integration, which you'll find on our website and here. I'd like to once again thank all the participants, and I invite you to go back to the audience.
Scusa?
Sorry, but we don't have a microphone.
Nel 2016 è iniziato il nostro progetto supportato dalla Fondazione Parent Project, che ha l'obiettivo di studiare in maniera osservazionale ma longitudinale lo stato nutrizionale, la composizione corporea e le abitudini alimentari dei pazienti affetti da distrofia muscolare di Duchenne e Becker. In questo momento stiamo continuando il nostro progetto, che si concluderà nel 2020. Il progetto era articolato in 3 work package, quindi in 3 macro aree di lavoro. La prima era orientata a studiare le abitudini alimentari dei pazienti e della loro famiglia, e per questo scopo abbiamo implementato una piattaforma in rete a cui tutti i pazienti potevano e possono tuttora accedere, con una compilazione online di questionari che appunto indagano le abitudini alimentari e lo stile di vita non solo del paziente ma anche della sua famiglia. Poi abbiamo una seconda macro area di lavoro che ha coinvolto e che sta coinvolgendo centri che si occupano di distrofia muscolare, e in questo senso stiamo cercando di attivare una raccolta multicentrica di dati di stato nutrizionale attraverso un protocollo standardizzato per il rilevamento delle misure che sia applicato in tutti i centri che partecipano. Questo dovrebbe permetterci di arrivare a fare una stima aggiornata della prevalenza di sovrappeso, di obesità, di sottopeso nei pazienti affetti dalla distrofia muscolare. L'ultima macro area invece è più di tipo metodologico. I pazienti in questo caso si riferiscono al nostro centro per le valutazioni diagnostiche e strumentali e sono sottoposti a valutazione del metabolismo basale con la calorimetria indiretta, allo studio della composizione corporea mediante la DEXA e a un'indagine molto accurata dei consumi alimentari. Questo dovrebbe portarci a stimare capire qual è il reale fabbisogno di energia del paziente affetto da distrofia nelle diverse fasi di malattia e capire se ci sono delle alterazioni dello stato nutrizionale in relazione ad alcuni comportamenti alimentari. Alla fine del nostro progetto ci aspettiamo di produrre un piccolo manuale dedicato alle famiglie con le indicazioni dietetiche più adatte ai soggetti affetti da questa patologia.
Ciao a tutti, sono Luca Ducciaroni, un dottorando all'ultimo anno finanziato da Parent Project Onlus. Lavoro da 5 anni nel laboratorio del dottor Puri in Fondazione Santa Lucia e studio la distrofia muscolare di Duchenne e il trattamento a base di HDAC inhibitors, proprio come il Givinostat. Ho iniziato a lavorare nel suo laboratorio per il tirocinio della mia tesi magistrale, poi essendomi appassionato alla Duchenne ho deciso di dedicargli anche il mio dottorato di ricerca. Il mio progetto di dottorato è guidato dal dottor Puri e dalla dottoressa Silvia Consalvi, anch'essa quest'anno finanziata da Parent Project. Il mio è un progetto esplorativo che ha come obiettivo quello di comprendere il meccanismo di progressione della patologia che determina la resistenza al trattamento con il Givinostat. Il Givinostat è per l'appunto un farmaco in clinical trial che sta avendo dei risultati estremamente promettenti nel rallentare ed ostacolare la progressione della Duchenne. L'effetto terapeutico del Givinostat è però stadio dipendente. Questo significa che in qualche modo i muscoli distrofici a stadio tardivo della patologia diventano resistenti al trattamento. Il mio progetto di dottorato quindi utilizza un potente approccio bioinformatico che è capace di estrapolare le differenze del patrimonio genetico ed epigenetico. Questo è allo scopo di comprendere il meccanismo di resistenza al Givinostat che avviene durante la progressione della patologia e identificare quindi marcatori di resistenza utili a velocizzare e semplificare l'entrata in clinical trial e identificare eventuali target molecolari la cui modulazione possa o sinergizzare con il Givinostat oppure attenuare la resistenza ad esso. L'analisi bioinformatica è stata ultimata ed abbiamo identificato tutta una serie di funzioni biologiche modulate dalla progressione della patologia che gli HDAC inhibitors riescono a contrastare a stadi precoci ma non a stadi tardivi. Il progetto è nel suo terzo ed ultimo anno. Ringrazio a tutti per l'attenzione, se avete domande sarò felice di rispondervi aiutandomi con il mio poster. Grazie.
Abbiamo quindi visto altri 2 video.
We just saw other 2 videos of researchers financed by Parent Project, and now please help yourself to the lunch behind you, and we will reconvene at 2. We ask everyone to kindly be punctual because we've got quite a delay and we've got a very rich and dense program ahead of us.
Posso chiedere di iniziare a riavvicinarvi per riprendere le sessioni?
I'd like to ask you to please prepare to resume our deliberations.
Niente, non parla. Non va, vero? Di accomodarvi cortesemente. Chiedo anche il contributo di tutto lo staff.
Could you kindly take your seats?
Vorrei anche chiedere la cortesia se durante
I'd also like to ask you very kindly, if you wish to engage in a conversation during the sessions, please leave the room, because often you may not realize it, but you might be disturbing those in the room and the speakers. Thank you.
Bene, preghiamo tutte le persone che
I'd like to ask all those who are at the bottom of the room to kindly take their seats so we can start. Or if you wish to finish your conversation, could you kindly leave this plenary hall so we can get on with our work? Thank you. So, let's resume our deliberations. This session will no longer deal with the genetic aspect and the restoring of dystrophin, but now we're going to look to a new target, which is muscle and therapeutic approaches. So the first presentation will be delivered by Paolo Bettiga on Givinostat. He's going to give us an update on how things are progressing in that area.
Buongiorno a tutti e grazie.
Good afternoon, everyone, and I'd like to thank, as always, Parent Project for having kindly invited me to speak to you about the update on the development of Givinostat. This is the institutional slide, the disclaimer. Italfarmaco produces Givinostat, which is not on any market in any part of the world. So it's an experimental drug, and this presentation is for scientific and educational purposes. The first thing I'd like to share with you is what Givinostat is all about. It's a potent deacetylase histone inhibitor, HDAC. We've been working on this for years. We've completed 23 clinical trials and 4 are under the way. The result of all this is that we have quite a long-standing experience that can help us determine the effects on man, on humans. We've treated more than 500 subjects with this agent, and many of them are children. We started out, when I presented this for the first time, we only had 31 children with juvenile arthritis, and now we have 70 children with DMD, of whom 16 have been in treatment for over 5 years. With regard to the drug's tolerability, Here are the main adverse events or effects. So one is the platelet reduction. This is actually a pharmacological effect of all HDACs. So we know that this has an onset very rapidly, and this is the reason why we have a very close control of hematology in the first few weeks of treatment. But the reverse is true as well. If the dose is too high and the platelet level goes way down, it's sufficient to discontinue the drug for a few weeks and platelets level go back to normality. There are adverse events related to the GI tract, especially diarrhea. But in DMD, it's actually quite the opposite. They have stypsis more than diarrhea, so this never limited treatment. And with the increased findings and the increased number of patients in treatment, we also observed a reversible increase of triglycerides in some patients. And so when treatment is discontinued, once again, the data goes back to normal. Over the years, we also have gained a better understanding as to how givinostat works in DMD. And I hope that we have also improved our way of getting the message across. You've already heard several times in the earlier presentations that the muscle tries to repair whenever there is damage that occurs. We have these so-called special cells that usually are at rest, they're dormant, but when it is necessary to repair the muscle, they become active and they restore the muscle's normal conditions. In DMD, of course, the main problem is the absence of dystrophin, which involves or entails two important things. First of all, the muscle is more prone to damage because dystrophin isn't present, and so it doesn't play its role as a shock absorber. Moreover, the repair muscle no longer works. The repair system no longer works well either. So at the end of the day, we're left with scars and fat and not a repaired muscle. Givinostat does not restore dystrophin, but it acts on the repair system. So what we expect givinostat to do is to restore an adequate repair system with a lower formation of scars and fat and a greater formation of muscle. This is a summary of the clinical development program for muscular dystrophies. Study 43, which is a phase 2 trial that I've presented a number of times. It's come to completion now, and the youth that were in trial 43 have entered trial 51, which is our long-term trial, in which we will follow up the children and youth enrolled in other trials up until the approval of the drug. The phase 3 trial, which we hope will give us good results so that we can register the drug, is called EPIDIS, and it's underway. In DMD, we're looking at another molecule too. It's in the preclinical trial phase. We also have a trial for BMD, Becker's muscular dystrophy. More details will be provided on that tomorrow afternoon in the session devoted to BMD. So what are the data that we've obtained thus far? Well, we've studied the drug in the ADX model, and we've observed a dose-dependent effect on histology and function. We then completed the phase 2 trial 43, in 20 children treated with givinostat, and at the end of the trial, we have been able to observe a significant effect at the level of muscle biopsy. There is an increase in the muscle component, a reduction of necrosis, fibrosis, and fat. And both findings have been published. What we can do with the children and youth treated in Trial 43 and that received treatment in February, well, that have been receiving treatment for over 5 years. These data are from the last interim analysis. Anyway, from this, we can look out for signs when it comes to the milestones of the disease. The first milestone, of course, being the loss of the ability to walk. And what we've observed is that the median age on the loss of deambulation in our trial is 14.5 years. And we wanted to determine whether we can use published data to have a comparison. And it is possible because recently synergy data were published. This is a very large natural history trial conducted in the US. And it looked at treatment with steroids. The patients with trial 43 were administered a combination of steroids plus givinostat. But anyway, with only steroids, the median age at loss of ambulation was 13 years, almost 13 and a half. So now we will have an additional year of treatment, and so we'll be able to establish whether or not that finding is confirmed. We must also look at other important parameters like respiratory function. Every year we did spirometry on these children, so we do have a couple of standard findings to evaluate respiration, breathing, and FVC. And we can also measure the peak expiratory flow. As regards the FVC, we have an annual decline of 2.3%, whereas in looking at the natural history trial data that have been published, we have a percentage that is between 4 and 5% per year. For PEF, we have no decline. So this is a very positive sign that makes us hopeful. Let's look at the clinical trials underway. This is the EPIDUS trial. It's a phase 3 trial. Hopefully it'll give us good results so that we can register the trial. It's a randomized placebo versus control trial. So 1/3 of the children enrolled in the trial will receive placebo. And the treatment will last 18 months. As you can see, we measure safety and tolerability and function every 3 months. And at baseline, after 12 months and 18 months, for a group of about 100 patients, we use MRI. An important point is that in this case too, as happened for trial 43, when treatment is completed after 18 months, the patients enrolled in the study will access trial 51, where they will be able to receive givinostat up until the time the drug is marketed. As to the inclusion criteria, children aged from 6 years onwards, any genetic defect is compatible. They have to be able to walk, they have to be able to go up 4 steps in at least 8 seconds, and they have to be able to get up in at least 3 seconds and less than 10 seconds. They have to be under stable treatment with steroids for about 6 months, and for the group that will undergo MRI, of course, they mustn't have any problems undergoing that, like claustrophobia. And then there are exclusion criteria that are related to other experimental agents, for example, or drugs that can interfere with the parameters evaluated in the trials. CAF contractures, and of course, the surgery has to be planned, and or if surgery is planned, and unsuitable health conditions. That's an exclusion criteria. Because of the way this drug works, the children who are enrolled in a trial of this kind can go on to experimenting with gene therapy if there's any that comes out. The drug is taken at meals in the evening and in the morning, and this is the list of procedures that happen during the trial. Of course, we need to have a written informed consent. We have to have vital signs, the ECG, spirometry, and functional tests. And of course 3 MRIs at 0, 12, and 18 months. One important aspect is this. MRI centers are only 2 in Italy. So all expenses connected with travel to undergo MRI are reimbursed. Plus, in the initial phase, where we have frequent blood tests taken, we've got a system of nurses that go to the home of the young patients to take the blood test. And finally, at the end of the EPIDIS trial, the patients can be enrolled in the long-term trial. These are the participating centers. The trial is ongoing both in Europe and in the United States, and here you've got a list of facilities where the trial is run in Italy. Trial 51, I described it very quickly to you. Basically, it's a long-term safety study accessed by subjects who participated in other trials with givinostat. So right now, the ones who are in this trial come from the phase 3 trial, and the ones that will be enrolled next will be coming from the EPIDIS trial. One last to mention is trial 53 for BMD and Becker's dystrophy. 1/3 of the group receives placebo. The treatment in this case is 12 months long, and there are 2 centers. One is Professor Comey's center in Milan, and the other is Professor Nick's facility in the Netherlands. The goal is to replicate some of the results that we obtained in trial 43. So the first thing is to evaluate the muscle biopsy and see if we get the same histological effect, and then this will be corroborated by MRI findings, and as always, of course, there will also be a functional assessment. The inclusion criteria are shown here. The most significant are shown here. There is no restriction on genetic mutation. Age is included between 18 and 65 years. The heart also has to function properly still. I'd like to conclude by acknowledging all patients and all the foundations that have helped us in performing the trials. Thank you.
Bene, grazie al dottor Bettica.
Thank you, Dr. Bettica. You'll come back to us for possible questions, I hope. On we go with another approach. Anti-myostatin. And I'd like to call upon the representative of Roche to speak to us about this new drug under experimentation right now.
Buongiorno. First thing I should say is that I'm not Roxana Dragici, as you may see in your program. So thank you to the organizers for inviting us to this wonderful event. And I'm very honored to talk about our RG6206 program, an anti-myostatin in Duchenne, on behalf of my colleagues at Roche. So first off, what is RG6206? So RG6206 is an investigational molecule being developed specifically for people with Duchenne. It's been specifically designed to bind to a protein called myostatin and limit this protein's function. This drug is administered at home on a weekly basis by subcutaneous injection, and it's actually able to be injected in one of 3 different locations: the abdomen, the thigh, or the back of the arm. What is myostatin? So myostatin is a naturally occurring protein that is produced primarily in skeletal muscle cells. And the function of this protein is to stop the muscles from growing too large. So it's thought that, in fact, it's been shown that limiting myostatin can increase muscle size. So we believe, and others believe in the field, that blocking myostatin could increase muscle growth and muscle strength in children with Duchenne. So if I were to summarize the RG6206 program in 3 bullet points, it would be as follows. So I've mentioned already RG6206 is an anti-myostatin adnectin, and that makes it a little bit different from some of the other anti-myostatins that have previously been studied, which have been primarily monoclonal antibodies. We also have accumulated quite a breadth of clinical experience so far and continue to collect clinical data. We've completed a phase 1b/2 study of 43 boys with Duchenne, and these boys are all in open label treatment at this moment and doing well. And we have an ongoing pivotal study called the Spitfire study in boys with ambulatory Duchenne, and this study is actively enrolling. So far, RG6206 has been studied in 4 clinical studies. There's been 2 phase 1 studies in healthy volunteers, the initial SAD and MAD study, and we've also recently completed a study looking at injection or site of injections. This was the study that allowed us to give flexibility to the injection site. And so we treated a total of 103 adult volunteers. And I should also add, in our initial study, we were able to observe muscle size increase in those healthy volunteers. Our second study, or rather our first study in Duchenne boys, was the WN40226 study. As I mentioned before, the primary part of that study is completed, and there are some 40-odd boys who continue in open label extension. And I'll be sharing more information about the results of that study in a moment. And then finally, as I mentioned also, we have our pivotal study called Spitfire. This is a global study, a safety and efficacy study called, as I mentioned, it's called Spitfire. So more on the exploratory study in boys with Duchenne. So currently there are 41 boys who are now aged about 8 to 13 who are still enrolled in the study. The placebo-controlled period was 24 weeks, during which time we studied 3 different doses of RG6206. We have now studied out to actually 120 weeks or more of treatment, and we're analyzing those data right now. And the boys are, actually, as of the last time we reported these data, none of the boys had lost the ability to walk. So as I mentioned before, the primary objective of this study was safety and tolerability. And certainly within this study, we did not observe any safety concerns, including injection site reactions or immunogenicity, which means none of the boys developed allergic reactions, and we didn't develop antibodies to the drug. The pharmacokinetic profile, or the levels of drug, were as expected, and we also had excellent target engagement. So we were trying to lower the myostatin levels to a very low degree, and we achieved that very nicely. Like the healthy volunteer study, when we looked at neuroimaging, which is DEXA scans, as well as MRI of the right thigh, we noticed that, or we observed that the muscle size did increase by around 3 to 5% compared to placebo. And then finally, with respect to functional endpoints, we did look at these in an exploratory way. And while we did not see any significant change in function, as assessed by time function tests, 6-minute walk distance, etc., at 72 weeks, this was a very small sample size, and the study was not designed or powered to demonstrate effects on these exploratory endpoints. So based on these encouraging data, we moved on into our pivotal study, and this is called WN40227, and more recently renamed the Spitfire study. And this is a study where we're trying to understand safety and efficacy of this molecule. Patients are randomized to either our drug or placebo. We're looking at 2 different dose levels, which means that twice as many boys will be getting the active drug as compared to placebo. After the 48-week double-blind placebo period, all patients move into the open-label extension period, where they will continue to get drug until the time the drug is hopefully approved. And just another note is that all patients have to be on corticosteroids coming into the trial, and they need to remain on stable doses of corticosteroids for the duration of the trial. And we're hoping to enroll 159 patients with Duchenne. So what are some of the key inclusion and exclusion criteria for this trial? Well, first thing to mention is that patients with all Duchenne mutations are eligible, and they have to have a diagnosis of Duchenne that is confirmed by medical history and by genotyping. They must be able to ambulate without assistance and be 6 to 11 years of age inclusive at the start of the study. They need to weigh 15 kilograms or greater, be on corticosteroids, and also be able to do the 4-stair climb in 8 seconds or less. And finally, because the primary endpoint is the North Star Ambulatory Assessment, we want patients to have at least a score of 15 at the time of screening. A few of the key exclusion criteria are that, firstly, they can't have any behavioral or mental issue that would affect their ability to complete the required study procedures. They can't previously or currently be taking medications like androgens or human growth hormones. Unfortunately, they can't be on a ventilator during the day. And of course, they need to be able to tolerate and be able to have blood samples collected and have an injection under the skin. So the primary endpoint of this study is the change from baseline in the North Star Ambulatory Assessment total score at week 48 in the RG6206 arm versus placebo. Some of the secondary endpoints include change from baseline in 4-stair climb velocity, stand from supine velocity, 10-meter walk or run velocity, the PODC. We're also looking at measures of strength, the 6-minute walk distance, and finally the stride velocity 95th percentile as measured by Actimio, and I have a slide to discuss this a bit further. And of course, we'll be looking at safety in terms of lab abnormalities and adverse events. So I think everyone in the field recognizes that many of the in-clinic endpoints are suboptimal for picking up changes or be able to understand if a drug works. And so we at Roche have really taken a look at what we could measure outside of the clinic and what could be more patient-centric, what could be more sensitive. And one of the endpoints that we are looking at is something called the stride velocity 95th percentile, as measured by Actimio, and I know other studies are also using this modality. So basically what it is, it's a wearable sensor that goes around the ankles, and this records real-world ambulatory measures with high accuracy. We're able to measure the stride velocity, the stride length, as well as things like the distance walked per hour. And so this device objectively detects these different gait parameters and the ambulatory capacity in a continuous manner every time the patient walks or climbs stairs. It measures things in the real-world setting, and it's objective and independent from motivations or other confounding factors. And I'm also pleased to say that recently, this outcome measure was accepted by the EMA as a secondary endpoint in pivotal trials for regulatory decision-making. So this study is a global study, and as you can see from this map, we're enrolling in North America, Argentina, 8 countries within Europe, Australia, and Japan. Within Italy, we have 4 sites, 1 site in Messina, 1 site in Rome, and 2 sites in Milan. Now, just before thanking everyone, I just want to note that this study is actually enrolling very well. Of the 159 patients, we have over 100 patients enrolled, and we are optimistic that we will complete enrollment sometime in the middle of this year. So there's still opportunity to participate, but that window is in fact closing soon, which is great news for the trial. So now I would very much like to thank all of the families and patients who take part in clinical trials in general and our program in particular. Thank you.
Proseguiamo. Chiamiamo Michael Binks.
I'd like to call on Michael Binks now, please.
Thank you, Chairman, ladies and gentlemen. I'm delighted to be back to give you a follow-up on the Pfizer anti-myostatin program that I think I first presented here in 2015. And it's great to be finally able to show you some data that have emerged over the last 6 months with this study. I would like to at first acknowledge some key efforts on our team to pull this analysis together. In particular, Shannon Marafino, who's been the clinical lead for this program, has worked tirelessly and with great enthusiasm and integrity to ensure that this study has been conducted to the highest levels of quality possible. I'd also like to acknowledge Dr. Lawrence Charnas, who's a pediatric neurologist who's joined our group at Pfizer, who not long after he joined us had to be locked away in a darkened room to ensure that nobody else was able to see the data to maintain the blind in case we were moving forward. For many weeks, poring over many thousands of data tables. So for both of you, many thanks. So we announced in August of last year that we were terminating 2 clinical studies studying domagrozumab, which we had been exploring as potential treatment for Duchenne muscular dystrophy. The phase 2 study didn't meet its primary efficacy endpoint, which was the mean change in the 4-stair climb time in patients treated with domagrozumab for 1 year. Compared to placebo. In addition, the totality of other evidence, including multiple secondary endpoints, did not support the existence of a significant treatment effect. The studies were not terminated for any reason related to safety. So since that announcement, we have been terminating the study, bringing all the patients back in for a final assessment to ensure that we have final efficacy and safety data recorded in our study database. And that database has now been locked just a few weeks ago, and full analysis and reporting of that data set will be complete later this year. So the analysis that I'm showing you is not necessarily the very, very final one, but based on data snapshots that were taken last year. Our intention is to publish the results in full through clinicaltrials.gov, UDRIS clinical trials database, on our own web portal, and in scientific journals. We're currently working with our principal investigators to pull the data together into manageable scientific bites that will be worked on through this year to get all this information into the scientific literature. So despite this negative outcome for the program, the data collected from this study, from these studies, is really of high quality and will support the development of new therapies for DMD. I'm not going to spend long introducing myostatin. I've talked about it many times, and you've already just heard a brief introduction. This is a monoclonal antibody to neutralize myostatin. As you've heard many times, myostatin is a local muscle growth regulator. We had data in the MDX mouse system to show inhibition of myostatin caused a significant reduction in the level of fibrosis and fatty infiltration, and had data in multiple species on increasing muscle mass. So this was the study that we conducted, in which we looked at boys aged 6 to 16 who were still able to walk and who had been on a stable dose of glucocorticoids. They received a monthly 2-hour intravenous infusion and study visits for functional assessment every 2 months, with an imaging regime that repeated MRI and DEXA scanning every 4 months in accordance with the dose that had been treated in the prior 4 months. Boys entering the study were randomized to one of 3 groups, 2 of them receiving increasing doses of drug over the first year, 1 receiving placebo, then switching over to a dose titration. 1 group receiving a high dose of the drug for a period of 16 months. And then boys moved on to an open label extension study, which was also just terminated. So this study was run at 31 sites in 8 countries. We screened 161 boys, of whom 120 successfully enrolled and received this treatment. 59 boys, in fact, were enrolled into the open label extension and had completed a full 2 years on the program. So this is the demographic table of information that shows you the characteristics of each group at baseline. Very similar between the groups. Some small differences in weight and height that weren't significant. The majority of boys recruited into the study were in the age range of 7 to 10 years. First of all, this was a safe drug. We didn't see any safety concerns at all. Here, in terms of reported adverse events, they were evenly distributed throughout the groups and were generally mild in severity. We had 5 subjects who were noted to have serious adverse events, usually involving an admission to hospital, but none of these were thought to be causally related to myostatin inhibition. We had 1 patient who discontinued the drug in the first year, but this wasn't thought to be due to domagrozumab. And then we had 6 serious adverse events in the open label extension study, again, not thought to be related to medication. There was 1 event involving a fracture and fat embolism following it that resulted in the death of 1 boy. So I've said the primary endpoint was not met, and this was the forced-air climb. We looked using several different methods using different statistical models to see whether there was anything below our primary endpoint that might be correctable with statistical testing, such as testing the forced-air climb velocity rather than the time, which has some advantages. And looking at a logarithmic correction on the data distribution. But although these tended to point to a small favorable effect, none reached statistical significance. We did a comparison over 2 years with an external natural history-based control group, and were unable to convince ourselves that there was anything going on there in that comparison, even though the numbers were quite small. With the North Star total score, we saw a directionally favorable response, so a trend towards an effect, but this didn't actually reach statistical significance. And our other measures, which included the 6-minute walk test, the forced vital capacity, the upper limb pull, time to stand, 10-meter walk, run, and strength, showed no trends towards a beneficial functional effect. Now, I should mention that we did measure the pharmacokinetics and the pharmacodynamic activity of the drug, and it behaved as we had expected from a distribution point of view in terms of pharmacokinetics, and we were able to demonstrate a greater than 99% inhibition of the mechanism. I missed one bullet here, which was on the patient-reported outcome, the PODCI, which is a sort of quality of life measure, where we saw some directionally favorable trends on 2 of the scales in the PODCI, but neither of these reached statistical significance. So I think this is what you would describe as a flat line response. So just to orientate you, this is the difference between the change in baseline, from baseline to week 49, that is the end of a year's treatment, with the placebo signal subtracted. And you can see here there's a statistical test, the p-value of 0.9, which suggests that these are in fact identical. When we looked out over 2 years, similarly, there was no evidence of any beneficial effect on the four-stair climb. When it came to the North Star, it did look as though there were a trend here, and we were excited to see this. The North Star performed in our study rather better than we had anticipated based on variability estimates prior to initiating the study. And we did perform some additional analyses called post hoc analyses, so we hadn't planned for them, so they would never be, it would never be possible to use them in a regulatory submission, where we looked at the time it took for loss of particular functions to occur in each individual North Star domain. And using that approach, we did identify a statistically significant difference, but it's not terribly helpful to us in this environment. But for future studies and for the development of endpoint analyses, I think it's an important piece of information. Okay, lastly, I just want to talk a few minutes about the MRI that was conducted in this study. I mentioned that we had MRIs conducted every 4 months, focused on the thigh, measuring muscle volume, total thigh muscle volume, T2 mapping, which is a T2 relaxation time, which is a particular MRI parameter, and an estimate of the fat fraction in the muscle. And I do want to shout out Sarah Sherlock here, who's our imaging lead specialist, who pretty much single-handedly put together a series of protocols that can be conducted in any MRI scanner at any hospital and ensured that there was a centralized analysis that conducted to a very high level. And this was conducted in 31 centers, 120 patients, with very, very little missing data. And it's really a major achievement to have acquired that data. And in support of the emerging literature around MRI, it does seem as though, at least in this experiment with this mechanism, we were able to impact and detect a change on the T2 relaxation time of a couple of milliseconds. Now, what that magnitude of change means is open to debate. It clearly wasn't enough to correlate with a clinically important signal. But nevertheless, it demonstrates the potential to measure in a global multi-center study high-quality MRI endpoints. And I think we'll be seeing more of these types of methods in the future. I'll say briefly that we included DEXA imaging in this study really as a quick and cheap way to measure lean body mass. And again, we were expecting to see a signal here, and there was a trend in the right direction, but it didn't reach statistical significance. However, if we look just at the arms, or just at the arms and legs together, or just at the arms, we could see a statistically meaningful difference. This is the total DEXA score, not quite trending in the right direction, but not different from placebo. So this phase 2 study didn't meet its primary efficacy endpoint. The sensitivity analyses didn't help us. The totality of evidence looking at multiple other endpoints, our secondary endpoints, didn't support a significant treatment effect. There were no safety findings of note, and further exploratory analyses are ongoing in collaboration with our principal investigators. So despite the disappointment, we think that the quality of the data set will benefit future studies, both for us and for the community as a whole. We will be, once our investigators have analyzed and processed the data more fully, we will be making this available to the DMD community. And there are a number of areas in particular where we think this data set will be strong around liver biomarkers, cardiac biomarkers, and the MRI. And we're already learning from this data set in the design of our gene therapy pivotal studies. So I thank you very much for your contribution, for your contributions to this study in supporting the design and supporting the recruitment at the 3 sites in Italy who recruited for this study. And would particularly like to thank Parent Project Onlus for their input into this study and for inviting us here to update you on an annual basis. Thank you very much.
Thank you, Mr. Binks. Diciamo che purtroppo non è mai
Well, unfortunately, for us families, we never seem to get good news, namely when such trials as this one does not provide us with the expected or hoped for results. But the important part is to try and understand what has not worked out well in order to improve, of course, to move towards some improvement at least. So defeats should not only be viewed as defeats, but rather as a way to reset, if you wish, what we are doing. Now, having said that, let me call upon Dr. Thomas Meyer to talk to us about Raxone.
Thank you. Good afternoon, ladies and gentlemen. It is a great pleasure to be here again, and I would like to update you on new data that we have acquired in the past year or so. And these are long-term efficacy data with idebenone on respiratory function outcomes in patients with Duchenne muscular dystrophy. And the special thing about these data are that these are real-world data. They were not collected in a clinical trial, but they were collected during routine clinical care where physicians were treating patients. And this is something that we probably will see more often going forward as regulators demand evidence also from real-world settings. So the agenda I would like to cover today is following. I would first of all set the scene by providing you some background information on the medical need for effective treatments of respiratory illness in advanced patients with Duchenne muscular dystrophy. Then I will very briefly, in only one slide, summarize previous data that we have collected in a randomized placebo-controlled trial, the DELOS trial, which was the starting point for longer-term data that we collected from patients that completed the trial and were treated with idebenone up to 6 years in clinical practice. And these data are relevant because they teach us about the long-term efficacy potential of idebenone on respiratory function outcomes in this advanced patient group. So, unlike other presentations you heard today, I am focusing on a different patient subgroup. These are the older patients, many of those already lost ambulation, and what they experience is a progressive loss of respiratory function. This typically starts to be clinically relevant at early teenage years, and within a period of about 10 years, patients linearly decline in respiratory function until they acquire or require assisted ventilation. This is initially during nighttime, but eventually full daytime ventilation. So this is the target that we address with our treatment idebenone. In the past, we have run a randomized placebo-controlled trial, the so-called DELOS trial, over a period of 12 months. The data have been published, and I have presented the data in this very same conference already 2 or 3 times in the past. So I will not spend much time in summarizing those. Suffice to say that idebenone slowed the decline in respiratory function in the primary endpoint that was peak expiratory flow, or PEF, and was statistically significant between placebo and active treatment. And that's shown to the chart on the top right. The gray line shows you the decline over 12 months in placebo, and the orange line shows you a relative stabilization, which was statistically significant at month 12 from placebo. We have also found consistent treatment effects for inspiratory function. This is inspiratory flow and inspiratory flow reserve, and the global respiratory function forced vital capacity, or FVC. Also of interest was the observation that patients treated with idebenone over a period of 1 year had a reduced risk of bronchopulmonary adverse events. These are airway infections and other complications, which impose a medical threat to patients of advanced stage. We also saw that these patients needed less systemic antibiotic treatment, and there was a lower risk of hospitalization due to respiratory complications. So this is what we knew from the phase 3 DELOS trial. But as you go to the next slide, long-term efficacy data would be desirable to eventually decide on the benefit-risk balance of such a drug like idebenone for advanced patients in a chronic disease stage. Now, collection of real-world evidence can provide such long-term data from patients, and as we said before, will be required from regulators more and more in the future. Certain countries in the European Union allowed the continuation of treatments of the patients who were previously in the DELOS trial under national laws and regulations. And this was, as I said, this was not a clinical study, it was not an extension study, it was just we provided under expanded access program, free of charge, medication to patients. That was in Switzerland, Germany, Netherlands, and Belgium. So we provided this, and now we have planned to collect these data in a prospective manner. So we wrote a protocol, we wrote an informed consent form, and a statistical analysis plan. We went to the hospitals, we collected the data, entered into a database, and analyzed them. So today I present the data from 18 patients that were treated up to, on average, 4 years, up to 6 years after the completion of the DELOS trial. Now, the first thing that we wanted to ensure is that these 18 patients, which are a subgroup of the DELOS trial population of 64 patients, is not a specific subgroups of patients much younger than the rest or less advanced in disease or any special subgroup that could hinder us in interpreting the data. So one of the things we did is we looked at the baseline characteristics. So they were, as we can see, of similar age as the DELOS population. they had a similar stage in the disease, the age at loss of ambulation was comparable to the entire population of Delos, and the respiratory function was also comparable. This and other measures, which I'm not going to show you today, demonstrate that the subgroup of 18 patients that we followed up for several years is representative of the entire population of Delos patients, which was the basis for the entire study. Now, this is actually what happens. So these are 18 lines in color, each line representing one patient. Orange color is the time when they received idebenone treatment, and gray color is the time when they received no idebenone treatment. This could have been in the placebo group of the Delos trial, or that they did not start immediately to be treated in the expanded access program with idebenone. And you see that the timeline goes out up to 7 years or even more for some of the patients. On average, the treatment duration with idebenone indicated in orange color is 4.2 years in this group. And with this, we collected a total of 76 patient years of exposure data on patients on idebenone. The primary endpoint was the question, what happens to patients that are not on idebenone initially, and when they come on idebenone during this expanded access program, what is happening to their respiratory function? So you see the switch from off to on idebenone treatment. Before the expanded access program, the mean change in forced vital capacity per year was more than 7%, a very steep decline, whereas it was reduced by about 50% when they came on treatment. So switching from off treatment to on treatment reduced the annual rate of decline in forced vital capacity. We have similar data for peak expiratory flow, but in the interest of time, I only show you the forced vital capacity data. Now, this can be perhaps more demonstrated more illustrative in the following way. These are individual patient lines. Each line that you see there is one patient, and you see the slope, that is the annualized rate of decline before they came on idebenone on the left and after they came on idebenone on the right. And you clearly see that the initial steep slope is flattening, and any reduction in this decline is a clinical benefit. And you see also that this is not driven by one or two outliers. This is the entire cohort of patients that reduced in the slope. So clearly we can demonstrate that switching patients from off idebenone to on idebenone slows the rate of decline, which is of therapeutic benefit. We have also then asked the question, now, is that really true over the entire observation period, up to 6 years? And the analysis we have done was the following. So we looked at 2-year bins. So year 1 to 2, and 2 to 3, and 3 to 4. And we looked at the annual rate of decline, again in orange, for patients on idebenone. Now, we needed a comparator group, and the comparator group are data from the natural history Synergy dataset, and we extracted from this 400 patient dataset those patients who matched with respiratory function the group of patients that we're looking at in each of these bins. So for instance, in year 1 and 2, you have 18 patient data under the orange bar, and you have 62 patient data under the black bar. And you clearly see that year on year, every year, the rate of decline in the idebenone treatment group is less than a matched natural history group. And you also see that the effect is maintained. If anything, over time, the effect gets larger, because you see that in the years 5 and 6, the annual rate of decline is actually lower than it was in year 1 and 2. So this provides further evidence that the treatment effect with idebenone is maintained year on year up to roughly 5 years of observation period. Now, I told you before that one of the observations we had in the Delos phase 3 trial, that patients on treatment had less risk of experiencing airway infections and other complications. We see the same again in this long-term follow-up. And you see again up to 5 years of observation on the x-axis, and you see the risk or the events of bronchopulmonary adverse events in gray, untreated patients, and in orange, treated patients. And you clearly see that the line in gray is much steeper, indicating a higher risk or more frequent adverse events, complications of respiratory functions compared to idebenone. We also have fewer hospitalizations that we observe, again confirming in the long term, up to 5 years, the evidence that we have seen in a well-controlled, placebo-controlled trial before. Now, I'd like to summarize and say that loss of respiratory function ultimately results in the need of assisted ventilation, which has negative impact on the quality of life in advanced patients with DMD. There's currently no treatment approved to slow the decline of respiratory function. The randomized controlled DELOS trial demonstrated that idebenone slows the loss of respiratory outcomes. Multiple outcomes were analyzed, and reduced the risk of bronchopulmonary adverse events and even the risk of hospitalization due to respiratory complications. The data I have shown today demonstrate that the treatment efficacy for idebenone on these respiratory function outcomes is maintained for up to 6 years, as demonstrated in this real-world evidence. And with this, I would like to acknowledge the study group that has helped us plan and conduct and analyze the study. You see multiple experts here that are also in the room, and I would like to thank them all for encouragement to do this analysis and this study. Without their help, we would not have been able to conduct this. I also would like to thank you as patients and families to come forward and participate in trials, not just in our trial, but in any trial that is currently ongoing, because without your support, we could not show any of these treatment benefits or could not assess possible new therapies. And with this, I would like to thank you, and also like to thank you, the translators, who do always a tremendous job. Thank you very much. Thank you.
Bene, grazie al dottor Mayer. Tra l'altro colgo l'occasione anche per...
Thank you, Dr. Meyer. Let me anticipate that over the next few months, we're going to be launching a survey on respiratory problems. And so we'll be able to evaluate those problems from the point of view of the families and the young patients. And on that basis, we will see how to best coordinate an effective response when it comes to the evaluation, research, and the therapeutic treatment of respiratory complications. But you'll have much more in-depth information when the survey is actually launched. I'd like to call upon our speakers, the presenters, and ask them to join me here so that we can start the question and answer session.
Perché io non saprei fare da interprete in caso. C'è qualcuno che ha qualche curiosità, qualche domanda da...
Is there anyone who has any questions that they'd like to ask on the 4 different topics that were discussed?
Una domanda qui al centro.
We have a question here at the center.
C'è già.
I have a question concerning Divinostat. Amongst the inclusion criteria, you also had minimum age of 6 years. Why? Thank you. Well, the reason for having a minimum age of 6 years is related to the ability, obviously, to take all the tests in the protocol. The other important reason for that is that we are trying to identify a patient population that has a disease that's not too advanced, a phase of disease that's not too advanced, because this substance, givinostat, works on the muscle, so there has to be sufficient muscle mass in order for it to work. But on the other hand, the disease must be evolving, ongoing. So children under 6 years of age are still, well, relatively developing their muscle function. So that's the reason why we introduce this as a limit. I'm sorry, but the mic's not working. The answer to that question that was inaudible is the studies will be conducted right now. We're assessing or evaluating the effect of givinostat in the pivotal study. After that, the program will be extended, and so it will include younger children. And this is actually a request on the part of the European regulatory authorities. And it will also look at youth and men that no longer have ambulation capacity.
C'era una domanda.
I think there was a question right here.
Qualcuno aveva alzato la mano?
Someone had raised their hand, but I don't remember who it was. Thank you. I think you've already answered that question, but Simone tried to get into the givinostat trial twice, and both times he was told the damage wasn't enough, it hadn't progressed enough, and the hospital told us that, we would have to wait. You partly answered that question, actually. Okay, well, let me just say that the protocol was recently amended. At present, the amended protocol is yet to be approved in Italy because it's still being looked at the ethics committee of the regulatory authorities. Hopefully it'll get their authorization soon. The new inclusion criteria involve age and the ability to take the 4 stairs, to climb the 4 stairs. We had included this algorithm previously to try to identify a certain population at a certain level of MRI fat content. We will use MRI at entrance only for baseline purposes.
In English, yes, can I? I have a question for Thomas Meyer. What is your feeling in the improvement of real-world data for accelerated approval drug? A fast-track procedure can improve the quality of real-world data?
So that's a general question, you mean? In general, what can be done? Well, that is not to me to comment on this. However, it is clear that long-term data cannot be collected in a controlled, placebo-controlled setting. Even the DMD guideline in Europe says very clearly that longer-term data will not be able to be collected like this. So I guess in future, regulators, being it the EMA or regulators, be it in pricing reimbursement, will want to see long-term benefit to the patients. So one way to collect these efforts are real-world data collection procedures, and there's more and more emphasis being given to such efforts. Now we have shown here for the first time very small data set. Nevertheless, I was quite intrigued how informative even small data sets can be if we have long-term follow-up times.
C'è qualche altra domanda?
Are there any other questions?
No. Bene.
That doesn't seem to be the case. Oh no, there we have one at the bottom.
Buonasera.
Good afternoon. I have a question for the gentleman who spoke to us about Divinostat. I'd like to know whether it will be possible to have an early access to the drug, maybe during the trial period, under the control of the clinical facility. Well, at present, the drug is used in the context of clinical trials only. Of course, we are planning to conduct a host of tests. There will be an ad interim analysis provided for in the study, and on that basis, we will see what can be done. Obviously, there are clear limitations to this, but there's also the possibility given by legislation to keep going. It depends on the results.
Bene, se non ci sono altre domande.
If there are no further questions, I am told that we could try to take the poll again, because this morning it didn't really work. So we'll try it again, because I think that technical changes were made. So could you please project this on the screen? Thank you, by the way, to all the speakers. Thank you for being with us.
Quindi ci sarà un link.
So you'll see a link, and you can click on it using your cell phone or the website of Parent Project. It'll redirect you. And on the screen, you should see a question that you'll be able to answer within a few seconds.
La domanda che dovrebbe...
The question that you should see on your screen has to do with the respiratory tract. It says, have respiratory infections appeared over the past year? And if so, how many? You can answer no, 1, 2, 3, or more. Is it working?
Ecco, vedete in diretta come stanno...
Okay, you see how the respondents are increasing.
Riusciamo a vedere? Altri 5 secondi.
I'll give you 5 more seconds.
Beh, diciamo che ormai la pole position è stata...
Okay, I think we have a prevalence of no's, but this is very much correlated to the age of the patient. I think we can move on to the second question now. You should see this on the screen. To what extent do respiratory complications change the quality of life, meaning the quality of the life of the patient and their family? And there are several answers. Very much so, slightly, not at all.
La situazione in evoluzione, vedo. Testa a testa tra poco e molto.
Okay, I think that we've got a tie here between those who say that the impact is great and those who say the impact is slight. I guess this largely depends on personal experience and age. Anyway, respiratory problems and complications in DMD, we know, are one of the factors that determine sometimes the life expectancy of the subject. So I imagine that those who answered slightly so have very young children still. But do bear this in mind, because this is something that could become even more significant as time goes by.
Successiva. Quale tra queste problematiche incide maggiormente?
So now, which one of these problems has the highest impact on daily life? Mobility, bone fragility, respiratory problems, heart-related problems, or nutritional issues?
Sembra che non ci sia storia in questo caso. Diciamo che c'è un netto prevalere della funzionalità.
So apparently mobility seems to rank first, which is quite understandable because, of course, we all have to come to terms with the difficulty in mobility, so, architectural barriers and so on and so forth. So a reduced mobility is rightly enough perceived as priority number one, even though I believe we should not neglect the other ones. Okay, let's move on to the last question.
Se poteste decidere di partecipare a diversi trial clinici.
So, what about clinical trials? Should you be able to choose which one to participate in? Which one would you choose? Investigational products aimed at improving mobility, or rather the muscular aspects, or the heart-related aspects, or else respiratory aspects?
Anche qui sembra che non ci sia storia.
Here again, it seems like we all agree.
Ok. Qualcosa è cambiato, ma... Bene, direi che la soluzione...
Well, yes, I believe we can now say that we have decided. Well, these are results that will be conveyed to those who might want to use them for the best, of course. And now we're going to show you some more videos about the trials now being funded by our association. So 2 more researchers will now be shown in their videos. Let's watch them before our coffee break. Thank you. Thank you very much.
Sono la professoressa Boucher del Dipartimento di Istologia dell'Università La Sapienza di Roma e lavoro da una decina d'anni. L'attività di ricerca del mio gruppo è concentrata sulla distrofia muscolare di Duchenne. In particolare quello che noi adesso vogliamo fare, grazie al supporto di Parent Project, è di verificare la fattibilità di un protocollo farmacologico per prevenire le alterazioni tardive della malattia della distrofia muscolare che avvengono a livello del cuore e del diaframma. Noi abbiamo già precedentemente dimostrato che l'inibizione di un enzima che si chiama proteinchinasi C-teta previene il danno e l'infiammazione a livello del muscolo scheletrico degli arti di topi distrofici utilizzati come modello sperimentale. Quello che adesso vogliamo fare è estendere questi studi a livello del cuore e del diaframma, dove ancora poco è noto riguardo alla risposta infiammatoria e alla cinetica di deposizione di tessuto fibrotico, e quindi vogliamo inizialmente estendere i nostri studi, la nostra strategia per studiare in dettaglio la cinetica e la qualità della risposta infiammatoria a livello del cuore e del diaframma, per poter identificare finestre temporali in cui intervenire con il nostro inibitore per poter verificare la possibilità di prevenire il danno in questi organi. Questo studio, oltre a dovrebbe essere una sorta di dimostrazione valutazione di fattibilità e potrebbe in ogni caso portare all'identificazione di ulteriori bersagli sia cellulari che molecolari per appunto contrastare la progressione della malattia a livello del cuore e del diaframma nelle fasi avanzate della malattia.
Buongiorno, sono Roberto Rizzi e sono un ricercatore del Consiglio Nazionale delle Ricerche. Mi occupo già da un discreto tempo della distrofia muscolare di Duchenne ed in particolare un mio progetto è stato finanziato dalla Fondazione Parent Project. In particolare mi occupo dello studio del rimodellamento cardiaco nei pazienti affetti dalla distrofia muscolare di Duchenne e più profondamente dello studio degli effetti di Givinostat sul remodeling cardiaco che avviene nei ragazzi purtroppo affetti dalla patologia. In questo momento abbiamo avuto dei risultati incoraggianti per il miglioramento della frazione d'eiezione, ovvero dei parametri fisiologici cardiaci dopo la somministrazione per un lungo tempo di Givinostat in modelli murini, in modelli animali della patologia, e abbiamo avuto delle evidenze molto importanti sul ruolo della matrice extracellulare cardiaca nel modulare l'innervazione nervosa, che è importantissima per la regolazione del ritmo cardiaco e anche per la deposizione del collagene, che è un effetto che è molto indagato in questa patologia. In particolare abbiamo trovato e stiamo approfondendo un coinvolgimento anche del sistema immunitario nella deposizione della matrice extracellulare e stiamo compiendo molti sforzi per cercare di individuare il meccanismo responsabile di questo fenomeno.
Mi chiamo Fabrizio Racca e dirigo il Dipartimento di Anestesia e Rianimazione dell'ospedale di Alessandria. Da 18 anni mi occupo dei pazienti affetti da Duchenne, gestendo sia la ventilazione domiciliare che le emergenze in pronto soccorso e in rianimazione. La qualità di vita e la prognosi dei pazienti affetti da Duchenne è nettamente migliorata negli ultimi decenni grazie alle terapie con cortisone, la ventilazione non invasiva, i farmaci cardioprotettori. Tuttavia rimane una significativa mortalità dovuta a insufficienza respiratoria e insufficienza cardiocircolatoria. Ci sono delle raccomandazioni che ci spiegano come trattare nel miglior modo questi pazienti quando hanno le ricotizzazioni. Purtroppo queste conoscenze non sono di dominio pubblico, nel senso che non tutti i medici che gestiscono le emergenze in pronto soccorso sono a conoscenza dei trattamenti che richiedono i pazienti affetti da Duchenne, perché è una patologia molto rara. Allora, le recenti raccomandazioni di Lancet 2018 sottolineano l'importanza di introdurre un emergency card per ogni paziente affetto da Duchenne, proprio per la gestione delle emergenze. Ed è questo il progetto di cui mi sto occupando e che Parent Project sta finanziando. L'emergency card è un mezzo elettronico e cartaceo attraverso cui possiamo condividere le informazioni più importanti con tutti i medici che vengono a contatto dei pazienti Duchenne, che riguardano sia le complicanze più frequenti, sia la loro gestione e sia anche i dati più rilevanti sul compenso cardiaco e respiratorio aggiornati di anno in anno.
Si attiva questo? Dove si attiva questo?
Il microfono è acceso, sta accendendo.
Bene, allora ci prendiamo 10 minuti di pausa per non andare troppo.
10 minutes for our coffee break. So Just a time for a quick coffee, okay? 10 minutes.
Vi invitiamo a riprendere posto, a sedervi, a riaccomodarvi per ricominciare. Se riusciste avremmo recuperato il quarto d'ora di ritardo che avevamo e sarebbe fantastico e perfetto. Grazie a tutto il fantastico staff di Parent Project che farà in modo che tutte le persone che sono ancora in piedi si possano sedere ai loro posti. Sta per iniziare la tavola rotonda dal titolo Avere cura di chi si prende cura. Modera questa sessione la nostra Emanuela Giulitti.
Moderating our session is Manuela from the National Parent Project Center. With her, we have Professor Elena Pegoraro from Padua, Professor Alessandra Ferlini from the University of Ferrara, and we have the great pleasure of also having with us our families, as always. Once again, I'd like to ask you very kindly to please not continue your conversations. If you'd like to continue your exchanges, do so elsewhere so we can begin our session. Thank you.
Allora, buon pomeriggio a tutti.
Good afternoon, everyone. This roundtable, which is formed exclusively by women, as you see, was devised to place emphasis on the role of women who come to terms with DMD and BMD. The various presentations shall focus on the importance of being aware of the pathology and the possible status of being a female carrier, stressing the need for a clinical approach in the case of female carriers. A special emphasis will be placed on the psycho-emotional role, and we will hear from different testimonials. And now I'd like to give the floor to Professor Ferlini from the University of Ferrara, who has worked with us for many years since the outset of a project called A Diagnosis for All, when in Italy there was a complete absence of standardized procedures and a very long time to come to a final diagnosis. Thank you for this introduction. We must never speak of time or years because years go by very quickly. But anyway, let me introduce this roundtable. It's a great pleasure for me to be able to share this session with Elena, Professor Pegoraro. We're going to try to have a very interactive session. We're going to see ads which should elicit questions in all the women who are attending here and who are listening to us. Of course, we're talking about dystrophies. We know exactly what this group of disease is. It brings together several clinical pictures. We know this. We know about the dystrophin gene. I think that we're all very familiar with it in this context. Just one brief reminder, this is a very large gene, and this is part of the reason why diagnosis, a genetic diagnosis of such gene mutations is quite challenging. It's a little easier today, of course, but you see this, the 69 constitutive exons plus other alternative exons, and these other locations that are the hotspots of these mutations, especially the deletions. We have all types of mutations of dystrophin. These are the percentages. for the characterization of the genotype, both in DMD and BMD and in the carriers. So 65% are deletions, 25% small mutations, and duplications, 10%, that we heard about this morning as well when we were discussing therapeutics. And today, too, we apply the frame approach. And usually, if that is maintained, it is associated with a slighter phenotype. If it's lost, instead, we lose the translation of protein, and so we have the absence of protein, and which leads to Duchenne dystrophy. Just very quickly, I'm going to cover some aspects that will be further illustrated by Dr. Pegoraro. Generally, today we can have two types of pedigrees. That can come up during a genetic consulting. On the right-hand side, we have the female carriers, which have these dots within the frames. This is genetic, so regardless of the genotypic characterization, these are carriers because they have a son that's affected, or two sons that's affected, or an affected sibling. So a carrier is an attribution that we assign on the basis of the pedigree. That's on the right-hand side. On the left-hand side, there is only one individual that's affected, and this is quite a large percentage. Indeed, over 40% of our patients are so-called sporadic cases. Studying these cases is really important to be able to understand whether the mothers and the sisters are carriers or not. Let's talk about the definition of carriers. We know that this definition is strongly related to their genotype, and in particular, the expression of the X chromosome. We know that females are mosaic, and one X chromosome in a stochastically random fashion inactivates certain tissues. This is a very old mechanism, which generally is true for all mammals. And as a result of this, only one X chromosome is expressed entirely. So females have a mix that expresses the mutated and the non-mutated allele, and that's why the phenotype is a mosaic phenotype. This will be shown later by Dr. Pegoraro. In picture D, you see DMD. In a female, this is the aspect of the muscular fibrous cells. Some are very evident, and they show a nice staining of dystrophin in the sarcolemma, and the others are totally negative, as in a mosaic situation. This is very different from a DMD and a BMD that's full-blown. There are particular pathogenetic mechanisms in symptomatic carriers, for example, that can be involved in determining this extremely rare phenotype. Indeed, in over 90% of the cases, the carriers of dystrophin disorders are asymptomatic, and in some cases, very small ones, with different degrees of manifestations, there can be clinical symptoms. For example, the dislocation of X chromosomes or others that facilitate the non-random expression of X, chromosome X, that has only the mutated allele. So the take-home message is that in all symptomatic carriers, it is recommended that the karyotype be analyzed, and or this new CGH technique. It's a genomic hybridization, which enables us to see whether there are other abnormalities. You see here, in addition to the dystrophin mutation, there's a broad deletion that involved adjacent or non-adjacent genes, so non-sequential deletions, and they can only be viewed with this technique, CGH, which can explain the symptomatic nature that we find, or symptomatic signs in certain carriers. The role of the X chromosome is quite controversial. In the literature, there are many findings that report on contrasting situations, on the fact that, can clinical severity be correlated with activation of the X chromosome? We don't know. We have different findings. And so today we're not in the position to scientifically determine the role of this kind of inactivation on the clinical phenotype. Let me come to my conclusions. This clinical part will be discussed in detail by Elena, so let me just say by way of conclusion that less than 10% of female carriers can be symptomatic at different degrees of severity. There are significant factors that lead to the onset of these symptomatic phenotypes, but we're not able yet to define them accurately. Surely, modifiers and certain transcription regulatory mechanisms can play a role. But let's remember that in all female carriers, symptomatic and not, it's important to undertake a cardiological follow-up. About prevention, genetic and medical prevention, there are different forms of prevention that can be observed. In particular, non-invasive diagnosis, NIPT, or pre-implantation diagnosis, prenatal diagnosis, and also, what should we do? What kind of approach should we take with minor female carriers? NIPT has become available recently only, and it can identify the genetic disease on the fetal DNA that's circulating freely in the mother's fetal circulation. This allows for prenatal diagnosis around week 10 or 12 of pregnancy without a placenta biopsy or any other invasive methods. Now, on this basis, the most immediately applicable technique is used for fetal sexing, and this can reduce by at least 50% the possible invasive diagnoses like amniocentesis and villocentesis, because if you identify a female fetus, then you just don't have to undergo the invasive procedure. This method, and I want to say this unequivocally, as it was said yesterday, is not available or made available in Italy by the national health system. There are many reasons for that, but anyway, it is available in certain European and non-European countries like Australia, the UK, and Canada. And let's talk about this during the question and answer session. Let's just say that it's not accessible in Italy. Many laboratories do provide it on a private basis, but not for the diagnosis of DMD, so for fetal sexing purposes only. And that's because the mutation diagnosis at this point in time is not direct, it's indirect, and it is performed through a very complex system which goes back to the linkage analysis. I don't know if you recall it, maybe some of you do, I certainly do, which made it possible to recognize the chromosome that carried the dystrophin mutation. And NIPT proceeds according, well, it's based on the very same approach. This is a study by a group of Manchester that did a prenatal analysis in a young boy with a mutation of exon 45. So we need to wait for the whole genome sequencing technique to become available, because it will make viable and safe this type of diagnosis. Pre-implantation diagnosis we're all very familiar with. Female carriers can subject themselves to this in the preconception stage with there's a protocol, of course, and guidelines. Today, pre-implantation diagnosis can be carried out with the blastomere using many techniques, including sequencing and NLPA, so they can technically identify the dystrophin gene, so it's feasible. Now, it does require a preclinical setup that's very complex, as is the case with assisted or in vitro fertilization. And there are still a few technical glitches as far as the enrichment phase is concerned, but technological progress is being made on that front. PD prenatal diagnosis is based on three possibilities. A placenta biopsy that happens between week 11 and 12 of pregnancy, amniocentesis, the harvesting of amniotic fluid, and the harvesting of fetal blood. Here we analyze the DNA of the trophoblast, so the response time is very quick, only 5 to 10 days. A bit longer from amniocytes, and long also for fetal blood. Who is prenatal diagnosis for? Well, for all female carriers, those who have who are heterozygotic for a mutation of DMD, and for mothers of DMD boys, because we do have the germinal mosaicism issue, and so guidelines do recommend prenatal diagnosis even for mothers who are not genetically carriers but who might have a mutation in the germline at the level of the oocyte. Finally, the diagnosis of female carriers in prenatal diagnosis was not recommended on the basis of WHO guidelines for genetic tests, because prenatal diagnosis with genetic tests requires a technical viability, and WHO does not recognize that dystrophin absence. It's not possible to predict whether a female heterozygotic is going to be affected or not. One last point, the diagnosis of carriers in women under age. Many of you, I'm sure, have dealt with this problem, again, on the basis of WHO guidelines and on the basis of the law decree of 2016, which protects privacy. To do this diagnosis, underage women have to have a tutor, and they need to motivate the request for this type of diagnosis if they're under 18. This is all for me, and we can further elaborate on the issue during our discussion. Thank you.
Well, now we give the floor to Mrs. Pecoraro from Padua University, and she is a very important reference point for the clinical management, and she has been working with us for quite some time. Well, thank you for being here. Well, the mutations of one gene can be expressed with different phenotypes and different clinical expressions. You may have DMD BMD, but there are also the carriers. And as Alessandra showed you, carriers can be the mothers or sisters of patients suffering from MD. But there are women who have no family history of MD, but clinical signs of myopathy. Well, in 10% of cases, these women are carriers. So these women must be identified, because by doing that, you can do what you must do in all carriers, offering them genetic counseling, as already explained. So the woman who has a deficit in strength and myopathy must be studied, because we must check whether she is a carrier. Well, carriers are in most cases asymptomatic. These women do very well. These are two twins. One twin has a boy with DMD, whereas the other twin has the clinical sign of the disease, as a deficit in strength. She has difficulties walking or climbing stairs. So in 10% of cases. And the difference between the woman who has this muscle weakness and the woman who hasn't, well, depends upon the quantity of dystrophin in her muscle. If you look at the two images, the one on the right, where the dystrophin negative fibers are more numerous, they don't have that orange circle representing dystrophin, whereas the image on the left shows that the dystrophin negative cells are very few. And this also determines the quantity of dystrophin present. So dystrophin by itself is the main determinant of the possibility for women carrier to show a deficit in strength. This symptomatic carrier hypertrophy, intolerance, cramps, or really muscle deficit, they may be severe, intermediate, or minimal. And then there are the so-called Duchenne-like phenotypes, where the girl has a behavior that is similar to the boy, so an early deficit in strength, and in some cases even not being able to walk. And why do we speak about this weakness? Well, weakness essentially in difficulties in activities of daily living, climbing stairs, lifting heavy objects. In some cases, symptomatic carriers may have an atrophy that initially is not associated with a deficit in strength, a sort of thinning of the muscle, whereas the muscle is still strong. And this strength deficit, asymmetrical, a slight asymmetry is very common in muscle diseases, but it can be very marked in carriers. And this also this macroglossia, it means an enlargement of the tongue. When you identify a carrier, you have to follow up, because we must study the clinical story of the patient and the data of her clinical story. That is the starting point for therapy and also for assessing the efficacy of therapy. 10% of carriers may show signs of muscle weakness, but 10% of carriers may also have dilative cardiomyopathy. And women who have a deficit in strength also have cardiomyopathy. So the two events may be separate. When I speak about dilative cardiomyopathy, well, you know that the heart is supposed to pump blood. If the heart is dilated, the efficiency of the pumping decreases, and this may be a clear cause of, invalidity and even demise. Up to 18% of carriers have a problem in the left ventricle that can be identified through instrumental exams. If we use a heart NMR, cardiac NMR, the number of carriers with some impairment, some deficiency, goes up to 47%. NMR is now considered important because we can identify a therapeutic window to treat a carrier before we have a clinical sign of cardiomyopathy appearing. So the heart involvement is asymptomatic. When we study them with ECG or ultrasound, we find some alterations. Well, we do not always find dilated myocardiopathy or heart failure. What is the frequency of LG? Well, in DMD carriers, apparently is slightly higher in DMD if compared to Becker. So the heart involvement may be so bad as to lead to heart transplantation. And so in the cardiac muscle, we have a mosaic of fibers having or not having dystrophin. In 2005, the American Academy of Pediatrics had drawn up a number of recommendations for monitoring. And first of all, carriers must be educated. They must be informed on the risk they may have of developing cardiomyopathy. They must be taken to a qualified cardiologist, and then you must have a regular follow-up at least once every 5 years or more frequently if this is clinically advisable. And then you must do more or less the same you do in males with cardiomyopathy. Some years later, this survey was carried out to see the adherence, to check the adherence, and there is very, very limited knowledge of the risk of developing cardiomyopathy in carriers, because in this survey, only 63% of carriers were aware of the risk of cardiomyopathy that can be associated to the fact of being a carrier, and only 64% had carried out checks of the heart. This was repeated in the guidelines for DMD, those that were published in 2018, and the guidelines for carriers say that cardiac NMR would be advisable and having a cardiac check regularly. And so in terms of prevention, there is still room for improvement. And another aspect is vaccination. The Italian Association of Myology some years ago, in 2014, had listed these recommendations. The anti-flu and the anti-pneumococcal vaccination is recommended for symptomatic carriers for individual risk and in carriers at large, so as to reduce the risk for this infectious disease to be transmitted to the family member who suffers from DMD or BMD. The other area is that of pregnancy. Well, pregnancy has not been studied in an exhaustive way in the area of neuromuscular diseases. So these findings refer to neuromuscular patients at large. So we're not speaking of carriers as such, but 2/3 of patients, so here we are speaking of symptomatic patients, may refer a worsening of this deficit of strength during pregnancy. Rarely, but there are some case reports, there was a cardiomyopathy immediately after childbirth, or an increase in the severity of this strength deficit. We don't want to create panic, but just to say that you have to plan pregnancy and you have to check cardiovascular risks and take all necessary measures to reduce risks to the minimum. And obviously, parents are the main caregivers of patients, both DMD and BMD patients. All carriers must face emotional, social, and psychological problems linked to this. In symptomatic patients, associated cardiomyopathy or strength deficit imply a number of extra problems, especially in the family management, and this is to be taken into account in planning and in the support we give to these families. Thank you for your attention.
Grazie professoressa. E in questa seconda parte
Thank you, Professor. In the second part, we decided to let the mothers speak, the mothers who grapple with the disease, whether they be carriers or not. So mothers and sisters, and now I'd like to give the floor to Daniela. Thank you. Good afternoon. Hello, everyone. My name is Daniela. I am Lorenzo's mother. We learned about the DMD diagnosis when our child was only 2.5 months old, and you can imagine how we felt. For me, I had yet to savor motherhood. It was a terrible blow. When you expect a baby, even before they're born, you can imagine what they're going to be like. There are many expectations, and you dream about their future. I remember how I felt when I was told that Lorenzo was affected by DMD. All of a sudden, I felt like I had no body, and like everything around me weighed so heavily that my mind and my body were not capable of supporting them. Initially, I tried to understand what was going on, and the greatest mistake was to get information off the internet because it was very painful for me, and today I recognize that it's not entirely truthful. At the hospital, I was advised to undergo genetic investigation. Being a carrier or not was not something that elicited anxiety in me. The only thought I had was, how should I behave in case I should become pregnant again? The result came after 2 years. I was not a carrier. The mutation had happened in Lorenzo's DNA. Since the diagnosis, I've changed my way of looking at life. I live in the present day after day. I don't think about the future. I look around and I see that the future is a huge question mark for everyone. Lorenzo has grown up knowing about his conditions from the beginning. We told him that his muscles were weaker than those of his peers. He didn't ask any questions, probably because we always explained everything to him every step of the way. To date, all of the phases of this illness have been tackled by us when they came up, and so we were able to face the present. I never asked myself, what will I do when he's no longer able to walk? When the time came, we faced it, and it was he who gave us the strength, and that the time had come for his first wheelchair, and I chose the color with him. And I was with him also when he started to play wheelchair hockey. For him, this sport came at the time in which he was losing the capacity to walk, and it helped him face this very important time in his life. Ever since he began playing the sport, he was able to experience new capacities, and his relational life is fuller. It's important for him to play hockey with other so-called normal kids and other kids who have different illnesses. I try to give him peace of mind, and I think I've succeeded today. Of course, we do fight, as any mother and child fight. It's difficult to be a mother, and even more so when you have a special son. I love my son, and thanks to him, I've discovered a strength, a force that I did not know I had, mentally and physically. I fight against society with him that often does not know what integration is all about. Our days are really filled between sports, medical examinations, work, and school. We get to the end of the day feeling exhausted, but with a smile on our face. Thanks to Parent Project, I've come to know with a lot of mothers. With some of them, we've become good friends. We exchange experiences, and some of them have gone through the most significant passages of this disease before me, and they gave me some very important advice. So thank you all for your attention.
Grazie, Daniela.
Thank you, Daniela. Thank you for telling us your story and how important it is to face things day by day. And now, Anna Maria has the floor.
Salve, sono Anna Maria Petruccelli.
My name is Anna Maria Petruccelli. I am the mother of Fabiano, Valerio, and Matteo, 31, 25, and 14 years of age. I had Fabiano when I was 24, and despite the lightness of the age, I managed. And then Valerio was born. Since I was older with more experience, I thought I could be up to the task, but I was wrong, because at that moment of my life, precisely that I had really to face the real challenge of my life. We were told the diagnosis of DMD, and when I still didn't know I was a carrier, then the third baby announced himself. I mean, I didn't know much or nothing certain, just likelihood. And then knowing that you are a carrier and this feeling of guilt that invades you in those cases, were there again. I had to be strong, never give in, and give my kids, certainty, peace of mind, the certainty of having a united family, an empathic family, a family willing to fight. Over the years, many things have happened, but always at the moment of need together, we did fight. And that was a true glue in the family. There are difficulties, that's undeniable. But we try and do our best to make it feel normal. We've been traveling around London, Barcelona, Nice, and so on and so forth. Many difficulties and, in inverted comma, an effort to be normal. In everyday life, we have many obstacles, but if we try and face the situation as a new possible reality and with a good adjustment skills, then you can overcome things. I've tried to face difficulties without running away, and I've always tried to transfer this to my kids. The three of them now are very determined. They have a good skill at adjusting, and they are following their dreams, trying to reach their objectives. I work part-time. My husband helps a lot. Fabiano has a phone selling shop, and that is very challenging, but he's always present with his brothers. Valerio goes to college and is doing his biomedical engineering. Matteo goes to school, has very full days. We try and live this adventure with optimism, always keeping hope, hoping that the future will hold something beautiful for us. Thank you.
Grazie Anna Maria.
Thank you, Anna Maria, for showing us the strength of your family in your quest for normalcy, and you bring to us the important message, which is to keep hoping. And now I'd like to give the floor to Franca. I'd like to tell you about my experience in the hope that it might be of help to someone. And I'd like to tell you about this extraordinary and challenging but significant adventure that has enabled me to grow and be born again as a woman and as a mother. And about rebirth, I'd like to recall the wonderful novel by Giuseppe Pontigia, Born Twice. The author talks about embarrassed doctors who at the time of the diagnosis of a severe pathology didn't know what to say, and they used metaphors. But he also speaks of a doctor who spoke words full of hope to the parents. He said, you have to live day by day. You don't have to think about the future as an obsession. It's going to be a very tough experience, but you will not deprecate it, and you will come out of it feeling better people. These children are born twice. They have to learn how to move around in a world which which is difficult. They were born twice, and their journey will be more challenging. But in the end, for you too, that will be a rebirth. Ever since I read those words, I've stuck to them, and they really reflect my life up until the present day. When the doctors very nicely told us that Matteo, our firstborn, was affected by DMD, I couldn't believe it. I felt that everything was falling on top of me, and when they added that I was the cause as a carrier, I felt very guilty, and I couldn't let go of that guilt. At the time, Matteo was 4, and Marco, the secondborn, was 1. Because this illness strikes or affects boys, we had a 50% chance that Marco would be affected by it too, and so it was. So anger, loss, and frustration were the feelings I experienced when I learned for sure that Marco too was affected by DMD. But I did not remain slave to that attitude. Neither did my husband. Together, gradually, we started to accept the situation. Initially, we were very stressed, and we alternated rational behaviors to crying fits and feelings of inadequacy. We didn't think that we'd be able to educate our children, to raise them, to listen to their needs. We felt isolated, and we felt that we needed to devote time only to our family. In this long time of transition, luckily, we never felt alone because we were surrounded by the love of our families and some friends who understood our feelings and sometimes shared with us the burden of difficult days, giving us a concrete help and by spending time with our children, thus making us feel that we were loved. In the meantime, we tried to learn more about DMD. The doctors explained to us what the disease was all about in a very detailed fashion scientifically, and they always concluded that there was no cure, there was not much to do. The sense of ensuing frustration and powerlessness would grow, but during sleepless nights in front of the computer, looking at the internet, we came to know Parent Project, and we discovered that actually there was a lot to do. That time was like a watershed, and our life took a different turn. Indeed, the association became for us a beacon, both because of their simple explanations to offset the complicated doctor explanations, and because of the support and advice given to us, and the hope in research and a cure. By talking to other parents, we realized that we were not alone in experiencing these feelings, and together we found that we looked for ways in which we could help our children and improve their quality of life and ours. That's when we began our rebirth. We reorganized our whole life. We found a new balance and gave priority to peace of mind with a spirit of patience and cooperation, but at the same time retaining our independence and autonomy. We were leaving behind our total dependence on research. There was a lot more that was equally important, but that wasn't over. Our future would have some news in store. In December 2001, we learned that we expected another child, and we were really astonished because we hadn't planned on it, and we were worried about everything that we would have to face. We knew how to proceed, of course, in order to limit the odds that the disease would affect the fetus. My gynecologist said that I should go to Milan for villocentesis. It was really tough. For the very first time, I felt distressed. The feeling of guilt was stronger than before. But when, after a few months, I got a phone call telling me that I was expecting a baby girl, I was relieved, and I tried in every possible way to regain that balance in the family that we had conquered. Today, Matteo is 23, Marco is 20, and they fulfilled the dream of being rather autonomous and living on their own. Mariana is 16 years old, and she plays volleyball. I think I can say that I have vanquished muscular dystrophy, not because there's a cure, but because I didn't allow it to adversely influence our lives. Thank you.
Grazie Franca, che ci ha parlato di quanto la sua famiglia ha avuto la...
Thank you, Franca. And now the floor to Giulia, who is the sister of a DMD. Good morning. Good afternoon. Thank you. I first wish to thank Parent Project for asking me to share my experience with you. I am the sister. I am a DMD sister, even though my brother died 2 years ago. And I am a carrier myself. Being a carrier is something that we were not told immediately, even though this possibility had been explained to us very well since the very beginning when my brother was diagnosed with the disease, so as a little child. And so the neurologist who was following him at the time had sent us to the Verona hospital to make this analysis. But these checks were essentially aimed at a possible transmission of the disease to possible children. But at the time, nobody told us that women carriers might have some physical problems, so we didn't know that. Anyhow, the result was that it was very likely for me to be a carrier because my mother had transferred the same X chromosome both to me and my brother. And even though at the time the mutation as such had not even been investigated upon. How did this affect my life? I mean, being a carrier, having a high likelihood, high probability of being a carrier, in some way may sound strange, but I was glad because, I mean, the most important thing was my brother. My brother had just received this diagnosis, and it was a diagnosis that was, I mean, something we could not face as a family. So I absolutely did not want him to have this disease. I thought it was unfair. And possibly, I thought it was unfair for him and only him to have this disease. And I started thinking, well, if I am a carrier myself, somehow we were equals. And I could have some failures, some impairments like him. And that is, I mean, and then time went by, the disease was there. I started scientific studies that enabled me to better understand the situation. And my boyfriend was also informed of the situation. And then we decided to look for the mutation in my brother's DNA. And then we spoke with Professor Ferlini, since we live in Ferrara province, so we are very close. And in 2005, we identified the mutation. It was a new mutation, a new splicing mutation, and it is a mutation that is present also in one of my dystrophin genes. So I was a carrier. It was confirmed. And even though years had gone by, I was glad to be somehow like my brother, or rather, I think it is just fair, even though it is very difficult to take some decisions. I believe information is paramount. Well, I mean, being able to know whether your kids might have the disease, or you might want to avoid them having the disease. I think information is essential. You are here with kids, you have other kids, or you may want to have other kids, but also for your daughters. At present, your daughters may be very young, but knowing that you are a carrier or you are not a carrier may be useful, so that when the time comes, they know what it is about and they can face this problem. And as for the problems associated with women carriers. We discovered them just a few years ago from Dr. Villanova in Bologna when he started following my brother, and he immediately informed us about this possibility. And that too is very important. Women must know, because doctors are not so well informed. Usually they are not well informed on Duchenne because it is a rare disease, and so also carriers having these problems is something they may not know. So if you and your daughters are aware of that, well, you can step in so as to make all necessary checks. As you know, now I'm an adult. My brother is no longer among us, and now I am thinking about starting a family with my boyfriend, being aware of our situation. And I'd like to close with a personal reflection. And these boys, we are talking about them here, and these boys are here and they are precious. We grow very fond of them because they need everything, but we need them just as much, and they are unique, they are important, both for what they can do in society, but also for our families. And as you said before, they can really extract from us strength, energy, abilities that we had not even dreamt of. And being a sister, well, my brother was the most beautiful gift I could ever have. So thinking that you can decide for these boys to be different from what they are, it is as if you were refusing them, as if you were excluding their value, losing all their wealth. I wouldn't want my brother different, slightly healthier, of course, yes, but I wanted him exactly as he was. Of course, this is something very personal. These are very sensitive subjects, so everything is very subjective. But in closing, I'd like to say that today women carriers have all the tools available to to decide whatever they think best for themselves and their families. And these conferences, these moments of information, of education are very useful, so that then women can use these tools at best and have the best possible life for themselves and their families.
Grazie Giulia. Abbiamo potuto vedere come
Thank you, Giulia. From the narrations that we have heard, what emerged was the changes that every family had to grapple with, together with a network of specialists who guided them by illustrating possible strategies, and a network of parents who, by exchanging experiences, support each other. I'd like to remind you about the material produced by Parent Project called Let's Take Care of Our Women, which you found in your pack yesterday. And now I'd like to open the floor for questions or for possible testimonies.
Sì, buongiorno, io sono una mamma portatrice.
I'm sorry, but we don't have a microphone. I'm a carrier and I'm a mother. After I found out about
being a carrier, I insisted on having the test on my daughter, who's now 8. And I'd like to thank the genetic expert because I really insisted that he evaluate my daughter, which he did. Now, Greta is much more aware, my daughter, of her condition, and I wonder, what should I do? I have a sheet that says that she's a carrier. I don't know what to do. I realize that she's very young, and I requested that she be analyzed because I wanted to find out. I needed an answer. So could you advise me on this? And something else about the feelings of guilt. I don't feel guilty, and I'm worried that I don't feel guilty because I'm thinking, will it come out? And maybe when I do feel guilty, I won't be able to manage that sense of guilt. I don't know.
Ma io do una risposta medica più.
I'll give you a medical answer more than anything else. From a medical point of view, if your daughter is well, I would wait a few years before instituting a medical therapy. And I would do so sufficiently, but not excessively. So my advice to you is around 16, 17 years of age, have her go a cardiological test. And if she's normal, which is likely to happen, do it every 3 to 5 years. Making or being watchful of symptoms like cramps in her legs, being short of breath, and her heart beating fast. But if she reports nothing of that, none of those symptoms, I would just let her have just grow.
Infanzia, quindi comunque lei ha già fatto una prima valutazione iniziale.
She's already undergone the first evaluation and she's absolutely fine. What do I need to do though? I don't know if you understand what I'm saying.
Qual è questo risultato? Tenere conto che anche le bambine adesso crescono rapidamente e che
you have to realize that girls become sexually mature very early on. So whenever you feel the time is right, explain to her that there is a risk that this problem will recur. Obviously, one today can plan on becoming pregnant when one is ready. Now things are different. She still seems young to me. You say that she's old enough, but I would wait a few years. For the time being. I don't know, maybe the mothers can actually pitch in here.
Io aspetterei.
I would wait. But if you think that it would be appropriate to talk to her, then go ahead. It's important that she's well and, proceed according to the advice that was given. You also talked about this feeling of guilt. That's one of the emotional reactions that you might have when you find out about this kind of diagnosis. It's very subjective, very personal. So you can experience it or not. If it does happen later on, then you will find the ways and means to face it, as you faced the first diagnosis in your boy.
Altre domande?
Salve, sono Sandra, la mamma di Gabriele.
I am the mother of Gabriele, age 11. I'm not a carrier. I did the screening, but I have a daughter who is 16. And I was told, since I was not a carrier, I was told to wait for control when she's older. If the mother is a non-carrier, is it advisable to have the daughter screened just the same or not? Well, there are guidelines and there are also legislative decrees such as the one on privacy recommending not to carry out genetic tests in people under age. Obviously, if we're speaking of asymptomatic cases, but of course, if there are symptoms, you are supposed to reach a diagnosis, but at present that would not be the case. From existing literature data, the dystrophin mutation in a carrier that hasn't any clinical sign is not in agreement with the guidelines and the decree before the person comes of age. There are very many reasons. I mean, if you have time to read this decree, this is a very innovative piece of legislation. And there are very important reasons for not carrying out this test. That is called, is not really a preclinical test. It is a test on heterozygosity before age 18. And there are many reasons. Postponing this test won't change anything if you do it after age 18, because you must also consider the minor. The minor cannot make the decision because whatever concerns him, is decided by parents or whoever is in charge of the minor. So I believe that it's up to the person. I mean, he or she must make the choice to submit to the test when he or she can truly make a choice, having reached the age 18. Even though you are not a carrier, the girl has a reduced risk, but still she has a risk of transmitting chromosome X, or anyway the gene for the mutated dystrophin. So from the medical viewpoint, it is very advisable to carry out the test. Yes, that is very important.
Allora, prima di procedere con le domande, vorrei fare un saluto.
I like here to welcome a person who came here today, Mrs. Marcella. Marcella is 102 and she had to face DMD herself. Age 102. Possibly she had children with DMD.
Grazie signora Marcella.
Thank you, Marcella, for coming.
Ci sono altre domande?
Is there any other question?
Hello. First of all, thank you for sharing your empowering stories. Yeah. But because the session is called Caring for the Caregivers, I'm curious to know how you, the mothers, cope with your own needs, emotionally but not only. Do you think you have an inner power, or you asked for help, and if you did, where?
Scusate, il microfono non ha funzionato. Può ripetere la domanda?
Could you please repeat the question? Sorry, but your microphone is not that we were not listening, we could not hear.
Is it working now?
Okay.
Yeah? Okay. So I was saying thank you, first of all, for sharing your empowering stories. And as it is a session about caring for the caregivers, I was wondering, how do you cope, you the mothers, how did you cope with your own needs, emotionally and not only? Did you ask for help?
C'è un problema con le traduttrici, un attimo. No, le traduttrici non sentono bene. Comunque
È stata se e come queste mamme che hanno raccontato la loro esperienza di portatrici sono riuscite a gestire emozionalmente il carico, a ritagliarsi una vita personale, dove hanno trovato aiuto a chi l'hanno chiesto.
Io insomma l'avevo già...
In Parent Project helped me enormously from the psychological viewpoint, both as a mother and as a woman. And I did find support, an important support. And not on my own, but of course with my husband. We were able to find a balance, also thanks to Parent Project.
E se non ci sono altre domande?
If there is no other question, I wish to thank... no, there is one question.
Con la diagnosi preimpianto si riesce a debellare definitivamente la mutazione?
Well, for example, the kids would not have this disease. Sorry, but the first part of the question was off microphone. With pre-implant diagnosis, it is possible to identify dystrophin mutation, both duplications and small mutations, and the technique has an accuracy of 95% because
che riguardano l'arricchimento del DNA che possono.
There are procedures concerning DNA enrichment that could be very precise. However, this is changing very rapidly. We are getting close to 98%. And that is the diagnostic accuracy that is accepted at present. There is a margin, yes, but you can modify dystrophin mutation at 98%, in 98% of cases.
Altre domande?
Any other question? If that is not the case, I think we can adjourn.
Allora, riprendiamo il sondaggio secondo le modalità indicate precedentemente.
We go back to the poll.
Link che trovate sul sito e la pagina che si apre collegandosi. Ci dovrebbe essere la prima domanda. Allora, quanto lo stato di portatrice?
To to what extent being a carrier affects the decision of being of starting a pregnancy.
Come possiamo vedere dalle prime risposte e come ci potevamo aspettare,
as we could expect, of course, being a carrier affects enormously whether you are going to start a new pregnancy or not, or a pregnancy or not. But there is 6% answering that it does not affect, and that is in line with what Giulia said.
Un'altra cosa importante è vi ricordo.
And also something else that is very important, the enrollment in the patient's register, not only for DMD and BMD boys, but also for women who are carriers. And here comes another question. Are you enrolled? Are you enrolled in the registry in Italy? Yes or no? 100% enrolled.
Quasi. Non si vede. Aspettiamo qualche altro secondo le ultime risposte.
Just a few seconds.
E poi passiamo alla successiva domanda, sempre che riguarda il registro pazienti.
Another question again on the patient's registry. How did you come to know about the registry? Well, health care staff through Parent Project Onlus, other families or other. Most of you got to know about the patient's registry through the Parent Project Association, through Parent Project Onlus. Of course, the registry is also mentioned by healthcare professionals or other families, people you get to know and that tell you to enroll in the registry. A few extra minutes.
A che ora è? Ricordiamo che
we wish to remind you that tomorrow morning our colleague Ilaria will be here, just here in the room, just in front, to update enrollments and also for those who want to enroll. From 9:30 to 11, she will be here just in front, just in the room in front, to Leptis Magna.
Siccome dobbiamo aspettare,
since we must wait till 5:15 for the video, and we will have here the companies with our President Luca Genovese, we wanted to share with Italian families, but it's also a good practice we can export to other countries. We wanted to share a success we have achieved and for the everyday life of our families. And so Luca will be sharing this experience with you. Well, for once we are early. That is very strange. And so we have this useful space to talk about this initiative, an initiative we will introduce we'll speak about very soon when we have the diagnosis with very small children. Well, then people have to go through commissions for having the acceptance of the invalidity first and the certification of the invalidity. And then there is a special piece of legislation concerning days off and leaves for parents. And some children had this that in Italian we call the 104 legislation, whereas some older boys did not have that. And so we got in touch with INPS, INPS, and Carlo Giacobini mentioned it yesterday. So we got in touch with INPS, that is the National Social Security, and then we had guidelines that will be translated into an internal circular directive, so as to have a uniform approach for the acceptance for the certification of all that. Also in terms of economic support, and so that is why the association is so important. And the slogan of this conference has not been chosen by chance. When we go to the Social Security Institute and we represent so many families, of course, we get some form of recognition. When it's just one single family, we can just complain. When we have, I don't know, to go to court to get something recognized. So this is an important example, and it is the way in which the association can do things. So over the next few days, this success will be made public, and we will inform all the families, but we thought it appropriate to inform you all about that. And of course, we are speaking essentially to Italian families, but in my opinion could be a useful approach in other countries where you have similar rights and similar cases of disrespect or non-abidance. And one final project funded by the Ministry for Social Policies. We have this Informa Azione. This is the name of the project. So Informazione, I mean, there is a pun in Italian. In March, we'll have local meetings, and in Florence, for example, we'll be meeting families in order also to understand if there are families who want to be delegates. So we really invite families to attend these meetings, and also to express their willingness to help us out. In this conference, there are many adult DMD and Becker, and this is important sign for us. We want to encourage that in our future programs. we really intend to have a sort of council of young people. Maybe that is not foreseen by our statute, but it is an important body, an important consulting body, because there are many of these young men who are becoming adults. There is an improvement also in the quality of life. There are many of these young men who go to college or work, real work now. There was a young man that is preparing a competitive examination for to become a civil servant. And so, in my opinion, well, that is something very important. So it is no longer just the parents' association, because in past years it was sort of necessary. Now we have our young men who have become adults and who can give their contribution. So the idea is having this sort of council of young people. We hope we can have the funding for a new project, also for training of these young men, and so distant training, e-courses, and also to work for companies, also working from home. Of course, after appropriate training, appropriate e-training. And this is a form of research. We try to prepare projects, as is the case for scientific research. Some of these projects won't be successful, whereas other projects may be successful and provide a special contribution. Beautiful words have come from the families, from the moms who spoke beforehand, and with a new form of hope to face this disease in a different way, with more hope, strength, and peace of mind. Not just looking for a therapy. We keep saying, well, we would like Parent Project to close as soon as possible, because that means that a treatment has been found, and so Parent Project will no longer be necessary. But at present, we are necessary. And Cristina is here. Cristina has been giving us big help. She is coordinating a team. Emanuele is here with me, and I mentioned her, but together with her, all the components of the CAD area of funding, collection of funds, events, and science. So there is this team, there is this Parent Project staff that has been working and is working, and Cristina is their coach and coordinator. She is helped by having excellent people in her staff, but Cristina must also fight with us, with the parents, because we are not as simple to manage, because we have our needs, but Parent Project provides the answers. We must also get questions with this project. For example, we are now creating sort of not truly a legal office, but having a sort of reference point inside the association, something that is going to be created over the next few months to try and formulate a number of guidelines on legal issues in schools, for example, as to the rights of our kids, of all sorts of kids with disabilities or the non-recognition of their rights by the healthcare commissions. And then the problem of architectural barriers. And our friend from Forlì, who is sitting here just in front of me, when he said, we must fight for our rights. Doing it individually as a family may create some problems because, you may have reprisals and not all have the force and courage to start the battle, let's say, as an individual. But the role of the association, as was historically the case for trade unions, for workers, the same goes for the association. The association steps in with a letter, for example, by our lawyer, whenever possible. For example, they say that a right has been violated, for example, not having a lift, and then paradox, well, the lift is there, but the lift is not working and no one is repairing the lift, or having a slide that is really poor quality just to say it is there. Well, we can do all these things through the needs that emerge from the families. We filter requests. Sometimes I make requests that are not really feasible, but some of these requests are feasible, are acceptable, and that is precisely what Parent Project is supposed to be doing. Always concentrating on scientific research, because that is the main objective of Parent Project, and we ask for funds to fund scientific research, and we have been trying to get things organized. And for example, there was a first call for tender for scientific research with the participation of some of the researchers who are present here today. And this call for tender was launched in 2018 when Parent Project reached the maximum amount for ever. We exceeded 500,000 euro for scientific and clinical research projects. And for us, this is a reason of pride. We want to grow, we need teamwork, and we work on the team because it is very important to find the right balance within the team. It's not easy to work as a team. We all have our own professional activities, and sometimes it is difficult to find the right balance. The real issue is being able to come to terms with other people. If I am unable to do that, I'm good for nothing. A team that really works well is a team that will reach ambitious objectives. And I close by saying it is that the first team is made up by families with our contribution. Even a phone call just to ask how are we getting on, or what can be done, or sometimes even brainstorming. Things are then filtered, and if they can be developed, of course we are not present everywhere, but we can easily reach all different areas. Well, for us, this is something we are supposed to do. Being members of Parent Project asking to be enrolled, feeling you are members also legally, and also paying the annual fee is not so much the economic value, but the symbolic value of belonging to the association, being present in the assemblies, participating, voting. Participate and become members, and also convincing other people, other DMD families to become members. Thank you. We had this time and it was useful to give some information.
E allora adesso siamo arrivati al momento.
So he has varied us right to the moment of the documentary, given the important role played by women and mothers and families in general who received the diagnosis of MDND and BMD. Because of their value, we decided to create an international documentary. With this documentary, Parent Project wishes to stress how certain families of this community in the world have implemented the spirit of resilience in their life. These families had the strength to transform their personal pain to improve the life of their children, encourage them to fully tap into their potential above and beyond the constraints of the illness. Most of them are part of a broader network that has improved the scenario of the entire community, from Italy to Spain, from Israel to Romania and the Netherlands. Parent Project features stories of parents and children with the goal of describing the challenges that they face on a daily basis, but also the strength and the ability to find the hope that makes less burdensome living with this disease. The new life of these persons is full of joy and fulfillment and can inspire all of us by contributing to a process of social change. Enjoy the documentary.
So, in 1984, a long time ago, two of my boys, my only two sons, were diagnosed with Duchenne muscular dystrophy. So at that moment, you feel quite alone and really unknowing about what happens next or what you should do. So I reached out to physicians, and most of them had never even heard of Duchenne, or if they'd heard of Duchenne, it was very briefly in their medical school career.
La distrofia muscolare di Duchenne è la più complessa tra le patologie genetiche.
DMD is the most complex. It's present from conception and the birth of children with DMD, and it compromises muscle regeneration. We all use muscles, whatever we do. Children with dystrophy don't.
That out, and it took some time because people, it was hard to find and there wasn't much research. And so I felt like what was missing in this whole environment is the patient voice, right? Because parents, and certainly boys with Duchenne or individuals with Duchenne, have to have a voice in here because it's about their lives and about the quality of their lives. So I felt that was really missing.
Io credo che come tutti i genitori che ricevono una diagnosi di Duchenne per il loro figlio, la prima cosa che si cerca di fare è di cercare informazioni, e quindi Parent Project in Italia è nato per questo.
It was really hard to find out because, first of all, way back then, which seems like the old age, right? We didn't have social media, we didn't have Facebook, we didn't have the ability to connect to other families.
Nel corso di questa ricerca entrai in contatto con alcuni genitori americani, in particolare con Pat, e allora decisi di aderire a questo movimento internazionale che stava nascendo, che si chiamava Parent Project.
So I took a loan out at the bank, and then I ordered all the publications around Duchenne to understand what we knew about Duchenne. And then I made appointments at different laboratories and different clinical offices and pretended to be a doctor and asked them questions about what do we know about Duchenne.
Quindi in quel periodo la cosa più difficile è stata proprio esattamente capire cosa fare e come farlo, quindi cosa spiegare alle famiglie, come insegnare alle famiglie ad affrontare questa malattia da un punto di vista clinico e soprattutto anche da un punto di vista psicologico.
So I think the fundamental issue is that this child is the same beautiful child and that their path is going to be far different than anyone predicts because it's unknown but full of promise.
I'm Elizabeth and I'm the mother of Justus. Justus is now almost 28, 27 and something. And I live in Amsterdam, or we live in Amsterdam. Justus is my son with muscular dystrophy, which we know for a long time already. And meanwhile, he's just doing whatever he likes to do, and I'm also doing what I like to do.
I'm Laura and I live with my husband Philip and our wonderful two boys. Louis is 7 years old and Jensen is 5 years old. We live in a little small village, Lancashire. It's very, very small.
Mi familia está compuesta por 3 personas, que son Isaac, que es el papá de Alonso, Alonso.
My family is made up by 3 people, and Isaac, who's also the father of Alonso, and myself. I'm the mom.
Suntem o familie normală, de oameni obișnuiți, avem 25 de ani de căsătorie și suntem 2 oameni care au avut...
I'm Tali Kaplan, I'm married, I have 3 children. The youngest is 10, Shira is 15 and a half, and Hori is 14.
De ani, Petru, care este centrul atenției și preocupărilor noastre.
אני טלי קפלן, אני נשואה.
I'm Tali Kaplan, I'm married, I have 3 children. Ido, the youngest, is 10, Shira is 15 and a half, and Hori is 14.
Alonso nacía por...
Alonso didn't want to stand up, and especially when he had to sit down, it took him a long time. And when he did try to sit, I had to put cushions behind him so he wouldn't fall because he lost his balance.
And we all got into the room and he got Louis pressing and running, and he just came out with the words where he's got muscular dystrophy Duchenne. And from that moment on, I don't think I heard any other words.
First thing I thought was, but we have time. No, he had a twin sister and she died after 4 days. Then I thought with Duchenne, you know, it's a shorter life, but it's still, we have 20 years ahead of us. So we're going to make the best out of it. So I remember when we drove back from the hospital, I said to him like, we have time, you know, we have time. We're going to make the best out of it.
El caso es que yo pensé que
I thought that for months. I did not want to relate with anybody that had DMD. I didn't want to talk to anyone at any associations, but then when I went through those phases, they elicited an amazing strength in me. That's what happened to me.
אבל מצד שני זה משנה את כל החשיבה.
There's still a lot of time to plan. On the other hand, everything changes, and you understand that your goal is to live and to gain experience.
Și din acel moment, de la vârsta lui de 6 ani,
From that moment onwards, when Peter was 6 years old, I decided to become the best parent possible. The best parent possible that invests time, that pays attention, and that is really well balanced. Very well balanced.
Diagnosed, I thought like, can I still laugh, you know, and have fun when your child has such a diagnosis? But then I realized that if he would have Duchenne and a depressed mom, that he would have a double problem.
Yo creo que feliz. Yo creo que él es.
I think that he's happy. He's got friends at school. He's got friends here in the neighborhood. He loves playing football. I think he's happy.
Boy, he's a character. He's always smiling. We have fun. We have fun.
Y lo que cambió sobre todo es nuestra forma de ver.
What really changed is our way of looking at life. We used to think things were important and that were real problems for us, but now we attach less value or importance to them. Everything becomes secondary after the diagnosis. All you do is think about the day-to-day living of your family.
Petru
is very strong and positive. And for me, he's like a ray of sunshine.
Este capabil să
He has the ability to understand deep things immediately at first glance. And he's very careful about the suffering of other people.
Young, I realized a few things. One is that he should be resilient. You know, if a child is afraid for everything and goes home or stays at home, that would not be good for his future. Second is that the way you're treated is also 50% is yourself. Justus just graduated from university, and he always, for him, optimistic, hope keeps you alive.
Un ringraziamento particolare va alle aziende che ci hanno sostenuto.
We wish to thank all the companies who supported us. And now we call the people who have enabled us to prepare this film. Silvia?
Isa. Elisabeth.
Elizabeth. Elisa? Pat? Ali? And Filippo.
E adesso chiediamo loro se ci vogliono dire qualcosa.
It was a real pleasure to take part in this documentary. We will overcome Duchenne with information, research, and closeness to the families. I wish to thank Parent Project Italy. We'll go on fighting together.
She said it was a real pleasure to participate in the documentary film. All together we will overcome Duchenne through information, research, and by being close to families. Thank you to Parent Project. We will go on fighting together.
I would like to thank Parent Project Italy. I think it was a very great experience, and I was happy to share our experience, and I think that we have a great community and we have a lot of things to look for and to live today but look ahead. So thank you very much.
I would like to join the others in really big thank you to Parent Project Italy, but also to the team like Fabiana, where are you, Marco, you were there. Everybody who was there worked so hard. I must say it was really fun to do it. So thank you. And I think indeed by exchanging examples, how we live, we see each other in conferences, but really seeing all of us in daily life is really nice. Also for me, it was nice to see your son, to see your helper dog of your son and everybody. Thank you so much. Thank you.
I was really honored to be part of this project. And because it's the first time I am able to speak in front of you, Parent Project Italy, I just want to say thank you, everyone, for including us in all your conferences and making us part of this journey and this fight. And because of you were so generous with Romania and our team, we were able to build a community in our country, and we were able to build services in our country. And we owe you, all of you, and Filippo and Pat, what we have done in our country. Thank you so much.
So when my boys were diagnosed, which was a very long time ago, as we traveled in this journey, someone said to me that this is a family disease. So I'm really grateful for your family, this family of ours, because I think together we can change the world, and we have already. We'll continue to do so. I'm so proud to be part of Duchenne Parent Project Italy and Duchenne Parent Project Netherlands and Romania and Israel. We've built a world community, and I think my sons are smiling, and I know that we can do this together. So thank you for allowing me to be a part of it.
I wish to thank Parent Project Italy because they did an excellent job, a real present to all of us. Of course, I'm honored I am the only man among so many women, which probably means something. I will start wondering what it means exactly. But it is a great honor for me because in these things women are more present, even though there are many fathers who are working hard. And this is a part of Parent Project. I mean, sharing difficulties of this life all together and also expressing them in our community to talk about them and solve about them together. I wish to thank Pat, Elizabeth, and all those who help us in the international community, as well as our staff that has become extraordinary, and our board, our executive board, that has truly grown together with the whole of our association. Congratulations.
Grazie a tutti.
Thank you, everybody. And now we go on with our session.
Proseguiamo con la sessione 5. E quindi...
So now we'll move on to session 5. So all chair persons are kindly requested to come onto the podium. Stefano Mazzariol, our vice president, and Pier Lorenzo Puri, who needs no introduction within this framework. So this session is devoted to the patients' support to the work in the labs. Is Professor Puri around?
Bene, non è facilissimo riprendersi dopo l'emozione.
It's not very easy to resume a session after watching that video, but actually we are doing a lot at a world level, and indeed this also includes our support to research. Actually, as you may know, one of the first activities of our association has precisely been the support we provide research. One of the aims being finding possible strategies that may turn into potential therapies for our kids. We've also given our support to that clinical research that might improve and possibly even widen, if you wish, the management of DMD kids. Well, a lot has been done in these past 20 years and more. We've funded several projects, and the research has a great, of course, representative here, Professor Puri, who again needs no introduction. We've been supporting clinical research in Italy. I'm thinking of the Italian patient registry, And we've also used some important professional figures within our staff, our biologists, in order to interact with all those working in the field of research. And of course, we've been engaged in other similar activities whereby our association is now widely known as a valuable interlocutor for the scientific community. Of course, we know that there are several steps that must be made for therapy to be one, actually, which, by the way, includes the various monitoring time that takes place within labs. We know about the very strict rules behind all of this, not to mention the investment, the large investment. We are talking here of millions of euros. And ours is a family association. Of course, we may be very well organized. We may benefit from the presence of many volunteers working on our site. However, we can hardly meet that kind of goal. So we have basically responded to the proposals that we got from the researchers in order to check whether their hypotheses were well grounded, hopefully leading to some improvement which might indeed stir up the interest of larger investors. And that, for example, was the case precisely of Professor Puri's research group. There was just a hypothesis that we supported as parent project, and this has led to some improvement in dealing with both DMD and BMD. So, for many years, we've been considering several projects that we were suggested by single researchers. However, now things have changed, and we have now launched the first tender, if you wish, for a parent project funded research project. Well, for a total of 500,000 euros. Now, this is the amount of the bid, if you wish. So, 80,000 euros for 2 years. Deadline was last September the 30th. 12 projects we have got so far 12 projects. We held a very long meeting with several people from our staff, including Filippo, during which we adopted the projects in scientific terms, but also in terms of the priority they could stand for from our standpoint, which is a patients' association. So 6 were moved to a short list that was then submitted to some scientists, some researchers. So thanks to our evaluation and the evaluation of some external auditors, if you wish, we decided to fund 3 out of the 6 projects, and you'll hear about them soon through some video interviews. So, as I said, we paid attention to the scientific side of the projects, but also the methods that the projects orbited around, since we are all very well aware of the need to pave the way to as many hypotheses as possible, of course with the aim of hopefully getting out of such an intricate forest that DMD is for us. The second tender, with no time deadline, regards towards projects amounting to maximum 20,000 euros. It's a fast-track kind of an approach, since there is an external approval that, however, goes hand in hand with an approval from, inside the parent project. 3 projects have been selected. 3 more are still being evaluated. Let me now introduce you to someone who is a friend to us, a friend for Parent Project, Professor Puri. We have been working together for several years indeed. I believe he can truly tell us about the real relevance of basic research and associations like ours in supporting such research. Thank you.
Well, it is the first time I speak of something that is not science, rather what is behind what we do. Usually I show data, and that is far simpler. And I realize it now, because you just have to say what a lab has produced, whereas today I want to tell you why it is important to receive funds. Funds coming... No, it's okay, we can hear you. Well, the importance of the funds, but also where these funds come from. Well, Parent Project really changed my life, and I say it very seriously. In some ways, it spoiled my life. I started doing research as a doctor, and my idea was solving a biomedical problem. But I would never have imagined that research could become part and parcel of my life to this extent, so much so that I took some decisions that really surprised me. Why is it so? Because, when I started doing research, I was a young researcher, full of ideals, but I'd never considered the burden of research. I thought it was just going into a lab and express myself. I went into the lab, I had an idea, and that is the beautiful thing about research. You have an idea and you check the validity of that idea. This is the most beautiful thing that can happen to a student. A student usually has more ideas than information. But undoubtedly, after a few years, research becomes an end in itself, or rather a vehicle, or a tool to reach some chair, and you start thinking of publications, and research becomes meaningless. I met Parent Project in 2002, and I believe I never came, or rather we both missed the place where we were supposed to meet. And then I met him, and that's when he spoiled my life, because when you start carrying out research, facing a huge problem, and then you start sharing this problem, and you gradually become the person that you would like someone else to do if you had a son with that problem yourself. And that is a huge responsibility. if you see it that way. So you start devising projects that are meant for people, not so much for your academic life. It's no longer a game. Of course, there is the good part of being in a lab, but you no longer see in the lab the experiment as such. And when the experiment doesn't work, I mean, you don't have only the frustration of an experiment that did not work, but there is also the pain of not having reached the target you meant to reach. Of course, this continuously generate energy. To carry out research, you need a lot of energy. You're always unhappy. But not so much because it is the research that makes you unhappy, but because you become increasingly demanding. Parent Project took me by the hand in 2002, and I would never have invested time and energy to such an extent to move from a biochemical experiment, bioacetylase inhibitors. I started working on that in 1999. It is something that has to do with biochemistry. I worked on enzymes that are called acetyltransferase, and I discovered there were enzymes doing the opposite. And I started thinking whether that could have some effect in guiding what we saw in vitro, i.e. the differentiation of the muscle fiber. Of course, behind that experiment, there was also the prospect of treating children who had problems. But until you know the parents and see the children, when you enter a community and where you feel the burden of what's behind all that, you will never be able to develop this type of research. For 17 years, actually. This is the personal aspect, but there is another one. Receiving funds from Parent Project meant giving also an identity to my research group. And that was possibly the most important aspect, because you can do nothing in isolation. All we did, from the first biochemical evidence up to the progression. I mean, when we reach the clinical trial with the acetylase inhibitor, that was done. They are all there in the back. I mean, they are half asleep. They are those people laughing there in the back, but they are there. And there are also people who were in my lab. Well, there is a sort of path in my lab, a route, so to speak. And almost all those who have worked in my group, either they stay with me, or if they go away, they keep working on DMD. It's not my own merit. It is Parent Project. You get bound to people, to faces. I mean, when you know that you are trying to solve a problem, this huge problem of a child, of a family, well, you stay there till 2 or 3 in the morning. It is no longer a problem. Motivation is everything. I think that can balance off the quantity of money you receive. I've had the opportunity to compare the rare cases where we obtained free funds, so to speak, funds that were not itemized, so to speak, and we had received a lot of money. In many cases, we squandered this money. When you have very little money that are directed to one mission, are usually well spent. And this is something that agencies giving out funds for research should learn, that the best way to give money for research is well, not so much itemized it to a project, but rather to a human mission, because researchers are men, they are human beings. And when you are involved in that mission, you can give 200, 300% of yourself. You no longer feel tired. You want to do something. You want to do that for people, faces, and then you meet these people every year. It is a sort of flywheel. It gives energy to a lab, to a group of researchers, generations that go through that lab. I'm very glad to talk about it because I've never talked about it in this way. I haven't even prepared my speech. I was told to prepare my speech, but possibly I said something different from what I wrote. I really wanted to express what I feel now. I didn't want to get a speech ready beforehand. This is the most beautiful thing that happened to me from the professional viewpoint, binding myself to this experience, to these emotions. Every time we have discovered something that we had sort of imagined might have had an impact on patients, there was always the risk that we might shed some tears. Well, when an experiment is successful, is wonderful. First of all, because it almost never happens. So when it succeeds, you think, oh, great, for once. And then you understand the evolution of that experiment and change the life of people. Then you become a father and a mother. Of course, you understand these things even better. And it is an enormous joy. I cannot find words to explain that. Well, maybe you should come to our lab where we work. It is a beautiful experience. We don't earn a lot of money, but it is one of those things where you can say, well, money is not everything. I'm doing something that goes beyond. I'm not speaking about myself, but that is something that brought together the people who are sitting there in the back. I've never thought that there is a Puri group or a special lab. We are just people who have decided to declare a war to Parent Project. And I see this young man who are 22 or 23 who comes to us. After a few months, they are so integrated. I've never asked anyone to do something extra. They volunteer. Well, Friday night, they work till 10 p.m. And this is one of the most beautiful professional satisfaction you can have. Today we are supposed to talk about those who did this research projects. One name is the one I think I guessed. Whoever gets this money, well, make use of this money with this in mind. Use them knowing that no matter what, you're trying to do something for parents and children all over the world. And I don't want to dramatize, but my job. If you are here because you hope we'll be giving you news. So apart from the responsibility, there is the joy you get when you can do something for someone else. And the social impact, in a world where everything is aimed at money making, everything is quantified. I mean, how much are you getting, what capital I invested. There are there in my lab you see young people who work without asking questions whether this work will help them in their career. They work because there is a problem, because I want to solve that problem, not because they are paying me. And this is not only a noble motivation, but is a motivation that fills our life as researchers. It is true that seeing patients, of course, spoils your life, but that is a minimal part. In fact, it filled our life, and I think I speak on behalf of all of them. We have our life filled with something beautiful, and the hope we can help some people to solve a problem for which they're certainly not guilty at all. At present, it is a giant that we simply cannot kill, but that gradually we see it is crumbling down, and that is something we have been witnessing over the last 20 years. Muscular dystrophy looks like a giant falling to pieces. Well, that's it.
Beh, io direi che con il supporto di persone.
Well, with the help of this kind of people, this is a cause that is certainly on our back, but they are on our side, trying to help us out of our problem. But it's nice to learn that it's their problem too. They carry it on their back too. So supporting lab research can have some perspective. It is something that may entail, say, €1, €2 after all. And of course we need fundraising campaigns to that respect, which is, by the way, where the money actually comes from. But this all amounts to the money that we give them, with the aim of hopefully finding something for us together with us. And of course, we need to show that the initial idea is the right one. In the case of HDAC, this is exactly what has happened. So we basically provide the final researchers with a good idea that they must simply try and implement, if you wish. For us, parents, as I said, DMD is like a forest, and the researchers are in this forest with us. They don't know how to get out. Well, in the middle of the road, maybe, but I like the forest as a metaphor. The point is that if we all go towards the same direction to find the way out, we might get it all wrong. Maybe if, on the other hand, we move in different directions, some of us may find the right way out. And who is the one that puts all these researchers moving to different directions together? Well, it's us, the parents, the families. We want research to always try and find new ways, hopefully finding the good ones. So this is what is behind the project that you're going to hear about in a minute. Let's try to find the right ways, and then everybody should know once we have found the right one. There's something else, by the way, I might add. When you are in research, in basic research, and you get to the preclinical or clinical stage, one usually, changes direction because then somebody else takes over. Well, I have always found Filippo once I got to that stage, the preclinical or clinical stage. Parent Project has been a sort of a guiding light for me, also in logistic terms, in organizational terms. A group of people that may help you find a way out of the forest, taking you by the hand. Basic researchers usually keep working in the field of basic research, unless, of course, there's a much higher, if you wish, motivation to lead them into unknown roads. Filippo may not be doing fine right now, but he's never said no. He's never given up.
Filippo, tutto Parent Project, Maria.
So this is about Filippo, but actually about Parent Project as a whole. We, as parents, thank researchers for what they do, and researchers thank us parents for what we do, which certainly speaks for such a high degree of synergy that must go on because it's the only way to win. Thank you, Professor Puri, thanks. And now let's start with the video about the projects that will be funded by us. Thank you.
Sono la dottoressa Elisabetta Ferraro e lavoro presso l'Ospedale Maggiore della Carità di Novara e presso l'Università di Torino come professore a contratto. Studio il muscolo scheletrico da circa 8 anni ed è la prima volta che mi occupo di distrofia muscolare di Duchenne. Il progetto da me proposto inizierà a marzo e il finanziamento durerà per 1 anno, ma gli esperimenti si protrarranno per circa 18 mesi. Tramite questo progetto intendiamo agire e ridurre l'infiammazione che caratterizza i muscoli dei pazienti affetti da distrofia muscolare di Duchenne. In particolare utilizzeremo dei farmaci che agiscono sul metabolismo e tramite questi farmaci ci proponiamo di modulare e direzionare il metabolismo dei macrofagi infiltranti in modo tale da indurre un loro differenziamento, una loro maturazione in senso pro-rigenerativo a scapito delle forme pro-infiammatorie. L'uso di questi farmaci nella distrofia muscolare di Duchenne è innovativo. È un enorme vantaggio che hanno i modulatori del metabolismo è quello di essere stati approvati per l'uso nell'uomo, in quanto hanno effetti benefici in alcune patologie cardiache, il che li rende estremamente interessanti per un potenziale uso terapeutico rapido ed efficace nella distrofia muscolare di Duchenne.
Sono Graziella Messina, professore associato all'Università degli Studi di Milano e lavoro su distrofia muscolare Duchenne da 10 anni. Ho recentemente ottenuto un finanziamento dal Duchenne Parent Project che inizierà a febbraio di quest'anno e durerà 2 anni. Qual è l'obiettivo di questo studio? È quello di validare l'efficacia terapeutica di un farmaco che già esiste in clinica per un'altra patologia, tumori, anche tumori pediatrici, nella distrofia muscolare di Duchenne. Qual è il razionale di questo studio? Abbiamo dimostrato recentemente, negli ultimi 6 anni, che l'assenza di una proteina che si chiama NF1X in animali distrofici porta a un miglioramento morfologico e funzionale del fenotipo distrofico, conseguente a una ridotta rigenerazione del muscolo scheletrico che abbiamo dimostrato essere in realtà un beneficio in un contesto distrofico. L'idea quindi futura qual è? Quella di inibire farmacologicamente questa proteina NF1X. Come fare? Abbiamo identificato recentemente qual è la via di segnalazione che attiva questa proteina ed è una chinasi. Queste chinasi sono per fortuna utilizzate e studiate molto in cancer e un inibitore di queste chinasi è già clinical approved sia dall'FDA che dall'EMA. Quindi la nostra idea sostanzialmente è quella di fare un drug repositioning per validare e verificare l'efficacia terapeutica di questo farmaco in Duchenne, utilizzando diversi modelli animali distrofici. L'impatto per nella comunità distrofica, quale può essere per la salute dei nostri pazienti distrofici, è quella di preservare e migliorare la morfologia e la funzionalità del muscolo. Non curiamo il difetto genetico alla base della patologia, ma preserviamo il muscolo, e questo significa una migliore qualità della vita dell'individuo e rendere il paziente stesso ancora eligible per diversi approcci più risolutivi della patologia perché possa rispondere anche meglio a qualsiasi approccio terapeutico, che sia di gene therapy che sia o di cell therapy.
Salve a tutti, sono Silvia Consalvi e lavoro presso la Fondazione Santa Lucia di Roma. Mi occupo di distrofia muscolare di Duchenne da oltre 10 anni. In particolare i miei studi nel team di ricerca del dottor Pier Lorenzo Puri hanno contribuito ad elucidare il potenziale terapeutico dei farmaci inibitori dell'estone di acetilasi, quale il Givinostat, per il trattamento della DMD, supportando quindi la loro attuale traslazione in clinical trials. Nonostante i risultati promettenti del Givinostat, siamo consapevoli che l'efficacia terapeutica di questo farmaco è limitata a stadi precoci della DMD, in quanto la progressione della malattia determina l'evolversi di un processo di invecchiamento cellulare che rende il tessuto muscolare gradualmente resistente al trattamento con Givinostat. Il mio progetto di ricerca, finanziato da Parent Project Onlus, ha l'obiettivo di sperimentare una nuova terapia farmacologica che possa contrastare la progressiva resistenza al trattamento con Givinostat e quindi estendere la sua finestra temporale d'azione a quei pazienti affetti da DMD a stadi avanzati. Il progetto partirà nell'immediato ed è previsto concludersi nei prossimi 2 anni. Attualmente, partendo dai nostri studi epigenetici, che sono l'oggetto del dottorato di ricerca del dottor Luca Tucciarone, finanziato anch'egli da Parent Project Onlus, abbiamo identificato la proteina sirtuina-1 come target potenziale di una nuova terapia farmacologica che potrebbe sinergizzare nonché estendere temporalmente l'efficacia funzionale del Givinostat. Questo progetto rappresenta quindi il primo tentativo di combinare il trattamento Givinostat con altri farmaci, al fine di offrire una prospettiva di trattamento all'attuale coorte di pazienti affetti da DMD a stadi avanzati.
Buongiorno, mi chiamo Giulio Cossu e sono professore di medicina rigenerativa all'Università di Manchester. Da più di 20 anni lavoro sulla distrofia muscolare col tentativo di sviluppare terapie cellulari che possano essere efficaci in fase clinica. Quello che si evince dall'ormai pluriennale esperienza di tanti centri di ricerca è che ci sono molte sperimentazioni cliniche in atto, ma ancora nessuna ha raggiunto un pieno successo clinico. Uno dei problemi maggiori delle sperimentazioni cliniche è che costano tanto, prendono tanto tempo e permettono di verificare una sola variabile alla volta, e questo ovviamente moltiplica tempi e costi. Il progetto che noi abbiamo ideato e che è stato finanziato dal Parent Project si basa su un'osservazione casuale che abbiamo fatto, in cui innervando, cioè coltivando insieme delle fibre muscolari normali o distrofiche, insieme a motoneuroni che provengono da topolini allo stato embrionale, si formano delle sinapsi, cioè delle giunzioni in cui il nervo si congiunge e regola la funzione motoria della fibra muscolare, immature, incomplete. Come conseguenza queste giunzioni mandano continuamente dei segnali e le fibre muscolari continuano a contrarsi indefinitamente. Quello che abbiamo notato è che in queste condizioni le fibre normali continuano a contrarsi per giorni, mentre le fibre distrofiche muoiono entro meno di 24 ore. Questo ci permette di sviluppare un saggio molto semplice in cui noi possiamo verificare se un farmaco, un vettore virale, cellule normali o geneticamente corrette possono in qualche modo migliorare la capacità di resistere allo stress indotto da contrazione della fibra muscolare e naturalmente questo porterà le fibre distrofiche a sopravvivere. E questo è quanto ci proponiamo di fare. Se il progetto, come speriamo, sarà coronato di successo, potrà offrire a tutti un mezzo molto rapido e semplice per un primo screening di molecole, di sostanze, di qualsiasi strategia terapeutica a un costo risibile e che può essere eseguito nel giro di pochissime settimane.
Sono Gabriele Siciliano, professore universitario presso la clinica neurologica dell'Università di Pisa e da vari anni mi occupo di malattie muscolari sia dal punto di vista clinico che dal punto di vista di quelli che sono i meccanismi patogenetici che stanno dietro la malattia muscolare. L'importanza di questo progetto, ricordo è un progetto che va a studiare nella distrofia muscolare di Becker quali sono le caratteristiche cliniche che differenziano in questa malattia le manifestazioni del disturbo muscolare. Ricordiamo che la distrofia muscolare di Becker, pur essendo al pari della malattia di Duchenne una distrofinopatia, si manifesta con una eterogeneità clinica molto elevata, molto variabile. Il nostro progetto ha lo scopo appunto di caratterizzare la storia naturale di malattia, individuare quali possono essere le differenze tra paziente a paziente in manifestazioni non solo muscolari ma anche per esempio per quanto riguarda l'interessamento cardiaco, che è uno degli altri elementi importantissimi nella prognosi della distrofia muscolare di Becker. È una malattia forse meno studiata rispetto alla distrofia di Duchenne, ma non per questo non altrettanto utile da caratterizzare nella conoscenza e nei dettagli. Questo ci permetterà in prospettiva di conoscere quali sono le differenti tipologie con cui la malattia si presenta e anche per avere la possibilità di uniformare i trials terapeutici che oggi si affacciano nella malattia. Questo è lo scopo del progetto che Duchenne Pare Project ci ha finanziato. Grazie.
Ringrazio Parent Project dell'opportunità di portare avanti questo progetto sullo studio della progressione e rimodellamento della cardiomiopatia dilatativa nella distrofia muscolare di Duchenne e di Becker. Io sono Rachele Adorisio, lavoro all'ospedale Bambino Gesù, sono una cardiologa e dal 2008 seguo i pazienti con cardiomiopatia dilatativa nelle forme di distrofia muscolare. In questo studio noi andremo a valutare gli effetti del rimodellamento dei meccanismi dell'atrofia cardiaca che sono alla base dello sviluppo della cardiomiopatia dilatativa. Ad oggi questa cardiomiopatia dilatativa decorre in modo quasi del tutto asintomatico nei pazienti e pochi sono i marker che noi possiamo utilizzare per valutare l'effetto precoce di farmaci oppure per poter identificare quali sono veramente i pazienti a rischio di poter avere un evento acuto nel corso del loro follow-up. Questo studio per la prima volta cerca di integrare delle informazioni sia di carattere muscolare, andando a studiare l'atrofia cardiaca, sia di identificare gli effetti dei farmaci, degli acidi inibitori che vengono utilizzati in termini di prevenzione, e sia attraverso un prelievo del sangue di identificare tutti i metaboliti che sono alla base del metabolismo del cuore, che può essere alterato proprio quando la patologia progredisce oppure in una fase acuta. Questo studio ci permetterà di identificare in modo più precoce tutti i fattori che portano allo scompenso cardiaco, ad individualizzare i trattamenti e a poter in questo modo precocizzare diagnosi e terapia. Grazie.
Bene, come vedete abbiamo esplorato tante strade, ricerche.
We have actually gone different ways. Basic research projects aimed at finding new therapeutical strategies, but also aimed at improving the knowledge or better management of our kids. And there is also another project that is specifically devoted to BMD, which I think it is a high time we should do, since of course BMD starts showing all its problems now. Okay, having said that, let me now ask Professor Racca from the Hospital of Alessandria to come to the podium to talk about another project that is running fast. The name of this project is Emergency Card.
Grazie a Parent Project per avermi invitato.
I wish to thank Parent Project for inviting me here. I'll try to be brief and tell you about this project that, as you can see, has been shared by many. We have all tried to make up a card that parents should wear or carry with them in case of emergency needs. We know that prognosis and quality of life of DMD patients have both remarkably improved thanks to cortisone or an IV, but also, cardioprotective drugs. However, there is still quite a significant early mortality rate for acute events, mainly respiratory failure or acute heart failure. So this study is particularly interesting insofar as it orbits around patients hospitalized in a French critical care unit, an ICU rather, and they had some neuromuscular diseases. They certainly showed respiratory failure, but mortality rate was relatively lower than the one we normally get from similar cases treated in ICUs. Anyway, we have seen that chronic neuromuscular there was still a low rate of muscular dystrophy, muscular disease patients. Another piece of data that came out of this study was that in order to have good results, some, of course, specialized knowledge was needed to best cope with these patients. Now, in these past few years, there's been quite a lot of reviews of the literature, but in the recent Lancet recommendations, one issue is particularly highlighted, namely that patients might be referred to an emergency unit where the physicians or nurses may not be fully prepared to welcome them in medical terms. Hence, one of the Lancet recommendations was precisely to allow families and patients to have a sort of a pocket guide, including some information for physicians and nurses. And Lancet also provided with some suggestions, some relevant suggestions. For example, the patient may not want to be resuscitated with, I don't know, tracheostomy, or maybe they'd better be treated with some relevant respiratory devices, or else it's important to know whether the patient uses a cough assistance device, and physicians should be made aware of this for what pertains to cardiac problems and to respiratory failure. So, I don't know, adrenal failure may be avoided in case patients are under chronic cortisone therapies, or else anesthesia may sometimes be necessary, but in that case one must know how to cope with it if the patient is a DMD patient. So, some colleagues have worked with me on these Lancet recommendations, thus producing an emergency card including the most relevant information for physicians, but also the most relevant clinical data pertaining to the single patient, of course, in case of emergency. Now, this is a tool that will soon be available. If you visit the Parent Project website, you'll find a Duchenne emergency card window. If you click there, you'll find a home page showing the authors, the name of the authors of the recommendations, and then what the tool is about and who it is for. The emergency card also includes two parts. Part one is a general part about all DMD patients in the different stages of the disease, and then there's a more customized part where the most relevant information about that single patient is provided. This part 2, is the one to be updated on a yearly basis because, of course, the single patient's conditions vary in time. Now, if we then click on the general part, we'll get the first of these two pages, the general part, indeed, which includes information for non-DMD experienced physicians about a DMD patient's respiratory problems rather than, heart-related problems or osteoporosis or the use of cortisone. Page 2, instead, includes some suggestions about how to cope with the single problems. If there's an acute respiratory event, what to do and what not to do. The same goes for heart failure or how to prevent the stress-related adrenal failure, how to cope with anesthesia in case it is needed, or dysphagia, which might lead to some choking time. So this is all summarized in a bunch of really few pieces of information in a very, again, fast, quick way. It's a pocket guide, actually. Otherwise, if the physician has more time to go deeper into the relevant topics, well, then there's a link to references to be downloaded, including Nikolov protocol regarding acute adrenal failure. You can just click on that and you'll find the protocol itself as well as the article used as a reference. Now, the emergency card shall be visible on the website, but also you can download it in a PDF version. And of course, you can also read the customized part. You click, of course, the personal on the personal part, and then, of course, you'll have to enter your password. Further to which you'll have in 2 pages the single patient's relevant information. Don't be afraid here, because you may find it's a bit crowded, but actually, if you answer no to one of these items, then the line simply vanishes. We have also included the phone number of the counseling center where the patient goes on a regular basis, but also information about allergies or DMD-related complications, say heart failure or cardiac complications, and so on and so forth. The drugs that he is on. This is all useful information for an emergency doctor, because of course, if the patient is on cortisone, then we might need some hydrocortisone, or if he is on ACE inhibitors, we might think of a possible hypotension. Therefore, beta blockers might help or not help. On page 2, we have non-drug therapies such as an IV or a cough device or nutritional devices, and then there's some room for other diseases that have nothing to do with DMD. I mean, other diseases may be present too, whereby the person is on other treatments. And then something useful for resuscitation, namely the predicted vital capacity at the last examination, ejection fraction of the heart, and finger saturation, either under ventilation or not. Well, these data might not sound that useful, but within a resuscitation framework, they are indeed very relevant. And what about intubation? Of course, if there's a respiratory failure, intubation might it might be useful to know whether intubation is possible or not. And then on the left-hand side, in the bottom, some limits to therapies. For example, no tracheostomy at all, because that is the will of the patient. So the personal side of the emergency card, as I said, shall be updated on a yearly basis. An English text will probably be produced too. Maybe the patient is abroad on holiday, so an English version might be handy. And then what about the age? I mean, at what age should a DMD patient carry this emergency card? Well, there's no age, in my opinion, because indeed recommendations for anesthesia are useful since diagnosis. Cortisone, this is also important, hence the relevance of Nikolov's protocol, and of course the relevance of orthopedic complications. Knowledge, these patients may, have related problems. And then, of course, heart diseases after they have reached the age of 10, and respiratory complications after they have lost their ambulatory ability. Now, as for timing, well, within 1 month, the general part will be uploaded in the website. Within 1 month, you'll be able to download that part. In the next few months, we believe that the first 20 pilot patients will fill in the personal side that will also go together with the English version, and then the parent project registry will be connected to the emergency card so as to update it on a yearly basis. And after that, say within the end of this year, the first 150 patients will be included with the, regarding their only personal part, and so on and so forth. Thank you very much.
Grazie, Professor Racca.
Thank you, Professor Racca. I believe this is a very important project, a very important evolution for our peace of mind, so to speak. Since we have a few minutes, is there any question for Professor Racca, or we can adjourn? I think we can adjourn. Just an announcement. Well, we have to get out of the room, so don't leave anything here, because they must prepare the room for our evening meal. So don't leave anything behind. We must clear the place, and we'll see you tonight. Thank you, and see you for dinner.